So today we will have also great presentations, and I invite you to enjoy these great presentations and also make a lot of questions. So now we will start with a great lecture with Dr. Prashant Sharma. Please be very welcome.
Dr. Prashant Kumar Sharma is a professor in the Department of Biomedical Engineering at the University Medical Center, Groningen, University of Groningen, and has 22 years of teaching and research experience. He obtained his PhD degree in surface chemistry of bacterial addition and bioprocessing of minerals from Luleå University of Technology in Sweden in 1997. His research group primarily focuses on biomaterials and biotribology. He developed biomaterials that synergistically function with the lubrication systems of the human body when damaged, aiming to maintain the hydration and lubrication of damaged surfaces. He also focuses on microbial addition, biofilm formation, and infection control. The specific pathologies of interest include dry mouth disease, ocular tribology, dry eye, cartilage tribology, and osteoarthritis. Please welcome Dr. Prashant.
Thank you very much for a kind introduction, and of course the invitation which was given to us, to our department, to come here and talk about what we are doing. One small correction in the introduction: I do everything what your colleague said, but I'm not a professor yet. I'm associate professor. Okay, so you can just call me Dr. Sharma, or Prashant is fine too. Okay, so let me come then to the topic of today. This topic is different than what I told you about on Monday. So that is one kind of research I do, which is on tribology, friction wear, lubrication, and so on. But this is also another aspect of my research, and you can see it is viscoelasticity and cell response. So viscoelasticity, maybe some of you could guess it, it is more biomechanics, how this affects cell response. Just checking. Okay. So, so in general, tissue engineering, the aim of tissue engineering is to get a functional tissue at the end of the day. And here I show you a small schematic which is not comprehensive. This is a very simplistic view of tissue engineering, where what we are doing, we have a certain type of cells. We are adding them into a scaffold, which is made of a biomaterial, and then we wait and see what happens, because the cells are the workhorses, and they do things, and ultimately we get the tissue. Okay? So we hope, after mixing the two things, of course there are some things in our hands which we can change so that we get the right tissue we want. And it also functional. So for example, if you want to have a beating tissue for cardiac regeneration, then it should beat, okay, and so on. So essentially a 3D culture. So here we are talking about cells which are embedded in a material. It's not on a top of a 2D situation. So we are talking about a 3D culture where the cells are surrounded from all the sides by the biomaterial, okay? That's an important thing to note. Of course, cells, I'm sorry for the quality of the picture, I don't think it's very nice, but the important thing I wanted to show with this picture is that the cells themselves interact with the surrounding material, surrounding matrix, through adhesions, focal adhesions, or even hemidesmosomes. So there could be different kind of adhesions which can help the cell to interact with the surrounding material. Now, what do we expect the cells to do? We could expect them to proliferate, increase in number. We may expect them to actually change their shape and also differentiate, because ultimately we want a cell type which is present in the target tissue, and it also gives the function it has to give. So, what are the things in our hands? So we do have few things which we can tweak here. Important ones are biological things, for example, nutrition. Enough nutrition should be there. If it is too much, probably proliferation will take over other functions. Air, oxygen, most of the time, cytokines, growth factors, which can take the cells in a certain particular direction. But then we also have in our hands mechanical stimulation. Okay, that is another thing which we can play with. Here are a few words. I'm going to explain those words, of course, in detail today. Stiffness, viscoelasticity of the scaffold, and on top of that, so these two things affect the cells passively, but active effect can be done by applying forces or stresses on this whole material. We can apply compressive stresses, shear stresses, and so on. Okay? All right, so let us do some basic stuff and then combine it with some understanding. So the mechanical stability of any material or tissue can be quantified by few different kind of tests we can do. So you can see here what we are doing. We are holding a tissue or a material and pulling it from two sides. Here we are pushing it into each other, and here we are sliding past. Okay, so these are respectively then called tension test, compression test, or shear test. And what do we get out of them is a modulus, which could be tensile modulus, compressive modulus, or shear modulus. Now, these E moduluses in the tension or compression are denoted by E, and the one shear modulus is denoted by G. And actually, these properties are interconvertible if you know something called Poisson ratio. And for most of the biological tissue and soft materials, Poisson ratio is about 0.5. So if you know any of them, you can convert into the other. Okay? Now, what does this modulus tells us? So this modulus actually tells us about the stiffness of the material. And what does stiffness means? It is resistance to deformation. Okay? So that is simply understandable. If you have something metallic in your hand, if you apply force to it, it does not deform. Whereas if you have a rubber band in your hand, it very easily deforms. So stiffness or modulus is connected to that. So scientists actually have realized long ago already that tissues have very different stiffnesses. Okay? So for example, starting from bone, which is the hardest, actually the hardest material in our, do you know what is the hardest material in our body? Stiffest, hardest? Sorry? Yeah, dental enamel, correct? Correct. But here, let's forget about dental enamel. So bone, in this case, is the hardest or stiffest. Going all the way to blood or brain. Brain is the softest tissue. So you can see there is a whole range of stiffnesses. Cartilage, among the soft tissues, cartilage is the stiffest. Bone is anyway the hardest. It's a very hard, brittle tissue. And the fact of the matter is that it has been well known that stem cell differentiation is affected by the stiffness of either the substrate in case of 2D cultures, and in case of 3D cultures, the stiffness of the surrounding matrix. And there are wonderful reviews which can tell you that how to play with stiffness in order to get a certain cell type. And here, Professor Van Rijn, he also uses stiffness as one of the important selection parameters for checking if cells go in a particular direction, besides other properties of a surface. But more recently, there has been a realization that viscoelasticity also affects cell response. So when I'm saying this, I'm not denying that stiffness plays a role, but on top of this, you have viscoelasticity which starts playing a role. So for example, here, 2011, the first paper came in this direction, the influence of substrate creep on mesenchymal stem cell behavior and phenotype. Then in 2015, hydrogels with tunable stress relaxation regulate stem cell fate and activity. And then matching material and cellular time scales maximizes cell spreading and on viscoelastic substrates. So this is all good. This is all in literature, and you can read those papers. But the problem is there are so many difficult words here. And I know that there are not a lot of students who are from an engineering background. Am I right? Most of you are from chemistry and biology. So I just wanted to give you a very small introduction of those things. So next time when you read an article like that, you are equipped well with the knowledge. Okay? So there are words like stiffness, but now we not understand what stiffness means. Elasticity, viscoelasticity, relaxation, creep, time scales. Yeah? So we have read these words. What does this all mean? Now, of course, we are going to understand all this, but ultimately, what you have to think about is this scenario here. So basically, the cell is inside a biomaterial, a matrix. It is pulling or pushing on the surrounding material, and depending on how the material responds, the cell will go in a certain particular behavior. Okay? So all these things, properties, will define how the material is going to interact with the pulling and pushing forces implemented by the cell. Okay? The material is not going to impose anything on the cell. It's the cell which is the driver of pulling and pushing, and the response comes from the material. Okay? All right, so this is a simple device which we have in our laboratory where what we do is we take a small piece of tissue or hydrogel, we place it between two plates, and then we just compress it. Okay, simple test, and then we measure few things out of it. And I'm going to describe you how those things look like so that you understand from basic on what we are talking about. So we basically control the position of the top plate. So when we go down, 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 then our material slowly gets deformed. Okay, it's get compressed. So this is what we basically do. We take the top plate down and we increase the deformation as a function of time, and at some point we can leave it constant. In order to do that, of course, we have to apply certain force. And in our device, we have the bottom plate connected to a load cell. It tells us exactly how much force we applied in the process of doing that. So the outcome or the measurement which we do is basically the force. So how much force did we need to give this deformation as a function of time? Next, what we do is we basically convert these two properties, or these two, deformation and force, into some other property. And you can see this one here is called strain. And strain is nothing but deformation, but what we do is we normalize it against the starting thickness. That's all. Okay? So strain basically means deformation. With respect to force, we do something similar. We divide it by the area of the top plate. So you can imagine here, if this plate is very thin, small, then we apply small forces. But if this plate is quite large, we apply more forces. So in order to normalize for that, we divide the force with the cross-section area of our top plate. Okay? So basically now you have force and deformation in terms of stress and strain. And this is plotted against each other. You can see stress is plotted against strain. In the beginning, you have a very nice straight line linear response, and at some point, something starts happening. So what we say is somewhere here, for example, the material loses its structure, its integrity. Okay, so this is the, let's say, the failure point. But in this linear region, we can actually measure the slope of this line, and that is the modulus. Okay, so modulus is coming out of force and deformation, but in a normalized situation. Okay. Okay, so now let us go and start to understand different type of materials. So here we are talking about elasticity and stiffness. So let us see what we are doing here. We have a rubber band, and we are applying it. The rubber band has a certain length, and we are applying force on it. And the consequence of that is, of course, that the length of this rubber band increases. So you have a certain amount of deformation. So you can see here, what we can do is we can plot force against deformation, and then we convert that into stress versus strain. Exactly the same as we did in the previous slide. The only difference is here we are doing tension, and there we were doing compression. Okay? So simply the same thing, and the slope of this line gives us the modulus. Okay? So just to repeat, E is the modulus of this material, and it gives us the stiffness of this material. Stiffness means resistance to deformation. So meaning, if this line is somewhere here, like this, then the stiffness will be higher. Modulus will be higher, stiffness will be higher. Okay, then an important understanding about this concept is, now I'm sure you can compare, you can imagine the difference between this process, which you can do in your hand, and the elastic cord in the bungee jumping. When somebody is doing bungee jumping, how does it extend? Yeah, and what is the difference between the two? Can anybody quickly say? So stretching a rubber band in our hands, or this cord stretching when we fall? True, gravity, yes. Any other difference? Can you think about how this material is being deformed? Slow or fast? Something like that. Yeah, so once the bungee jumping is loaded, that means first the bungee cord unfolds, and then at some point the person basically starts extending the cord. And that process happens at a very fast rate. Yeah, and this process, of course we can do very fast, but generally we would do at a certain speed. Nothing compared to the speed at which this material is deformed. So what I'm trying to say here, what we are talking about is the rate of deformation. How fast do we deform? Okay? And you can see for elastic materials, which can be represented by a spring, this modulus E is independent of how fast we deform. So if you take the rubber band, you deform slowly, fast, it will give you the same value of E. Within limits, of course. If you're going into astronomically high speeds, then maybe things will change, but for practical purposes. Okay? So stiffness for elastic materials it doesn't depend on deformation rate. Now, let us then take this argument a little bit further. So here, this is something we understand now completely: stress, strain, modulus, stiffness. But most of the materials in biomedical situation are viscoelastic in nature. Okay? So they are not elastic, but viscoelastic. So if we do the same thing, exactly the same test, you can see there is deformation as a function of time. We measure the force as a function of time. And if we now do this test faster and faster, you see what I'm doing? I'm increasing the slope, so deformation is having faster and faster. And you can see that the force response changes. If we now do all our magic, and look at the stress strain response, actually that is also going to, change. And you can see that the stiffness actually is changing. The modulus is actually increasing if I do the deformation faster and faster. So you can see this is completely different from what we observed before. So for a viscoelastic material, the stiffness or modulus becomes deformation dependent, the rate of deformation dependent. Okay? In the next slide, we will try to understand why that happens. So it is all about water. So we are 70% water. Even hydrogels, it could be 90 plus amount of water, percentage water. So it is all the play of water. Now, I'm sure Mexico is the nice place to do all those activities, I think, going to Cancun and either walking nicely on the beach. So when the water is warm enough and you are walking very slowly in water, actually you don't feel the water, okay? Unless the water is very cold, you just can slowly walk through it. But if you fall in water, we know what happens. We get hit by the water. Okay, so what is going on here? What is the difference? The difference is here your legs are deforming the water very slowly, whereas here your whole body is trying to deform the water very fast. Okay, so when we talk about fluids, in this case water of course, then this deformation rate becomes important. Okay? So slow movement, no feeling, whereas when you try to deform at high rates, you do feel water. Now, if we try to understand this a little bit on an engineering way, the fluid, or any fluid or water in this case, is represented by this nice symbol. And I will explain that symbol to you. Look, this is kind of a bucket, you can see, where you can pour water or something. And this is a piston or a plunger which is setting in here, and it can basically move up and down. Okay? Now, if we fill this bucket with a liquid, then, and if we now pull this piston upwards, this piston can move upwards only if the liquid from above this plunger can move below the plunger. Right? So there is a very small space here, you can see, where the liquid can flow from top to bottom. Okay? So the faster the fluid can flow, the faster can this plunger move up. Okay? So the speed at which this, we call it a dashpot, responds really depends on the viscosity of the material, of the fluid, I mean. So water is really thin and less viscous, whereas oils are more viscous. So if I fill water here, the plunger will move up faster. If I put oil, it will move slower. Okay? So this is a symbol which takes care of the fluid properties in a material. And we already saw that the elastic properties are taken care by the spring. Okay? So in a viscoelastic material, both the fluid properties and elastic properties are combined. And very often we represent it with this symbol. You can see the spring is giving you the elastic response, or taking care of the elastic response, whereas this dashpot is taking care of the fluid-like behavior, which is present in your material. Okay? Okay. So stiffness, now we know for a viscoelastic material, or even for viscous material, the stiffness modulus is dependent on the deformation rate or strain rate. Okay? So what are we all talking about? We are talking about time. Time coming in. When we are talking about rate, that means time starts playing a role. So here I would like to give you a caution that if you have a viscoelastic material, and if you only measure stiffness, and if you have 3 different materials, you can compare the stiffness, yes, but only if they are all measured at the same strain rate. Then you are allowed to compare and say, look, this one is stiffer than the other. Okay? In absence of any other testing, this can be done, but caution at the same strain rate. If you don't know the strain rate, for example, there are many reviews where people talk about hydrogel stiffnesses, and they do a literature survey. They have no idea at what strain rate different stiffnesses are measured, but they do compare them. Okay? So often it goes okay, but it should not be done in principle. Theoretically looked at, it should not be done. Okay. So then I said that only having this value of modulus is not enough, because we are not really taking care of everything of this material. What can we do? So viscoelastic properties of a material actually is, illustrated or measured in terms of 2 different kind of tests. And these are actually time-dependent tests. And I'm going to describe to you, and you can understand the words relaxation or creep in this slide. So first test which we do, and which we actually do in our lab, is called stress relaxation. What do we do in this test? Okay, the second test, which we don't do, but some people do, is called creep test. In stress relaxation, what do? We actually impose strain, meaning deformation, very quickly, and then hold on to this strain. So I compress my material and keep it that way. Okay? And so if it would be an elastic material, then the response on the stress side would be looking like this. So when I want to deform this elastic, I have to apply certain stress, but when I hold on to this stress, then I have to keep the stress constant. Because if I reduce my stress, this deformation will go down. Okay? Simple elastic behavior. But if you think of chewing gum, and I'm sure you have done this in your life sometimes, if you have chewed on chewing gum, and if you try to stretch it, when you want to stretch it, you have to apply force, but after some time, the chewing gum actually starts deforming by itself. You don't have to apply any forces. And that is exactly what is called stress relaxation. Okay? The material cannot hold on to stress. It just flows and relaxes. So in this situation, if I would have a viscoelastic material, the response would look like this. So during deformation, I have to apply stress, but then the requirement of stress goes down. And that's why it's called relaxation. Now, in creep tests, how do we do that? Is instead of applying strain, I apply stress. Okay? So you can imagine I take a rubber band and hang it on a nail, and then at the bottom, I put a small weight. Okay? So I'm applying constant force, which can be converted into constant strain. Yeah? So I apply the load and then keep the stress constant. Now, if it would be the rubber band, you would expect a certain behavior, and I think you can now predict it. But if it is a viscoelastic material, you can see this is what would happen. During the application of load, the material will elongate, but it will keep on elongating afterwards as well. So think of this chewing gum. If you hang this chewing gum from a needle, from a nail, and then apply a certain load, this chewing gum is going to deform forever. Yeah? So this is the response which you expect for a viscoelastic material. Of course, this red line tells you that if it would be a rubber band, it would be constant strain as a function of time. Okay? So any of these two tests can be done to find out the real viscoelastic properties of a material. In our lab, we do the top one, so I'm going to describe you that one a little bit more. So stress relaxation done this way, application of strain, stress is what is measured. What you do is you just divide the stress with the constant strain, and you get something called relaxation modulus. And what we do next is we fit something called a Maxwell model, generalized Maxwell model. And don't worry, I'm going to explain to you that. But at this moment, this is the model which we fit. It has a certain equation. You can see this equation can be fit to this measured data. Okay? Each of this, we have seen individual of this element, a combination of a dashpot with a spring. This we have seen before. Now, each of this element actually remains or becomes active for a certain period of time. And that period of time is dependent on the viscosity and the modulus. You can see here, this relaxation time, tau, is the ratio of the viscosity to stiffness. If some people know about the unit analysis, Pascal seconds divided Pascal is seconds. So when you take this ratio, what you get out is time. Okay? How does this work? Let's look at a practical example. This is measured data, and the red line, which you don't see at the back, is actually the fit. Okay? And that fit is composed of multiple Maxwell elements. So look, if I zoom in into the first 5 seconds, you can see it better. The red line is the fit, and these are individual Maxwell elements. That means this Maxwell element was active for 1 second, whereas this Maxwell element is active for longer time. Okay? So every element contributes to fit the experimental data to the model. Okay? Again, the time for which each of them remain active depends on their viscosity and stiffness. Okay? Let us see how does this all work. So imagine this is a single Maxwell element, and it has a certain viscosity and stiffness. If I now stretch it, give it strain, because this takes time to respond, all the deformation goes into the spring, and we will see what happens as a function of time. As time passes, you can see this bucket starts moving downwards, and the spring starts contracting. And up 60 seconds, you can see that this dashpot has completely done what it can do. So it has done its deformation within these 60 seconds. Okay, so you could see that there were Maxwell element which was active for only 1 second. So the whole process would happen in 1 second, whereas the other ones go longer and longer. If we now look at a complex system, which is bunch of Maxwell elements, you can see the viscosities are the same. The only thing which is different is the springs are stronger and stronger. Okay, bigger and bigger. So you can see this is less stiff spring, this is the stiffest spring. Now if we do the same thing, if we pull it, and if we see what happens after 10 seconds in example, you can see that this strongest spring has already pulled the bucket down, and this element is done. It has done its job. This one is halfway, and this one is almost not even started. Okay, so you can see that the ratio between the viscosity and the stiffness matters. Since the eta by E here is smaller, this element goes very quick. So the value of tau is very small. At 60 seconds, everything is done. Okay, but the important thing which I wanted to say is each element is active for a certain period of time, and that time depends on the ratio of this eta and E. Okay, all right, good. Now here I just show you some measurements which we did in our lab. You can see liver tissue, adipose tissue, fracture hematoma, and you can see the first thing we can see clearly and understand is the amount of relaxation. So we start here at 40, but you can see we have after 200 seconds we reach about 5. So there is quite a bit of relaxation. But for example, adipose tissue in 200 seconds would relax only 50%, liver 66 or 67%, and fracture hematoma 90%. Okay, so each material tissue can behave differently. Okay. Okay, and let us not go there at the moment. You can see these are all the after fitting. You can find out what are the different elements here, and you can see there are elements which are lasting for 3 to 4 seconds, 50 to 70 seconds, and more than 100 seconds. So 900, 2000, and so on. Okay, now can we try and understand what is the meaning of these Maxwell elements? I just want to give you a physical interpretation of it, although the answer is not exact. Okay, so what are we doing? We have a material, and then we press this material, we compress this material. During that process, what we are doing is we are generating a region of high pressure, because we compress it. So there is a region of high pressure which develops just below the plunger. And now the material which is here would like to move out of this high pressure region. Okay, so you can imagine that it depends really on how big each of those things are in your material. So if we are talking about hydrogels or tissues, water is a smallest molecule, and water can probably escape this space the fastest. Okay, so maybe the fastest Maxwell element links up to water, movement of water in your material, for example. Yeah. Then comes bound water, because we discussed about glycosaminoglycans, proteoglycans. These are all present in tissues, and they have a lot of bound water, and that bound water will move slowly. Okay. Then the whole network itself can change, the matrix. And the last would be cells. If there are cells, they would then respond to your stresses. Okay. Still, this is not final, because there is no systematic study which shows that this element actually belongs to water. Okay. So this is just to give you a physical understanding that something is going into your material, and that can have manifestation in terms of time. Okay. Now, from the cell side, so we have now materials with different elastic and viscoelastic properties. From the cell side, what response can we get? So, few things which we always think about and measure is spreading, for example. And spreading not only happens on 2D situations, but also in 3D. You can have adhesion of cells, and that can be quantified in terms of focal points. You can even quantify the size of focal points, density of focal points, and so on. Next thing is proliferation. Under certain conditions, cells may just proliferate, increase in number. And last but not the least is differentiation. And very often in tissue engineering, we depend on differentiation because we don't start with the type of cell which we ultimately want. Okay? I have only 7-8 minutes, I think, and we are oh, you reduced it even. Okay. So I think I cannot show you everything, but let us quickly see the 3 papers I showed you. What have they shown in principle? So this is the first paper, 2011. What they used was human mesenchymal stem cells, and they used polyacrylamide gels, and they were able to make 3 different gels with the same stiffness. So they were able to create gels which has the same stiffness, but they had very different creep behavior. Now, when I say creep, you can also understand this as relaxation, because the material will behave in the same way. Okay? So you can see here, there is almost no creep, and here there is quite a bit of creep. Okay? So increasing viscous contribution. Let's think of it that way. Okay? So they basically did it through a particular chemistry, which I am not very good at, but I have colleagues which can help me understand that. So here, basically what did they measure was average cell spreading area and then fraction of focal adhesion. So they basically looked at the size of focal adhesion things. So, and basically the message here was higher creep, that means higher viscous component, better was the cell spreading, and smaller the focal adhesion. Okay? In this study, they did not talk about time constants. Okay? So they did not really think about time at this moment. In more viscous contribution, different cell response. Then we come to this interesting study where they actually used, again, human mesenchymal stem cells, but they now played with alginate. And they used alginate with different molecular weights, but they also used polyethylene glycol as spacer. So by that, they basically made materials of 2 different stiffnesses, but very different relaxation behavior. You can see the relaxation response is different. Lower molecular weight or presence of spacer caused faster relaxation. So basically, you can see these curves are on log scale. So if you convert it into linear, it looks like the curves we saw. Yes, okay, let's go to the, oh yeah, another thing, important thing, alginate and PEG are both not bioactive. And with bioactivity, I would mean that you have something on which the cells can adhere to. So in this study, they actually externally added some RGD moieties into the gel. So first thing they did was to find out how much RGD do they need to add in order to get a certain response. And look here, faster and faster stress relaxation. So this is slow, meaning this is less viscous, this is more viscous. And you can see that if you go from left to right, cells tend to spread like the matrix and do something which the other cells are not doing. And they found out that you need a certain concentration of RGD in order to get a good cell response. So then they did the rest of the experiments at 1500 micromolar RGD. And what did they look for was adipogenic differentiation and osteogenic differentiation. Now, these were present. You can see this is less stiff gel, high stiff gel, more stiff gel. Low stiff, less stiff, high stiff. In less stiffness, we got angiogenic differentiation. So you can see stiffness still is an important parameter. And in high stiffness, we got osteogenic differentiation. But if you now look at the results, you can see here, more viscous at high stiffness gave you more and more osteogenic differentiation, whereas less stiff, but how do you say that? No problem. You get adipogenic differentiation. I think I should better stop here because then I will not have any questions, right? Yeah, I stop here. So, but you get the idea that stiff viscoelasticity is also important as compared to stiffness for getting a certain cell response. Thank you.
So we will be starting with a round of questions. All right, sorry. So, quien sea que tenga una pregunta puede alzar la mano y les pasamos los micrófonos.
No questions means. Thank you, Patrick.
Thanks
saved
a
my
lot.
day.
No, but now I finally understand how a stress strain curve is being built up. So thanks a lot. I was wondering. So you showed these different RGD concentrations and then increasing relaxation properties, right? And what you could notice is that at the far end of the low concentration, it matched more or less the high concentration, but then a little bit before. So it almost seems like with stress relaxation, you're able to promote more or less. So if you would go higher in stress relaxation, you would at a certain point almost see that it behaves like high RGD concentration.
Really?
So would that be a possibility?
No. Because cells do need minimum adhesion points, I think, in the matrix if they want to show something. Yeah, but there is also limitation to how much viscous you can make your material, because ultimately you need a hydrogel which is workable. So yeah, I mean there is a limit to how much.
No, because you also showed that if you change your relaxation, then you change the areas of your focal adhesion. So if you downscale your focal adhesions at lower RGD concentration, then that matches, right? So you have less interesting.
Yeah, but then we have two handles to control things. One, I would say the mechanical properties are a little bit more difficult. In this case, RGD is easier. Just add it in this situation, you're done.
Yeah.
So we have another question in here.
Hi, Doctor. Really nice presentation. I just wanted you to ask, which do you consider are the factor more difficult to standardize when studying friction-related diseases?
Friction-related diseases?
Yeah.
But, okay. So that is not connected to this talk, huh? No. Can we talk about it later? Okay. I can talk
sure.
a lot about that, so that's the problem.
Don't worry.
Yeah? I will.
Yeah, thank you.
Hi, thank you. Well, I was wondering if, since we have different stiffness for different tissues, if you want to apply something like a material into some kind of tissue, you need to consider also if this tissue is going to be scarred, so you need to change the stiffness to the new tissue that is going to form.
So what I can say on that is you don't always start with the final stiffness or final viscoelasticity you need, actually. Because the cells have to do still a lot of work, produce a lot of ECM on its own, and then the final property should look similar to the tissue, what you need. So the starting viscoelasticity or stiffness, I would say, is to tweak the cells in the right direction. So use those two things as stimuli rather than already giving the properties we need, because then cells have nothing to do, more or less.
Okay.
right? So I would say use stiffness and viscoelasticity as a stimuli. Yeah?
Thank you.
Hi, thank you. I was just wondering, how do you maintain the variability of the area for the stress calculation?
How do I maintain the?
The area of different samples. Like if you want to test different samples, how do you maintain that variability?
No, look, the tissue can be larger, or a piece of my hydrogel could be large, but I only press it with a plunger which is having a certain diameter. So my area of cross-section always remains the same.
Okay. And do you use also nano indentation?
No, we do micro indentation.
indentation.
So we have the same device I showed you. We now have it in a micro scale. So we can do really small pieces of tissue and gel, for example. And the problem, so nano indentation we don't have actually. We do have AFM. But the problem with AFM is you cannot control the strain rate. And since I know that, I stay away from AFM for this purpose.
Okay.
For other things I use AFM, but for this one, no.
All right.
Yeah.
Thank you.
Yeah.
Hi, doctor. Thank you. My question is, physical gels can behave or have these specific viscoelastic properties or only cross-linking gels?
Please repeat the first part of the question.
Physical gels
Yes.
can have these specific viscoelastic properties or only cross-linking?
Both.
Okay.
Both in principle. But if you want to, for example, have a stiffness of a certain level, then I would say you have to go to a chemically bound, covalent bound.
Okay.
Okay? So
Yeah.
I wanted to show some examples. So now what we are doing is we are using gelatin methacrylate, GELMA, which is basically with UV, you can cross-link it, covalently cross-link it. But then it is really a stiff gel. The relaxation is very small. So what we are doing is adding, for example, polyethylene glycol or chondroitin sulfate or gelatin, free gelatin, just to be inside the gel so that water is kept in the gel. So by just adding these hygroscopic molecules, you can tweak the viscoelasticity. You can increase the viscoelasticity. So the properties can be in both gels, but the covalently bound, chemically bound, are workable. You know, you can use them and more easily.
And a second question, as a chemist, how can I put the cells inside my hydrogel?
Well, I think, so the strategy we use, for example, with gelatin methacrylate, we do work with organ-derived matrices also. So basically, you first need to make a kind of a pre-gel or a mix, and then you cross-link later on.
Okay.
That's the easiest way, and that's the only way. See, because hydrogels are not, their porosity is on a nanoscale.
Yeah.
You cannot let the cells go by themselves. So you have to pre-mix them and then cross-link.
Okay. Okay. Thank you.
Yeah.
Good morning, doctor. So I was wondering that regarding your compound about the hydrogel, you mentioned us that the morphology, the proliferation, and the differentiation are important for these properties of the hydrogel. So I was wondering, when a cell is dividing, does this actually changes the properties of the hydrogel, the stiffness and the tension that you have already mentioned?
So this is a very simplistic view of how things are happening. You can imagine two things happening. When you have seeded hydrogel implanted in vivo, so the body is interacting with the hydrogel, and also the cells are changing the hydrogel or the material from inside. It is either degrading the material around with MMPs or depositing its own extracellular matrix. So the scaffold properties will change drastically during the process. So in 7 days, 21 days, the properties will be completely different.
Okay.
Yes.
Thank you so much.
Yeah.
Yes, just now a very clever question arose. The difference between a physical hydrogel and a chemical hydrogel. What if the physical hydrogel breaks during the process? a chemical gel will not really break. So how
During
does that
which
influence-
process you mean?
Well, if you apply stress,
Yeah.
and all these components happen, but also your matrix deforms and breaks up because the non-covalent interactions are not strong enough to
Yeah.
withstand. How does your story then?
Yes. So see, when we do mechanical testing, then of course we know what are the limits. So we just tweak we check the modulus within the limit when it breaks. Now in vivo or in situ or during tissue engineering, well then I have no idea how this goes. Of course, for the cell, when it pulls on a physical gel, it's easy. The physical gel will respond much more easily as a covalent gel. But physical failure, I don't know. Never thought about it. I don't know if it happens.
Well, maybe it's related to muscle tearing if you exercise, right?
Yeah, could be.
All right. Thanks.
Yeah.
That was the last question. So
Thank you. Thank you very much.
Thank you for your conference, Dr. Pashant. Up next, our next conference will be on charge of Dr. Theo van Kuther. Hi, Doctor. So before we start, can I say a few words?
You can say it.
Yeah. Okay. So. Okay. Okay, so Dr. Theo van Kuther is a biomedical scientist with a background in studying body implant biomaterials and their interactions with cells and tissues. He has worked on various projects over time, leading to changes in his area of interest. Education has also become an important aspect on his work, and he has taught courses on the biomedical and biological evaluation of implants, the immune response to biomaterials, and the interface biology at University of Groningen in both bachelor and master's programs. And currently, his research focuses on controlling the differentiation of epithelial cells of the crystalline lens through interactions with biomaterials, as well as the degradation of polymers by macrophages and as an in vitro model. Moreover, he's involved in macrophage activation and biofilm assays on biomaterial surface. Overall, his main research and teaching subject revolved around the biocompatibility of biomaterials. So, well, give us a few minutes and then we can start with the conference, please.
Thank you very much for the introduction. So for today I want to talk about some aspects of our research and especially some in vitro parts with cell culture systems
Then I want to go to a recent approach on studying the foreign body response. And then finally something about the process which is called EMT, epithelial to mesenchymal transition. I probably have too many slides, so I probably have to cut somewhere, but I will notice your signs there. First of all, I want to say something about my discipline. I'm a biologist, so if I stand here, you can see this very nice bird. It is the Aquilucho siniso, and it's going every year from Africa to Europe, Spain, to the Netherlands, and even to more northern regions. And so we study that bird. We are looking for that bird in different countries. And when I was doing my study in biology, I started with things like ecology, animal behavior, plant physiology, and so on. And only later I specialized in what at that time was called human biology. Nowadays it's very comparable with biomedical sciences, I would say. So that's my background, and this is still my hobby. So this is my free time. Thank you. So this is my free time occupation as well. Okay, in the summer school I showed you that the foreign body response is composed of different stages with different cell types coming from the immunology part, so neutrophils and macrophages mainly, and that later on fibroblasts are going to construct or reconstruct tissue. So talking about these cells, I will now focus on macrophages and fibroblasts and show you how we study those cells in vitro in cell culture systems. So those cells are very important for the foreign body response, but they, of course, can also easily be cultured, especially fibroblasts, of course. Macrophages is a bit more difficult maybe. There is a lot to tell about that, but you can culture them. And it all belongs to this ballpark, wound healing and the foreign body reaction. And wound healing is nothing else than inflammation. So inflammation is an important aspect in our body. And I also, in this talk, want to combine it a little bit with infection as well. So we look into the macrophage functions, and I also showed you this picture. This is about degradable polymers. This is one type of polymer, polyurethane. It can be made in a kind of semi-inert way, but it can also be made in a degradable polymer. And if you if you put it in an animal body, the foam of polyurethane will be degraded mainly by macrophages. You can also do it with other polymers. This is another polymer, poly-3-methylene carbonate, and you see the macrophages. Those macrophages are eating their way, here you see one macrophage, into that material. They do that by secreting enzymes, and the enzymes are degrading the polymer chains, and the polymer is disappearing. It is a real hallmark of macrophages. That's important to understand. Here you see the response by macrophages is really the one that gives this erosion, surface erosion of this material. If you look at other cell types, this is a fibroblast. The fibroblast totally likes the surface. That's no problem. It will totally cover the surface, but it will not do anything to the surface. So the surface is not affected by the fibroblasts. So this system is something we did together with the University of Twente, and we looked in into many aspects. In Twente they made many different biomaterials or polymers containing poly-trimethylene carbonate, but also other parts in copolymer systems. And we studied that with macrophages, what was the exact degradation. But it's also a very nice system because it's very visual for students. So I also let students play with this system. I have been doing that for quite many years now, playing with this system and look at all kinds of different aspects of the system, the behavior of macrophages, and you can do that with bachelor students, with master students, but also even with students studying medicine. So the good thing is that they really see what the cells are doing. And that is different from many experiments. I'm sure that many of you are doing sometimes experiments in the lab, and you have to wait till the final moment that you get some quantitative data out of your experiment. But for the rest, it has been a black box. Cells are doing something, but you don't know what they are doing. And here you can see that. So that is the plus of this system for students beginning in the lab. So we had several aims. I just mentioned two of them here. One of the first things we wanted to exploit is to see which enzymes were being used, and also if reactive oxygen species were used for the degradation of PTMC. And later on, then we started combining the response of macrophages and fibroblasts to see what they were doing. And this is such an experimental setup. So we work especially with mouse fibroblasts. They are easy to culture. And if you look in the literature, there are several cell lines of mouse fibroblasts. And in consultation with David Granger in Utah, we came up with this macrophage cell type J774. And this cell type is currently being used in our lab quite a lot, actually, for many different purposes. So the J774 mouse macrophage is then directly cultured onto the PTMC. At that time, when we were looking into the roles of the different enzymes, we decided to use different inhibitors for different enzyme systems. So you see the inhibitors over here. The inhibitors, that's an entire row of inhibitors. And we decided to also test if complement has an activity here. Complement is a system which is present in the plasma and or the serum that we add to medium, and it can have an effect on how cells respond in certain systems. And there are also some people who say, I always take out complement. It's quite easy, 30 minutes heating to 56 degrees of the serum will inactivate the complement. So we did that, added complement, or we kept the complement, or we inactivated it, and then we did these studies. We followed this with fluorescent microscopy. We also looked at the cytokine response with ELISAs. And since this material PTMC is degrading, you can measure the weight after the cells have done their work. So the weight determination is an important aspect as well. And this is a typical response that you see. So you see a percentage mass loss. The higher the peaks are, the more of the material is disappearing. And you can easily see now that especially this one, the DPP, is inhibiting degradation. And this is the one which is inhibiting cholesterol esterase. So we have a quite easy answer already here. Cholesterol esterase certainly plays a role. For the others, we are not totally sure. It's possible that we don't have the proper concentration. It's possible that the half-life of these enzymes in a culture system is quite low, for example. All those things can happen. So that is something that would deserve further studies. But you can see here the cell densities are quite similar for most of them. I can say that the cocktail and EDTA were toxic to the cells, so the cells disappeared out of the equation. And of course then you don't have any cells in your field, but you also don't have any degradation. And this is how it looks like. So you have a bunch of macrophages sitting on the surface. The J774 macrophage is behaving like a kind of spherical cell. If you activate it with LPS, it will spread out quite drastically, by the way, but in many cases it stays quite a spherical cell. It's not a real spreading behavior going on. And if you look with confocal microscopy, you can indeed then pinpoint where the degradation is taking place, and of course you can see in this part that they go into the material. So for the enzymes, at least we know that cholesterol esterase is involved. Others can still be involved. We also have some inconclusive results for the role of reactive oxygen species. My feeling is that they also play a role, but it's quite hard to study that. We had one inhibitor for that, apocynin, but apocynin was also doing stuff to the cells themselves. It was not a very linear connection with taking away the reactive oxygen species or preventing the formation of that. So that was very difficult to study, actually. So the fibroblasts and macrophages together, you can bring them together in a co-culture system in a kind of juxtaquin fashion, which means they can touch each other. You can separate them with a semi-permeable membrane, the so-called thin insert system. So you put a basket in the well, and in that basket you can put the fibroblasts, and you can put the macrophages on the PTMC in the well itself. Paracrine influence is possible because signaling molecules can travel through the pores of the system. And what also is quite useful is looking at what cells are producing themselves. So we have an entire panel here from fibroblasts that have been cultured on a normal culture plastic, tissue culture plastic, on PTMC itself, and even on polystyrene. So that is the bacterial grade cultureware that is the basis for TCPS. And if you do that, what you can do is you can culture the fibroblasts, and after some time, 24 hours or 48 hours, you can sample the medium, and you can use that medium, after taking out the cells, of course, to give to the macrophages. It's called conditioned medium, and the only thing you do is you use the factors secreted by the fibroblasts to tease the macrophages. So we have that at several levels, and if you look at this, especially the CDE, and this middle column is going to give a drastic response. First of all, the coverage, so how many cells are there, very comparable. You see there is not a real effect on cell density. These cells, by the way, can proliferate, so it's not like a monocyte that you take out of the peripheral blood of a human. That cell can go into a macrophage state when you culture it on TCPS, for example, but those cells are not going to divide. They can do their macrophage function, but you don't get more cells. With J774 you get more cells over time. But this is the response in weight loss, and this is this group of conditioned media here. You can see the difference here with the other systems is quite large, and we have found that in many cases actually. So the conditioned media of fibroblasts is going to increase the response of the macrophages. It has something to do with the interactions of these cells, and the funny thing is, if you have them together, sitting together, or even paracrine, then there is an inhibiting effect. So there you will see that the presence of the fibroblasts together with the macrophages is slowing down the erosion of the material. So clearly there is some kind of two-way communication that is very important in this process. And of course this is what you can expect in a foreign body response as well, that there will be two-way communication. So this is what the Granger group proposed in 2011. There we have the fibroblast, there we have the macrophage, and if you look at this, there is an entire panel of chemokines and cytokines involved in this two-way communication. You can look at all of these with ELISA techniques. You can put them in multiplex systems and measure that. But they all have an important role in the communication. Although we don't know exactly how this all fits together, of course. That's the difficult part. Okay, so we have done many of these studies, and you can also study the influence of all kinds of anti-inflammatory agents, the steroids, the non-steroid anti-inflammatory drugs, and even more of these compounds, which we have also done, by the way. The question is, can we study the foreign body reaction in vivo then, over time? In the summer school I showed you that the very classical approach of a subcutaneous implantation, and at some point in time we wanted to have some more detailed insight in the processes going on in these animals, and we came into contact with a certain model which is called window on a mouse. So this is a collaboration between many people actually. So our group within the BME is involved together with my colleague Jelmer Scholma, but we also have a good connection with DSM, which is a company making different polymers that they want to introduce as coatings or materials themselves in biomedical applications. And we have also collaboration with the group of pathology and medical biology in the person of Ruud Bank. There is a fourth group involved right now. That's a group in Maastricht who has done a lot of these interaction studies with human macrophages with these materials. Also there we have a lot of information now about these systems. So we are actually looking in this entire ballpark where we have the different macrophages, the different types of macrophages. We have dendritic cells, we have the neutrophilic granulocytes, we may have giant cells, and we combine this model also with the presence of an infection. So we are also adding bacteria to the equation here. This is how it looks like. So the original publication was in Nature Protocols in 2013. What did they do? They made a titanium window. In that titanium window, they put a glass window. They glued the glass window. They put it in the skin of the mouse, but they used this to look at abdominal organs, to see what was going on in that mouse in real time. So this is how it looks like. It's quite simple, about 15 millimeters in diameter. Fits on the finger. And we thought, well, this is very interesting, they can do that. Why not use the window itself as a foreign body? Because we put it in the skin. So it is a biomaterial. It's going to be in contact with the tissue. And maybe it gives very valuable results, and we can look at that response. So this is how it looks like. We put it in the flank, the side of the mouse, and then we can look at mainly the fascia, a kind of muscular structure underneath. And what you will find is that there will be a tissue response developing in this space. And we can look at that tissue response by putting the mouse under a microscope. And with that microscope, we can look at the reaction of stained or labeled, it's not stained, it's labeled specimens. So we have macrophages which are expressing GFP, green fluorescent protein, and we have, if there is an infection, bacteria which are expressing M. cherry, which is in the red ballpark. Let's see if this works, I'm not sure. Okay, that works. There was an animation. So the mice are implanted with these windows. Then they will be subjected to the microscopy. They are under anesthesia for 45 minutes max, and we can do that according to the ethics committee 4 times in 14 days. That is what is allowed. So 4 times in 14 days we are looking at that mouse, same material, and we follow the tissue response. So for the methods, animals or mice, 8 to 12 weeks old, they are called mafia mouse, by the way. Mafia is a strain which is well described in literature. They produce macrophages and dendritic cells with GFP. And the bacteria, as I said, were M. cherry labeled, and we used Staphylococcus aureus with a certain strain, as you can see here. The skin is cut 17 millimeters quite exactly. The window is placed inside the skin, so the skin is surrounding the window. There is a kind of groove in that periphery of the window, and then we suture the skin in that in that part, and then it will hold together for several weeks actually. And if we put injections of bacteria in, that is an amount of 20 microliters, and the total amount of bacteria going in is 10 to the power 7. I want to remind you that 10 to the power 7 is a quite high load. And that is always a discussion item, because people say, well, in real life, if an infection is developing in the presence of a biomaterial, usually there are far less bacteria to start with, which is true. But in order to have a kind of detection system, there is no other way than to start with relatively high amounts of inoculates. So this is what is the experience with these animal models. If you give a lower amount, mice will clear the infection quite easily. So skin is sutured again, and the imaging is then performed with in this case, with a size microscope together with a 2-photon laser. Why the 2-photon laser? The 2-photon laser allows you to go much deeper into the tissue than a conventional confocal microscope does. Excitation wavelengths were being used, which are fitting to the GFP and M. cherry, and then we get this kind of result. So we get a 3-dimensional site of the distribution of macrophages in GFP close to the surface, underneath the surface that we implanted. With bacteria it looks like this. The reddish part in this part are the bacteria, so there are bacteria taken up by macrophages already. Those macrophages get a combined green and red color, so they become a bit orange, you could say, of green. And that is a sign that bacteria are in the system and that the macrophages are taking them up. They are phagocytosing bacteria. So it is possible to do several things with them. You can take static images, you can take set stacks of images, you can do a time lapse as shown in this particular case. And what we see is that we get a lot of data out of it, so we are still a bit struggling, but for how to cope with the amount of data. It's data in the order of going into the terabytes, so it's really a lot. After that, after the period of 14 days, we sacrificed the mouse and we cut out the window together with the tissue. Then we try to get rid of the window first of all. That can be done by etching, but I have also discovered that in cases of infections, for example, it's quite easy to lift it off from the tissue because the connection is not strong anymore. And then we are going into the common procedure for preparing these tissues for all kinds of microscopy, light microscopy, electron microscopy. One of the things we do is we develop these samples for STEM imaging. And STEM imaging is that we make cubes of around 100 times 100 times 80 micrometer times 80 nanometers. So very thin ones, and those are scanned with scanning, transmission, electron microscopy into a Google-like image. That's how it looks like in STEM. So this is the overall slice we have, and we see now that if we zoom in, we can look at all the different elements that are involved there, belonging to the foreign body response, even the collagen fibers, just to mention. So also the collagen fibers can be seen easily. And that's something we can also do if we have infected specimens, so we can see the bacteria. You can even see that within cells there are dividing bacteria, like this one. So some of these bacteria are still alive. And we can also see another process that is this process of netosis. That is when neutrophils push out the DNA content. Here you see such a thing. So this is a stretch of of a kind of matrix with a lot of DNA, extracellular DNA. And this is something that we can see throughout the entire sample we have. Also that requires a lot of studying, so you have to go to all these different spots, see what's going on. So in this project with DSM we decided to look at two different coatings on the glass. If the coatings are thin enough, we can still use the glass to have a good microscopy session. So the two coatings were PLGA, polylactic glycolide acid, and the other one is PEA, or polyesteramide. DSM had already done research in which they demonstrated that PEA has very favorable biocompatible properties. It is, like PLGA, degradable. PLGA, as you know, is hydrolytically cleavable, so the influence of water will already start degrading this polymer. For PEA it is twofold. There is a bit of water that has an influence, and there is also a large influence of enzymes again, like with the other polymers I mentioned before. So we did a lot of things. We wanted to look at the fibrosis, we wanted to look at the foreign body response, we wanted to look at the healing underneath the window, and we also wanted to see what the relative inoculation with bacteria, or what the inoculation with bacteria would do to the response. With the hypothesis that PEA would induce a less severe fibrotic response. That was our hypothesis, and that was also not combined, I will not show much of that, with the use of antibiotics in the coating. Okay, so looking at this, we had glass 12 millimeters, we had the different coatings. That was done by DSM, so they prepared the coatings, they put the coatings on the glass was shipped to us. Our technician did a good job in gluing this glass into the titanium windows. Then we went for sterilization. That sterilization typically is short exposure to UV or ethanol, mostly ethanol, very short. So there is no irradiation involved. And here it says that the titanium rings were sterilized with gamma irradiation. That's not completely true, because if you have PEA, it is possible that you damage that particular layer with this irradiation. And then we went to an entire scheme where we looked at the different responses, the different kinds of microscopy. One of the things is if you have bacteria, these bacteria expressed M. cherry, but these bacteria could also be detected in the so-called IFIS system. I'm not sure if you are familiar with the IFIS, but in the IFIS you can put an entire animal under the camera, and the camera will register if there is luminescence or fluorescence present. And you can make an image out of that. So we also had those images, and those images can then be used to see how the infection is developing mainly. So the infection clearance, and I think this is a very interesting picture, because I was involved together with our technician in doing the animal experiments. That's because I have a license to work with these animals, and I also have the experience with the microscope. We looked at those those mice, and if you just look at the PEA or PLGA coating, it is a normal foreign body response, nothing strange going on. There is tissue development, and you can see that quite easily. But the surprising thing came when we had the inoculation of bacteria and then followed up on that and see what happened. And actually, I would say you don't need microscopy at all. This is the response. We looked through the window. This is the window with the PEA. And you can also see blood vessels appearing here. We are actually looking at a very healthy piece of tissue. There is vascularization going on. It has this very nice pink color. There is really no sign of any bacterial infection. That's also what the IFIS is telling us. If you look at the IFIS, there is no signal at all. This is the signal on the PLGA coating. But if you look macroscopically, this is what you see on the PLGA coating. It's an enormous amount of pus, of mucus. Mucus secreted, for example, by the bacteria. The bacteria are probably forming some kind of biofilm-like condition here. So this is a huge difference actually between these two coatings. This is without antibiotics. So without antibiotics, somehow the polyester amide is able to clear the infection together with the body. And now we are looking into how this is possible. We don't know. We have all these observations. We are trying to connect all these observations. I think we have been discussing now for more than 1 year in frequent sessions with many experts how to present this to the scientific audience, because of course this is going into a publication. But it's a very difficult matter because it's very complex. So looking at the macrophages, this is how it looks like, this kind of images you get. The upper panels are the images close to the window. So these panels here and these panels. You see the day 2, day 6, day 10, day 14. And what you see with the PEA, polyester amide, is that you have an intense buildup of macrophages close to the surface. Very many macrophages are present. If you compare that with PLGA, I will skip this one. If you compare that with PLGA, the buildup of macrophages close to the surface is much less. So we think it is somewhere there, that it has to do with how many macrophages are going to be attracted to your interface. And this may be the answer, but then of course we still have to explain why this is happening and why this plays an important role. How many minutes do I have? 15? Ah, that's good. Okay, then I will go through this a bit more. So what we actually do also can do with the confocal microscope with two-photon microscopy, that is using the so-called second harmonics. If you use second harmonics, you can image collagen. So the collagen can be imaged without any further labeling. And this is what you see with collagen. So far from the window, for example, this is PEA, infected by the way, but you can see very nicely the buildup of all kinds of collagen fibers. You see thick collagen fibers, you see thin collagen fibers. The density of collagen fibers can be very high, actually. So this is an additional mode of microscopy that we also applied, and that also gave us valuable information. And these are things that can be put, for example, in machine learning algorithms to quantify the amount of collagen that is being formed depending on the depth below the window. This is the kind of graphs we get out of that. Also there we have to interpret this in terms of the response, in terms of the anti-infection behavior of PEA, for example. And of course in the end we can also finish with classical histology as shown here, just looking at the tissue response with histological stains. That's also a possibility. Again, this is such a STEM image, PLJ, sterile, many cells belonging to this foreign body response are there. We can characterize the cells, we can see blood vessels, we can see collagen. And one of the things we do with the collagen is has been done by someone within DSM. You can imagine that a large company as DSM is also very much interested in big data and how you can extract as much information out of that as possible, and then you get into the area of artificial intelligence and machine learning. So there is someone within DSM who has a lot of experience with this, and this is what he did. He went into this STEM imaging and then started identifying where the collagen was, feed this into the algorithm. In this case, and I'm not familiar with this, so don't ask me what it is, but he used neural UNet for that. And then he came up with this kind of full-scale image with collagen in the red. And if you look in hindsight what the effect is, so if you release this algorithm on all the images, it is actually very precise. I was really amazed what is possible with this. So the conclusions from our window WOWS model, as they will probably appear in the paper, are listed here. So again, a very interesting difference between two coatings, where one coating is attracting a lot of macrophages and at the same time has a very positive influence in preventing spreading of the infection that was applied to the mouse. Okay. This has also a bit to do with the foreign body response, a lot actually, and it has to do with fibrosis, so there may be a fibrotic capsule formation. And if you look at that, they are connected with EMT. EMT is the epithelial to mesenchymal transition process, and therefore I want to take you in the final 10 minutes to the intraocular lens implantation system that I've been working on quite a long time, and from which for me is still an open issue in the end. I will show you what the open issue is. What are we looking at? We are looking at the intraocular lens in our eye. The best lens, or the main lens in our eye, by the way, is the cornea, just to be clear. But the role of the intraocular lens is that we can accommodate. So we can easily read a paper here, or look to the back and see a sharp image. That lens is surrounded by a capsular bag, and the yellow part here is indicating the lens epithelial cell layer. So there is on the inside of that capsular bag one layer of cell. It's a one-layered epithelium. It's called lens epithelium. And for the rest in that lens there is no cell present. It's acellular. Over time, there are quite many people who develop this particular condition. It's called cataract. Many elderly people develop this, and cataract is nothing else than troubling clouding of the lens. And if this process goes on, it will make you blind. But luckily, in our Western world, we have a very good solution for that, and that is the intraocular lens implantation. That works about like this. So the surgeon would cut away the middle part here of the capsule. It's called capsular waxis. It's about several millimeters usually. It can be smaller than that, by the way. Then you have they take away this lens material. Usually they have to use some ultrasonics for that. And then they put in such a kind of lens. There are several types of lenses for this, but this is an intraocular lens. The materials for these lenses are the acrylates and silicons, roughly. And within the acrylates we have hydrophobic and hydrophilic ones. Both can be used. These so-called haptons, these are sitting in the equator region here and are holding the lens in place. Well, there are really many people who undergo this surgery. It takes about 10 minutes for the surgeon to implant the lens on average. It's a very fast procedure. People do this polyclinically, so they go in, lens implanted, they have to wait a little while and then can go out again. It's a very successful operation, I would say, as well. In 15%, this is a rough estimate, 15% of the patients, we see a condition which is called posterior capsular opacification. You see the result here. You see a bit of a tissue formation on the lens, or behind the lens in this case. And that tissue formation is very much a fibrotic response. It's caused by the lens epithelial cells mainly. So if this happens, you are going to see fuzzy again. So it's not a sharp image anymore. In order to prevent this, or in order to cure this, the ophthalmologist will go in with a jack laser, and that jack laser is shooting away that particular tissue, and at least the center part of the lens is freed, and you have a clear vision again. This is all very easy, I would say, so even that condition can be restored to good vision, and this is okay. But then, at the time when we started collaborating with an industrial partner, the question was, can we do something different? And if you have just a piece of plastic, of polymer in your eye, it's not accommodating. So you have clear vision, but the accommodation is gone. That's not a real problem. I mean, many of your parents who are young here will probably notice that they use reading glasses. The elderly people here will recognize that they need reading glasses to read something close by. Or if you are lucky, you may have one eye like I have, where I still can read pretty well on close distance. But many people don't, and when they are becoming elderly. So our idea was, you fill the lens with a kind of hydrogel-like component, at least a gel-like component. That gel-like component can be deformed by the capsular bag and the attached zonular fibers, small muscle fibers. And in that case, accommodation can also be restored. So this is the material that was developed by the industrial partner, silicon-based, refractive index 1.43, a little bit more heavy than water, so that it's not going to float, and then the stiffness comparable to someone who is 20 years old and 40 years old, two different ones. This is how it looks like. So this is a lens refilled with this silicon. It's totally clear. It's acting as a lens. It allows transmission of light 100%, and we are looking in the ballpark here of 20 years, that's this one, or 40 years. Two different formulations. By the way, this is the curve, the change of lens power that we see when we become older. So with 50 years, roughly, we don't have any accommodation left. This is natural process. The process occurs because our lens epithelial cells are constantly adding new lens fibers to our lens over time. So you can imagine if you add more and more volume to a lens within a capsular bag, it will be more on the tension, more and more, and at some point the accommodation, the change in shape is no longer possible. From in vitro studies that was mainly done in company together with ophthalmology, it was clear that it's possible to do this, so you can indeed fill the lens and you have a restoration of accommodation. We also noticed that it's quite biocompatible, or easily biocompatible, I would say. And this is the type of response that you then see. So we have two things, refraction, and we have accommodative amplitude. What you would like to see is that you post-operatively will see an amplitude of about 4 to 6 diopters. Then you have the proper accommodation to read at a certain distance and at the same time look in the distance. We also need a certain refraction, and I'm not familiar with this one here, with the units, but I know it has to be in this upper ballpark in order to have a clear image. A clear image is associated with refraction. You see already now that the capsule has been treated here, whereas here it has been untreated, and if we don't treat the capsule, it's going down quite rapidly. In half a year you lose visual acuity. And Also there, the response is because of fibrosis. So which problems can we expect to occur? Fibrosis, and then you would say yes, but we implant a normal intraocular lens. So there also you expect fibrosis, and you have shown that with 50% there is posterior capsular opacification. And then, yes, that is true. What's happening in the other 85% of the patients? Well, also fibrosis. It looks like this. This is the intraocular lens when you implant it. Here you see space where fibrosis is going to occur, maybe also here, but usually the central part is still clear of fibrosis. So and that's important because that's where the light is traveling. So those lenses, and here you can see that how it looks like. This part is relatively clear, and here is all the fibrosis. It's a massive fibrotic response. This one is also not accommodating, as you can see. This is the main culprit, that is a growth factor TGF-beta. TGF-beta is released by the epithelial cells. You go into this system where the epithelial cells are, and there is no chance that you can avoid that you injure these epithelial cells. There is injury going on. They will release TGF-beta, and then the TGF-beta is going to lead to all these changes in matrix formation. So these cells automatically will go in this response. If you fill the lens with a gel, you cannot use JAK lasers. That will be clear. You cannot just shoot away the capsular bag, because then the gel is going to leak into the rest of the eye. That is not an issue. So that cannot happen. So we have to find other ways, and we have tried a lot with trying to kill the epithelial cells, for example. There's no way that you can kill them all in a short time window. So we had to look in other ways of what we could do. This, by the way, is this fibrotic response of this. And as a biologist, although I like these birds I was talking about in the beginning, I also like this very much because there is a lot to see and tell from what these lens epithelial cells are doing. They have proliferation, they have transdifferentiation, and they are going to form these strands of fibrotic tissue. Not fine for the patient if this happens in the patient, but biologically it's very nice actually. Another example shown here. So this is post-operatively, and this was done in a monkey. And in the monkey you can see that this is where the extraction was done. It's only less than 1 millimeter. There is a plug inside, and this is all filled with silicon. So directly after the operation it looks very nice. And after 2 years it looks like this. So we have put this lens on the confocal microscope, and this is where, I'm going too fast. This is what you see. You have a ring of fibrotic tissue surrounding this, and you can now imagine that the sonar fibers, this muscle system, can pull on this capsular bag, but nothing will happen anymore. It's very stiff. So there is no accommodation possible anymore, and that's what you see here. The accommodation amplitude is going down to 0 after, say, half a year. Hintz and his group and co-workers described the EMT process in 2007. This is how it looks like. There are many cells actually which can become fibrotic. Fibroblasts are one of them, coming out of the fibrous tissue reaction. But we are here talking about epithelium, which is going into this process. And look at this cell. This cell is a very large cell now. It still has a kind of actin cytoskeleton, but most of the cytoskeleton is now alpha smooth muscle actin. And somewhere in 2010 or 11, I think, I made this confocal picture. This is the outgrowth of a lens epithelial cell on tissue culture plastic. This cell has a size of about 400 by 400 micrometer. It's a huge cell. And you can see now that the double staining here is very comparable to here. So we have the green, which is alpha SMA, the red, which is the normal actin, and you see all those elements in here. So this is a totally transformed cell. So actually the lens model is a very interesting cell to use as a model for fibrotic disease, as recognized by Wormstone and all. And this is also something that we want to study further. For the last 2 minutes I will use for 2 more slides which refer to what I was looking at, that I still have some open issues. I also want to show this one. This is one of the responses you can measure. So you see that the cells dense epithelial cells are forming a kind of fibrous strands going and finding their way into the silicon material. It looks two-dimensional, but this is in 3 dimensions. It was a very interesting feature, so you see a growth cone even here of these cells which are finding their way into the material. But it's also possible that you get this response. This is with hyaluronic acid as a filler. So hyaluronic acid is a very nice natural material, and the response is totally different. So this is indicating that with different materials we can generate different responses. And that's what we actually did with the in collaboration with another industrial partner, where they were making nanofiber systems. These nanofiber systems can be put into a kind of hydrogel. They are self-assembling, so they are like flat stones like this, which are stacking on top of each other. And this is how it looks like, the synthesis, and they can be equipped with these adhesion molecules that you find in laminin and collagen and so on. And we get a lot of different responses with different combinations of these adhesion molecules, also in different ratios. And one jumps out. I want to show you that one. That's this one. Here we are using with two motifs, ICFAF and YIXER from laminin. And they are offered in 2 different ratios, 1 to 0.5 and then the reverse 0.5 to 1. The green color refers to the alpha SMA expression and points to transformation of cells. And you see the difference here. And here there are a few, of course, there are a few cells which have been transforming in the culture system we used. But here there are many more. And this is giving me the confidence that we can, at some point, probably develop materials or hydrogel-like materials that offer the exact right amount in a right ratio of different adhesion motifs that can drive the epithelial cells into a quiet condition. And if that's the case, then you can indeed develop an injectable lens system, close it, and restore accommodation in the patient. So that's where I want to end. It's just in time, I hope. Thank you. Questions?
Yeah. Now we begin with a session of questions. Anyone?
Patrick has a question.
Yeah, thanks a lot. So you showed the bioluminescence and the CFU count on your window, right? And what I noticed was that the bioluminescence had several orders of magnitude difference, while the CFU count was only a factor of 3. So how do you correlate those 2 findings?
I know that so Jan Scholma has continued with this exactly this question, because that is indeed a kind of discrepancy that you see that. But it seems that under certain conditions the bacteria can still be alive, but they will somehow there is an impact on that particular bioluminescence system, so that the bioluminescence system is shutting down somehow. And he just told me last week that his PhD student has been able to show that in vitro as well now, that this is a possibility, that you still can have a high CFU count but low bioluminescence.
Did they then kick out the Cherry gene or something, or?
Well, somehow they have an inhibiting mechanism. It's not that they kick it out, I think. They are still able to bioluminescence again. But at that point in time, they somehow have a mechanism in place that it's not just not showing up.
They're adapting to fool you.
Maybe, yeah. But of course, we know from, for example, the biofilm shape of condition or the biofilm condition that these things are possible, that they can silence themselves a bit.
All right, thanks.
You're welcome.
Hi, Doctor. Real nice presentation. Talking about biomaterials, which do you consider are some adverse effects when talking about the inflammatory process?
Again, what
adverse effects could these biomaterials have when talking about this inflammatory process?
A very strong foreign body reaction would mean that there is a high activation of macrophages, and if macrophages are activated very much, then they could, for example, secrete many enzymes, even reactive oxygen species, that could then lead to local tissue damage. And if you have that situation, you end in a kind of vicious circle, because the tissue damage itself will also add up to the inflammatory response. So a high activation in a short time is a bad thing, I would say. If it's moderate or mild, then probably the body can cope with it.
Okay, and another
And the other thing is, if the outcome is that you have a very thick fibrous capsule in a situation where you cannot use it, that's also an adverse response.
Interesting. Now, can these biomaterials change or polarize the macrophage to an anti-inflammatory
well,
state?
In general, you see very typically that macrophages start in the beginning with the pro-inflammatory state, and at some point will switch to anti-inflammatory. And whether or not those are new macrophages or the same macrophages going to the M2 state, that's both possible. But of course, the time point at which this is happening can be quite different for different materials. And it's even possible in some cases that this M2 state is not even reached because of the chronic inflammatory situation. Yeah. But it's very highly dependent on the type of material when this is occurring. But usually there is such a transition.
Okay. Thank you.
And anyone has another question?
Good morning, doctor. So I was wondering if these polymers that you mentioned us could be altering the phagocytic capacity of these macrophages when they are uptaking the bacteria that you mentioned us.
I heard it quite.
Sorry, if these polymers could be affecting the phagocytic capacity of the macrophages when they are uptaking the bacteria on your study.
If the macrophages are taking up the bacteria or the material?
The material.
They will, I think. The PLGA mainly is a material that will break down at some point in time. So it becomes quite fragile if it's bulk degradation. So there will be fragments coming out, and we have seen instances where we find also that material inside macrophages. For the PEA, it's different a bit, because that is mainly going down by surface erosion. So it's much slower in small layers, more or less. So there you can expect, very likely at least, that it will be more at a molecular level. So they won't take up particles, but probably molecules.
Thank you so much.
There's one there.
Hi, thank you for the talk. And I was just wondering, instead of preventing the foreign body response and preventing fibrosis, have you worked with the other way around, triggering the foreign body response, for instance, for cancer treatments, or
I think it's going to be quite an exciting decade coming in. Of course, this cancer anti-cancer treatment is very promising. At the same time, you see in the biomaterials world that groups have focused now on what they call immune instructive biomaterials. That can be biomaterials that are going to instruct the immune response, for example, for a vaccine development or for an anti-cancer treatment, but of course very likely also for this local foreign body response, that it's going to instruct it in such a way that you will have a quick recovery towards this, for example, anti-inflammatory phenotype for the macrophages. So I really expect that there will be many more publications coming in the coming years exactly on this topic.
All right, thank you.
Thank you.
Thank you. So now we will go to the coffee break, and we will return to 11. But 5 minutes? Okay. Nos vemos a las 11:05.
Okay, now we will resume with Dr. Saleh. So, Dr. Muhammad Ali Shavasi is a prestigious researcher at the University of Groningen in the Department of Biomedical Engineering. Dr. Shavasi received his PhD from the University of Helsinki, Finland, and has more than 12 years of experience in cancer therapy and tissue regeneration at UMMCG. Within his impressive professional and academic career, he received various awards, including the Best PhD from the University of Helsinki and the Best PhD thesis in Pharmaceutical Sciences in Finland in 2015, as well as the Young Researchers Award in Pharmaceutical Sciences at the 20th RASI Festival, and was laureate of the prestigious USARN Award in 2020. Currently, he serves as an editorial board and reviewer for world-renowned journals. He is co-founder of the company Capsamedics OI, as well as the spin-off company Medix Micro, both of which are involved in the development of 3D printed transnational dental adhesive devices and microneedling. Respectively, Dr. Muhammad Ali's core research topics include nanotechnology-based regenerative hydrogels, photothermal materials, scaffolds, and microneedles for wound healing, as well as bone repair and localized treatment delivery. Please welcome Dr. Saleh.
Hello, everyone. Thank you very much for the nice introduction. I'm very happy to be here. And today my talk is a bit different from what I did on Monday about photothermal nanomaterials and biomaterials for tissue engineering and cancer therapy. That was mainly a, let's say, conference talk. And today we have more like a teaching course and teaching talk today about the application of biomaterials for wound healing. And here I will explain you why we really need these biomaterials to accelerate the healing process of wounds in different parts of the body. Usually, when we talk about wounds, it comes to my mind that we mainly speak about skin wounds, that in many cases researchers are mainly focusing on this topic, making different bioprinting, hydrogels, scaffolds, microneedles that I will talk about them today, injectable hydrogels, electrospun patches for wounds that are on the skin. But besides that, we have some new fields that unfortunately today, due to the lack of time, I think 2 hours was not enough to talk about them. But at the beginning, I would like to let you know that there are also a lot of works going on the application of biomaterials for internal wounds, like post-surgical injuries and wounds that we have in the abdominal area after surgeries. There are a lot of injectable hydrogels, electrospinning patches, and so on that during the surgery, even for heart surgery, patches that can stick to the damaged tissue for improving the regeneration. So mainly today I will talk about wounds on the skin. But please consider that this is just an example, and the same technologies can be also applied for internal wounds. Let's start with this slide and ask you several questions at the beginning. I would like to know how many of you have seen a newborn or infant baby just born healthy, like healthy newborn babies. How many of you have seen a baby totally healthy? Just raise your hand. Almost all of you have seen. So it means that most of the newborn babies are healthy. But how many of you have seen a newborn baby that has some organ dysfunctions or problems in one or several organs? Still, really? Interesting. But I expect that maybe you have seen, but the number of the babies that are born with a problem or organ dysfunction are much less, like most of us that we were born healthy. So this is not a big issue, but the main issue is related to the next two photos that I have here. How we can remain our body and spirit active when we are adult, and how we can live long and stay healthy. This is actually two main problems for most of us, and we have experienced problems and diseases in different tissues and in different organs, injuries. Most probably most of us has experienced surgeries which bring a lot of organ dysfunctions and pain to us. And that's why basically we are working on tissue engineering, because as you see here in this slide, there are many reasons that results in organ dysfunction or different diseases over time. For example, our lifestyle, the job that we have, most of the time we need to sit. We have several surgeries maybe during our life. Accidents are very common nowadays because of the huge number of cars that are in the street. Many days we don't eat healthy foods, and over time, if we do a lot like this, anyway, problems and diseases and organ dysfunctions will appear sooner or later. That's why, as a scientist, we are working on tissue engineering field, but I would like to know which one of you, or who knows how we got inspired to work on tissue engineering, how the idea came. Yeah? Just exactly, from the nature. If you look at these animals that are just some examples, you see that there are a lot of examples in the nature, and different animals actually can regenerate different parts of their body. For example, in the case of salamander, it can almost regenerate any tissues, eyes, tails, heart, brain, kidney. And we know that in our body, still there are some organs like kidney that can be regenerated automatically in the body, but brain or cardiac regeneration is extremely challenging because we don't have enough stem cells there in the cardiac tissue. And that's why, for example, Professor Santos, during the past 5-6 years, is highly focusing on cardiac regeneration because of the challenges that exist and we have there. Or in the case of deer, it can easily regenerate antlers every year. Zebrafish can regenerate, again, the heart tissue. Brown bat can regenerate part of the wing. And as you see here, sea star can regenerate its limbs automatically without any problem if it's lost. But on top of that, as I mentioned, in our body also we have one clear example, which is liver, that even 1/3 of the liver can end up and turns to a complete liver. So if we have some liver damage, it's not a big issue. It can recover by itself. But for many organs, this is not the case. And tissue engineering can really play a significant role here. And we as a scientist, we can work on different types of 3D networks that are safe for the body. There is no foreign body reaction. That is the main field of Professor Tove and Koten. And then we can apply it to regenerate the defective tissues. This is a real example that I really like, and in almost all my presentations and lectures, I showed this real example to the student. This is this girl, the name is Hannah. She's still alive, but when she was born in 2013, doctors realized that she cannot breathe very well because she didn't have a perfect windpipe. That's why right after her born, scientists and researchers started to make a 3D printed windpipe, and then they separated the stem cells from the patient. That was Hannah, cultured the stem cells on this 3D windpipe. It was very well going on. The stem cells could grow there, and then they implanted this windpipe to the Hannah, and she's still alive, which is really great. This shows the importance of our field and how we can help patients in different ways to make them live better and without any pain or problem. This is also one of the pioneering works in the field of cardiac regeneration or cardiac therapy. Professor Taylor is one of the pioneers in the University of Minnesota who was, yeah? Sorry? Yeah? No, there is no, just one time they implanted and it works. Forever. Yes. Yeah? So Professor Taylor started to work on making a real heart, and her idea was first to get a heart from a dead person, use different detergents to remove all the cells from the connective tissue, and what you see here, the white heart that you see here is basically the extracellular matrix of the heart without any cells, anything. And then she started to separate the stem cells from the patients, from the cardiac tissues cultured on this extracellular matrix of the heart, and finally she could get a heart that can beat. Of course, there are different challenges, a lot of challenges to move to the clinic, but as a starting point, this is really amazing, you know, to fabricate something that can basically survive many patients that have cardiac diseases. So when we talk about tissue engineering, there are some extremely important elements and factors that we need to consider to include and put together to make a successful tissue engineering approach or strategy. There are mainly three different elements that we need to put together. The one here you see is a scaffold, which is the backbone that we need for cell attachment and growth. The second one is bioactive factors. We need different kinds of proteins, peptides or biomolecules to induce the differentiations of the cells that we have inside the scaffolds, and obviously, as I mentioned, cells. It means that the third element is cells. So we need to have on the scaffolds both biomolecules and the cells, and then implant it to the body for tissue regeneration applications. But as a scientist in tissue engineering field, like many different researchers around the world, we are working on some polymers or scaffolds or materials that can be used to skip these two elements. So we can design some scaffolds that by themselves, without using any biomolecules, can induce the differentiation of stem cells. There are already some polymers that can do this job, and we can make scaffolds out of those polymers. Or we have some polymers that can recreate that can recreate stem cells or the cells that we need for tissue regeneration. So there is no need to initially in the lab culture the cells inside the scaffold, but when they are implanted to the human body, they can recreate the cells, and then we can get the regeneration. So by doing this, the final idea is what I show here, to go from replacements of the tissues to regeneration of tissues to their original function. So we don't need anymore to replace tissues or organs, but instead we can make some systems to make them really working like an original and healthy tissue or organ. But of course, for that we need to consider that there are several important factors during the fabrication process. The first one is to consider using materials that are really biocompatible. By biocompatibility, I mean that they don't induce any toxic effect on the organ that they are implanted or to the human body. I mean, even when they degrade, those degraded parts cannot go to other organs or tissues to make some side effects. This means biocompatibility. The next important factor is the mechanical property that should mimic the real mechanical property of the host tissue. This is again one of the big topics that my colleague and friend, Professor Prashant, is highly working on, the mechanical property of different materials. Why this is important? Because for sure you know that the mechanical property of the bone tissue is totally different from the mechanical property of the skin or mechanical property of the hard tissue. And the cells and our body is really smart and clever. And one of the factors that affect the regeneration and differentiation of the cells is mechanical property of that. How they are under different pressures defines how they differentiate. The next factor is the ECM mimicking that we need. ECM means extracellular matrix, or let's say the collagen that we have in our body as extracellular matrix, that our scaffold should really be able to mimic the ECM of the body. Again, this is important because cells are very clever. They realize what is the geometry orientation of the scaffold, what is the pore size, and based on that, they decide how to attach and distribute inside the scaffold. Mechanical property is 2 times here, so I skip this one. The next one is the surface chemistry and texture of the scaffold. That again is the focus of research of Professor Patrick in our department, and he made a great talk about that yesterday, and I'm sure that today he will also talk more about the texture and surface chemistry and its importance in tissue engineering. But just to shortly mention, we know that, for example, if on the surface we have amine groups for the scaffolds, it might induce high toxicity to the cells. The cells, they don't like to attach there and grow. But if we have some proteins or peptides like RGD, then they can really easily attach there and grow. The next important factor is interconnected porous network for the scaffolds. And does anyone know why this is important? Why it's important to have interconnected pores? Of course, we can have pores that might be separated, not connected, but having interconnected pores in the scaffold is extremely important. Does anyone know why? Exactly, thank you. We need actually the cells to penetrate to all parts of the scaffolds for proper tissue regeneration. If they just to penetrate to part of the scaffolds, or if we just have interconnected pores around the corners of the scaffold but not really inside, we cannot get a proper tissue regeneration. Thank you very much for that. The next one is to avoid immune cell responses and host body reactions that I'm sure by now you know a lot about that. The other factor that we need to consider is how fast we can get the scaffold resorbed in the body. Because for tissue engineering, at the same time that the cells migrate and grow, over time we need to get the degradation of the scaffold to make really all the cells and connective tissues that we need in the defect area. If the scaffold remained for very, very long time, for example, in the case of bone, ideally we need 1 to 2 months the scaffold to stay there. But if it stays there for 1 year, it means that we cannot have a real bone tissue because the cells cannot really expand and proliferate in the defect area, yes? So we need to have a combination of, and a mix of, let's say, everything. We need to have some biomolecules or just the scaffold by itself to induce the cell proliferation and differentiation. And at the end, one of the main factors that most of the researchers, including myself, actually ignore, we don't pay enough attention to this, is that how easy and cheap we can produce these scaffolds for tissue engineering. We are doing a lot of fancy and complex systems, but which is good, of course, for moving forward and pushing science to move forward. But when we go to the companies and we ask, okay, we have this system that is multifunctional, it can induce blood vessel formation, it can suppress the immune cells, blah, blah, blah. The first question that might ask is that how cheap it is. What is the materials that you use? What is the process? And if we show that we have a complex process to fabricate these hydrogels or scaffolds, then definitely it will not go to the market. So this was an introduction about the importance of the tissue engineering, how and what parameters we need to consider to have a successful regeneration of the tissue. And since the main topic of today is the regeneration of the skin, of course, if we want to regenerate as a researcher one tissue or organ, first thing that we need to know is what is the anatomy of that organ, no? Because based on that, we design our regenerative system. In the case of skin, we have mainly 3 different layers in the skin: epidermis, dermis, and subcutaneous tissue. And as you see here, the epidermis layer is covered with stratum corneum, that the main function is to protect our body as a barrier and doesn't let the bacteria or different microorganisms to penetrate to the skin. And the second function is to keep the moisture of our skin tissue. It doesn't let the evaporation from the lower part of the tissue. That's why our skin is always fresh and flexible. The second layer is dermal matrix that is composed of collagen, elastin, fibronectin. There are a lot of fibroblasts there. We have nerves there and also capillaries. And the third layer is subcutaneous tissue that is mainly made of fats, fibroblasts, again, blood vessels, and sweat glands. And you see that by this image, the skin is already a very complex system. And if we want to do a proper regeneration, we need to have a lot of consideration to make it really work. But now again, a question. Why really we need to do regeneration of the damaged skin? Why it's crucial to do? What do you think? I mean, if you want to compare with other organs, maybe some people have less interest to do that. But yeah, please. Yeah, exactly. Thank you very much. You are really good students, and we were just discussing yesterday about all of you and how motivated you are and how good you are. True, because that's the first barrier of our body that protects our body from external environment, yes? And the surface area of the skin is really large. And if you have some damage, there is a high risk of virus infection going to the blood circulation, sepsis, and that's why a lot of people in the hospital die, for example, because of sepsis infection, yeah, which is a hidden killer basically in the hospitals. So for wound healing process, we have 4 different phases that I show here in this schematic, as you see. The first phase is hemostasis and coagulation. That is by the migration of platelets to the damaged area to stop the bleeding. The second step is inflammation, and in this step, a lot of neutrophils and macrophages move to the damaged tissue. This is the second phase, and I'm pretty sure you know why, because it's an open damaged tissue. There is a high risk of infection, and our body is very smart. Since there is a risk of infection, neutrophils, of course, there is a cascade and a lot of biomolecules involved in this mechanism. But anyway, at the end, we get a lot of neutrophils and macrophages there to protect our body as much as they can. The next step is proliferation. And by proliferation, we mean that a lot of keratinocytes from the edge of the wound migrate to inside, and then fibroblasts will be proliferated, and we have angiogenesis and blood vessel formation in this phase, during the proliferation phase, which is actually the main phase to get wound closure. And then at the end, after wound closure, we have remodeling under the wound area in this part, which means a lot of collagens will accumulate to the real structure that gives us intact skin without scar tissue and without fibrosis, okay? But there is a high risk that we get delayed in this process, and if we get delayed in the proliferation phase, we will get at the end the scar tissue, which is not nice, especially if it's somewhere like in the face, yes? So why do you think this might happen? How we can get a delay in this process of the proliferation? What can be the reason for delayed proliferation phase? Yes, please. Someone raise hand? No, not you? Yeah, please. Size of the wound actually define how fast it can be healed. But delay is different from how fast it can be healed. Age can be a parameter, a general parameter, of course, yes. Yeah, you?
A reduction in nutrients, and also if we have no vascular, a correct vascular proliferation in the area.
Okay, so you mean that low blood vessel formation, yeah?
Yes.
Yeah, this can be a good reason for that. Anyone else? Yes? Yeah. This is actually the right answer, that the consequence is all that you talked, like lack of blood vessel formation. It comes from prolonged inflammation there. Basically, the step before that. If we get prolonged inflammation in the wound area, then the proliferation phase would be delayed. Okay? And I show it here. Actually, prolonged inflammation phase results in the delayed repair. We cannot have a normal repair of the wound tissue. And there are several factors that can induce this inflammation phase to be prolonged. The first one is infection. If we get infection in the wound tissue, then this pathogen does not allow proper proliferation because of the inflammation. If we have poor local vasculature there, excessive level of pro-inflammatory cytokines in the wound tissue, protease enzyme, and at the end, reactive oxygen species, ROS. These are the main factors that prolong the inflammation. And this is a big challenge for patients that are diabetics, they have malignancies, they have immunosuppression, and they have vasculopathy. Yeah? And that's why in the clinic, there are a lot of efforts, especially for these patients, to avoid chronic wounds in the patients and do a lot of therapies to them to make sure that there is no like leg amputation or hand amputation in these patients. There are several clinical treatments at the moment in the clinic. One of them, of course, is antibiotics to avoid infection. There are some protocols to change dressings on the wound. For proper healing, there are operative debridements. In the clinic, by debridements, I mean cleaning of the wound tissue and the removal of the bacteria and dead cells. This is very important. If we have a lot of dead cells in the wound area, it will delay the treatment because the space is occupied with the dead cells and then new healthy cells cannot grow and proliferate there. So the protocol is called debridement, that there are some sponges or foams in the clinic that they do this. Okay, and the final one that is used is flap repair. It means that if there are some large wounds in these patients, some parts of the skin from healthy areas are detached, and then they place it in the defect area. These are normal protocols in the clinic for these patients, but besides that, we have adjuvant therapies. Remember, they are just adjuvant therapies. They are not the main therapies. If we just do this as a monotherapy, they will not work. They are adjuvant therapies. One of them is platelet-rich plasma that you see here. What does it mean? In this procedure, clinicians take blood samples from the patients. They do centrifuge to separate red blood cells. And then we have a lot of platelets in this area and some platelets in this area. And then they try to concentrate them and get platelet-rich plasma. Why would they do that? Because platelet is full of anti-inflammatory biomolecules, different miRNAs, proteins that all of them together can do angiogenesis, can do anti-inflammatory responses, and many other things that are not known. And that's why we use it in the clinic. The next one is HBOT, hyperbaric oxygen therapy. You see these patients with big wounds that is covered, but definitely it was a big wound. That's why they did HBOT for this patient. And with HBOT, the defect area will go to a chamber that you see here in this picture that is 100% of oxygen in a pressure above normal. Why the pressure is above normal? What do you think? And why oxygen is needed? Why they put the wound area in 100% oxygen and with higher level of pressure? Any ideas? Yes, please.
Under hypo-oxygen conditions. So this level of oxygen could allow to kill that bacteria that grows.
Or at least stop the proliferation of the bacteria. Yes, thank you very much. This is one of the reasons. Any other reason comes to your mind? Okay, I help you. The other reason is that in 100% oxygen and high pressure, especially if it's high pressure, we have high chance to get more oxygen to the defect tissue. Yes? When the pressure is higher. And with higher oxygen delivery, the proliferation of the cells in the damaged area would be much more. The next approach is negative pressure assistant therapy. What does it mean? In this technology, there is a suction pump, there are tubings, and then a dressing that is connected to the suction pump. And here the idea is that this suction pump can suck all the dead cells, exudates, and the fluids that are in the defect area to make space for the growth and proliferations of the new cells. Okay? And there are also some more advanced technologies that here we have two tubes. One is for input of biomolecules or buffers for cleaning, and the other one for the exit of the exudates. So we have very dynamic system there in the wound area. And this helps the proliferation of the cells and accelerated wound repair. So besides this, here I would like to show you the function of conventional dressings. With conventional dressings, I mean, here we have the wound and we have a dressing there that can do two jobs there. One is to seal the wound area and doesn't let the bacteria or viruses to penetrate. This is one of the functions of the traditional dressings. And the second one is that it can keep the moisture in the wound area. These are two main functions of traditional wound dressings. And we have some examples already in the market. Tegaderm is one of those to do this job, or more traditional one, of course, you know, is cotton gas there. Yeah? So, besides this, we have also a lot of creams, ointments that are antibacterial in the market. But now, with different technologies that we have in biomedical engineering, a lot of researchers working on biomaterial sciences, then they have decided to push the technology forward. For example, what we do in our lab is the fabrication of different microneedles for wound healing applications, making electroresponse patches that are made of really tiny nano-sized fibers that have a very big and large surface area that can mimic the ECM. We have injectable hydrogels, bioprinting to make very nice design and orderly designed structures from the gels for implantation. These are new technologies in the market that a lot of research going on in these fields. And to let you know the importance of the field, here are some values for advanced products that are already in the market. As you see here, in 2020 was about $12 billion in the market of wound treatment with new technologies and advanced materials. And it's expected to reach to 18.7 billion in 2027. Yeah? So this is really important. So we have many different new approaches to improve the regenerations of the wound area. How much is it so far that I have talked? Sorry? Okay, great. So we have different technologies and strategies for wound healing, and I give you examples one by one. What are those? The main ones, of course. The first one that I would like to talk about it is the oxygen-generating biomaterials. Some biomaterials or hydrogels or formulations that can be placed on the wound area and they can generate oxygen. So it can be an alternative to what I showed you here. Yes? So instead of having such a big device, having the patients here and a clinician expert to do this, it can be a personalized or a formulation that the patient can use it by himself or herself. This is the benefit of oxygen generating biomaterials for wound healing applications. This is one example. Hyaluronic acid and polydopamine were mixed together to make a hydrogel. I don't go to the details of the hydrogel and mainly focus on this part. They then incorporate calcium peroxide there plus ICG. Calcium peroxide is there because of this reason. It can react with water that we have in the wound area and produce hydrogen peroxide. But we know that the hydrogen peroxide, low level of hydrogen peroxide is actually good for the regeneration, very low level. But we know that if it goes to the high levels in the wound area, it has negative effect. It kills the cells, it induces inflammatory responses and so on. So to avoid this process and the increase of the H2O2 in the wound area, after this step, this H2O2 can convert to oxygen. That's why we call it oxygen-generating biomaterials. How? Due to the presence of MnO2. This is one of the functions of MnO2 that we call it chemodynamic therapy, basically. Okay? It can convert hydrogen peroxides to oxygen. So we can increase the level of oxygen by this mechanism in the wound area. And inside this system, there was also ICG, which is a molecule for photothermal therapy that I discussed on Monday. So I don't give more details about that, but just to let you know that they can do multifunctional job, increasing the temperature to 40 degrees for wound healing and also oxygen generation. This is a multifunctional system. And to see how this oxygen generation and photothermal therapy can affect anti-inflammatory responses, TGF-beta expression in the wound tissue was imaged and screened. And you see that in 3 different groups, that the group 1 is the hydrogel without anything. Group 2 is the hydrogel plus carbon peroxide. And group 3 is the hydrogel CaO2 plus photothermal therapy. And you see that the expression of the TGF-beta as an indicative of anti-inflammatory responses has been increased in these 2 groups, yeah? Which means that we can get better regeneration of the damaged tissue. The next strategy is antibacterial approaches. Of course, we have some ointments and creams in the market with antibacterial properties, but patients need to use it in different intervals, like every 4 hours or every 8 hours, that reduce the compliance of the patients. That's why we develop wound dressings or hydrogels that are antibacterial. Why we do this, why antibacterial property is important? Because of these 2 reasons. The first one is that in many cases, the infected wounds have more than 1 bacterial species. Usually, Staphylococcus aureus and Pseudomonas aeruginosa are the main ones that infect the wounds. And the second one is the presence of drug-resistant bacteria and biofilm formation. One example is methicillin-resistant Staphylococcus aureus, MRSA. Because of these reasons, we try to develop antibacterial dressings that, of course, this is one therapeutic approach, but can be combined with other technologies to give multifunctionality. And there are common strategies to stop bacterial colonization in the wound area. These are 3 main common strategies for that. One is to limit the formation of new bacteria. The second one is to remove the bacteria that are formed in the defect tissue. And the third one is to kill the bacteria that are present in the wound area. Instead of using antibiotics, that we know that we have a global challenge with the resistance of the bacteria to antibiotics. Now scientists are switching to find out some polymers that they can induce antibacterial properties. They can induce the killing of the bacteria in the wound tissue. One example is chitosan. It doesn't have a very potent antibacterial properties, but if we modify it with different functionalities and the polymer change, we can enhance the antibacterial property of the chitosan. For example, in this study, this quaternized chitosan was developed that has amine group, OH group, and this functionality that you see here. Polyethylene glycols with 2 aldehyde groups on 2 ends were also used. These 2 were combined. And by interaction between aldehyde groups and amine groups, we can form a nice hydrogel that is self-healable by shift-based reaction. And these functionalities were shown that has very potent antibacterial properties. So we don't need to use any antibacterial agent inside. And then this hydrogel were used and studied for biomechanical properties and self-healing. And you see here that storage modulus and young modulus were checked in 2 different strains in different intervals, 10% and 250%. And you see that by changing the strain, the mechanical property changes, the storage modulus significantly drops, and then by again stopping this high strain and switching back to 10%, again we have the gel properties. It means that the gel is really self-healable and we can apply it for wound healing application. And here you see antibacterial assays that were done in these studies, the control group with a lot of bacteria in the agar plate and intact healthy bacteria. But when we have these 3 different samples with different amounts of this QCS, this modified chitosan, you see that the growth of the bacteria is much less and they are not healthy. And actually there is no bacteria in this one.
Ali, one question here. What is the mechanism of the antibacterial activity of the chitosan? Is a membrane active, or?
Basically, they destroy the membrane integrity.
So it's like this amphiphile peptide or something that interact with membrane, forming pores?
Amphiphilicity is one of the things. The second thing is that we have also a negative charge in the cell membrane, and this negative charge can interact with the positive charge of the amine group, and because of that they can lose the real integrity, and then the bacteria will die. Yeah? So the next example for antibacterial dressings is the usage of metal ions and metal-based nanomaterials. So instead of antibacterial polymers, we can also use metal ions and metal-based nanoparticles. And studies shows that there is a broad spectrum of antibacterial effect for these ions because, yeah, please. PEGDA?
Yeah,
PEGDA is just for the gel formation. It has two aldehyde groups in two ends that can react with the chitosan functional groups to make the hydrogel. But the killing effect comes from the chitosan.
It's for the cross-linking?
Yes, exactly.
Thank you.
Yes, please.
Antibacterial activity of chitosan depends on the origin, shells and fungi.
Not usually, because at the end you have chitosan. The chemical structure is chitosan. The chemical formula is same, no matter what is the source. Okay? Yeah. Yes, exactly. Any other questions so far? No? Okay, we continue. So this is the next one, metal ions. This is one of the studies that we did, very short study as a proof of concept. We use a natural polymer called farsigam, and we combine it with AgNO3 as a precursor for the fabrication of silver nanoparticles. And we showed that this farsigam can be a reducing agent to convert AgNO3 to silver nanoparticles and also stabilize that. So just by mixing these two, AgNO3 and farsigam, we can produce silver nanoparticles, and then we applied it as a sprayable hydrogel on the wound. Interestingly, we observed that for these two different species of bacteria, it can effectively kill the bacteria very efficiently. And here you see the MIC of this system for these two bacteria, which were basically 1.1 and 2.2 for S. aureus and P. aerugineza, respectively. Okay? So what is the mechanism here? You see, breaking disulfide bonds in the proteins of the bacteria. This is the main mechanism, antibacterial mechanism of most of these metal ion based nanomaterials for wound healing applications and antibacterial property that we can get out of it. This is another example. The previous one was with the silver. This one is with copper-based antibacterials that are fabricated and stabilized in the presence of bovine serum albumin, BSA, with this approach that you see here, combination of copper nitrate, Na2S as oxidative agents, and at high temperature to accelerate fast production of the nanomaterials, and after formation they can be applied in the wound area, and definitely we can get here because of the CUS antibacterial effect. But guess what is the antibacterial mechanism? What is your idea? Why we get and how we get the antibacterial effect? I can help you. Here you see this wavelength, and here you see the laser. So what is one of the mechanisms? Yes, hyperthermia, or let's say photothermal therapy, because by increasing the temperature we can kill the bacteria. But this is not the only one. There is also one more mechanism here. Sorry? No. More or less, it can be. But anyway, as a continuation of what you said, in the previous slide I talked about silver ions as well. Here the release of copper ions is very important for antibacterial properties. Basically, studies shows that when we apply near infrared light, in addition to hyperthermia, killing of the bacteria, we can get accelerated release of copper ions. So it's not just a matter of copper nanoparticle, copper sulfate nanoparticle by itself. Of course, it has antibacterial properties. But with photothermal therapy, we can increase the release of the ions. And the release of ions at the end results in what I said here, breaking disulfide binds of the bacteria. This is the other mechanism that we have. So I showed you the loading of copper sulfide, the load of, or let's say in situ forming of the silver nanoparticles with polymers to make a gel for antibacterial properties. But besides this, we can also load many different types of antibacterial or antibiotics inside these hydrogel systems for wound healing applications. And even we can use these hydrogels or patches to get sustained release of these antibacterial agents, no matter it's an ion or it's an antibiotic. And this is a huge benefit of hydrogels or dressings that we have in the market or under development. Why? I ask why. I will not answer myself. The tonation was different. Sorry. Can someone talk about this graph? Do we have a pharmacist here? Who is the? Many. So pharmacists should know very well this. Maybe in the second or third year of pharmacy, you study this, and then later in biopharmacy, no? Yeah, please. Mm-hmm.
After some time, the concentration will lower, and then we have to keep once again the drop in order to keep those that effects in mind. So that's the up and down that we see in a kind of solve.
Yeah.
Figure. But in the red line, we have a control release where we have sustained active effect because the concentration is just stabilized in a level that it still gives effect.
Okay, thank you very much. Thanks, but let me, because we have just less than 1 hour left, let me complete what you said, and sorry others, I know that you wanted to add more, but I just shortly explain more this. Actually, by giving you an example, why ointments that are in the market, clinicians and specialists says, okay, this patient should oops, thank you, sorry. This patient should use it in 8-hour intervals or 6-hour intervals. Why? Because at the beginning that we apply it on the skin, the concentration of the drug molecule can increase to reach to this window that we call it therapeutic window, this area, that we have a minimum effective dose and maximum effective dose. In this area, we call it therapeutic window. So for certain times, period of time, we have the level of the drug molecule in the therapeutic window. But even if it goes higher, then we have toxic effects, more than that, okay? So to the certain time, it increases and then decreases. By decreasing, at some point, it's still in the therapeutic window, but we reach, after a time, to concentrations that are not therapeutically effective. And then that's why this can be 6 hours or 8 hours later, and the doctor says, okay, this is the time for the next usage of the medicine. And this is the case for tablets, capsules, whatever that we have. Yeah? So this is the story that currently we have, but with these dressings and hydrogels, the idea is to get a single dose. The concentration in the wound area increases very fast, and it reaches to a level that is inside the therapeutic window, and for a long time it will stay there, remain there constantly. And with this approach, we have less intervals of changing the patch or hydrogel, and we can get better therapeutic response. Yeah? Besides that, another benefit is with these systems, we can control the release of the drug molecule. This is another benefit. We can have these kinds of trends for very fast therapeutic effect, a bit slower, zero-order drug release, which means that the release of the drug molecules from the dressings or hydrogel is not dependent on the concentration of the drug over time that is inside the hydrogel, which is basically the best release trend that we can get. All researchers are trying to get something like this. We can also combine different drugs or even one single drug that release at the beginning fast and then later with sustained release. We can have delayed release or pulsatile release. So different trends based on what kind of polymers, what kinds of strategies, what is inside the dressings or hydrogels or scaffolds, we can get different release patterns. But imagine in the case of hydrogels or different dressings that we have or we are working on that for wound healing, what are the main factors that play on the control of the release rate? How do you think we can control the release rate of the drug molecule from these 3D networks? What are the parameters that affect? Porosity is one, but how we can control the porosity? Do you have any idea? Excellent. Yes, the concentration that we use of the polymers can have a remarkable effect on the trend of the release. Imagine in the certain volume, we use 1% or we use 2% of polymers. This means that we can get different porosities. Less porosity means sustained or a slower drug release. Yes? This is one factor. Any other factor? Do you have any idea? Yeah. Thank you very much. Yeah, composition is also another thing. Hydrophilic polymers, hydrophobic polymers. What is the degree of hydrophilicity? All this, which is related to the composition, can define the release rate of the drug molecule. If we have, for example, the chitosan and with some surface modification of the polymer, or let's say polymer modification, we make it more hydrophobic or we make it more hydrophilic. With this, we can control the release. Another thing is, sorry? Temperature and pressure can be after fabrication, basically, yes? This is the one factor that is post-fabrication. But if we want to talk just about the fabrication parameters, plus these two, the amount or concentration of the cross-linker that we use is another factor that can play a key role in the release rate. These are basically the main parameters that we can play with to change the release of the biomolecules or drugs from the hydrogen. The third approach, I talked about debridements and what does it mean. In the clinic, we have these kinds of foams, as you see in this picture, that are used for removal of the bacteria, dead cells, exudates, and so on. So we get more space for healthy cells to grow. This is commonly used in the clinic for the patients. But besides this, there are also hydrogels and dressings that can be used instead of regular usage of wound debridements that are in the clinic, these kinds of foams. Okay, hydrogels can be used. So I have several questions here for you. The first one is how hydrogels can work as a debridement. What do you think? Have you heard about the swelling property of different materials or hydrogels? So it plays a key role there again, okay? Hydrogels, of course, they have by themselves a lot of water inside. That's why we call it hydrogel, because it's a mixture of polymer plus water or plus buffer. But it depends how much water or solvent we use to fabricate those hydrogels. Yeah? And still, after making the hydrogels, most of them, they have a very good potential to absorb more water. And if they are in the wound tissue, they can absorb exudates from the wound area. That's why and how they can work as a debridement. Thank you very much. So this is the answer. Rehydration and suction of the dead and damaged cells. Yeah? The next question. Imagine there is a wound with substantial exudation, a lot of secretions and fluid in the wound tissue. Can we still use hydrogels? Of course we can use, but it will not be very efficient. We can always use. Yeah, if we have, it depends on the type of the polymer that we use for the hydrogel, but usually hydrogels are used for wounds that they don't have so much exudation. Yeah? If it's a little, hydrogels can work very well. But if we have high level of exudation, then hydrogels are not good, because in many cases, absorption capacity is limited. Yeah? The next question. For these kinds of wound, what do you suggest? What should we do? Imagine you are a clinician or a researcher in this field, and a patient comes to you and asks for a solution. We usually have these common connections between researchers and patients in UMCG. They come and the faculty or UMCG organize some events with the patients that they talk about the challenges that they observe during the therapy process. Okay? Imagine the patient comes and raise this issue. What's your suggestion? What can we do? Mm-hmm. Basically, you will look for other alternatives. Yes? True. That's true. You need to have some materials that have high capacity of water absorption. That's the first thing that you need to do. So make your hydrogel with polymers that have very high capacity of water absorption. And the examples are carboxymethyl cellulose and polyurethane. These are the main polymers that can be used for this case. Yeah? The second one is to use some technologies or dressings that are in the dry form, not hydrogen. Yeah? If you use electroresponse patch or something else that is in the dry form, then you have high potential of absorbing more water or exudates from the wound tissue. The fourth mechanism for the fabrication of the multifunctional wound dressings is to incorporate antioxidant reagents or molecules there. So antioxidative dressings can work very well for wound repair. Why? Because in the wound area, we have mainly these three rust reagents, super oxide anions, hydrogen peroxide, and hydroxyl radicals, that they induce these two undesirable effect, let's say. The first one is significant damage to the cells. How they do that? By this mechanism. They induce membrane damage, lipid peroxidation that basically induce this membrane damage, and they decrease the antioxidant biomolecules that we have in the cells, and finally they make DNA breakage and oxidative damage that end up to apoptotic cells or necrotic cells. Yeah? This is one bad effect of this ROS. And the second one is that some studies have shown that this ROS production can impair angiogenesis. If we have a lot of ROS in the wound area, the blood vessel formation would be much less than same wound with the same characteristic but without ROS. Yeah? So let's go to the example. This is again another hydrogel made of metacrylated chitosan, which is connected or conjugated with gallic acid. Gallic acid is a phenol molecule that we have in many different types of plants. And in herbal medicine, it's used a lot as an antioxidative agent. So this one is conjugated to metacrylated chitosan and chlorhexidine as an antibacterial agent, plus this polyronic F127 that again works as an antibacterial agent. Together were incorporated inside the hydrogel for two purposes. One, to reduce the generation of ROS using gallic acid, and the second one is the antibacterial property of this compound here. So I show here the main results. You see that this is an experiment called DPPH that is for checking the antioxidant property of materials. DPPH is a free radical, and here you see that by using and treating this DPPH that has the original color of purple with the main formulations. This one is chitosan, metacrylated chitosan with gallic acid, and these three are metacrylated chitosan with gallic acid plus FC, which means this F127, and chlorhexidine, basically the antibacterial agents. And here we checked all because we want to show that this antibacterial agents doesn't have any negative effect on antioxidative property of gallic acid. And in all cases, you see that the color change means that we don't have free radicals anymore there, and the scavenging effect is near 100, more than 95% there for all the formulation compared to the polymer alone without gallic acid. So this approves the antioxidant properties. This scratch assay that cells are grown on the plate, and then you make a scratch and you treat the samples or cells with different formulation, shows migration of the cells and their proliferation. And again, here you see that when gallic acid is there in these two cases, the migration was much more than the other samples because of the antioxidant properties. So the cells are more healthy and can proliferate and grow. And in the next step, the expression of VEGF and CD31, that are two main markers of angiogenesis expression and angiogenesis in the wound area, were checked. And again, here you see that for the final formulation, the expression of angiogenetic markers are significantly increased. Why this test has been done? Because here I told you that ROS can impair angiogenesis. So by having something antioxidant, we can increase the angiogenesis rate. Yeah? Let's go to the fifth mechanism for wound healing, that is immunoregulation, that has a key role in wound healing and tissue regeneration. If you remember from the earliest slides, I told you that the proliferation phase of the wound treatment was blocked or delayed because of the inflammation. Yes? So let's go to more details. We have two key immune cells here, neutrophils and macrophages. Neutrophils has two main jobs in the wound area. The first one is to release antibacterial substances and molecules to kill the bacteria so we don't get infection in the blood circulation. And the second one is that it can release different biomolecules that can promote angiogenesis. So this is a good thing. Yeah? So in the normal process, without any treatments, neutrophils can do this job, which is very good for the next step. and the proliferation phase. Basically, proliferation phase is started with this mechanism by neutrophils. The second immune cell is macrophage that has phagocytic effect. It can phagocytize bacteria and debris in the wound tissue. To let you know, we have two different types of macrophages, M1 and M2. M1 is pro-inflammatory or inflammatory macrophage, which the population is a lot in the wound area because of this defensive system and doing phagocytosis. It's done basically with M1 macrophages, which is inflammatory. And M2 is anti-inflammatory. And the effect is totally different. If we have M1, a lot of M1 macrophages in the wound area, it suppresses a lot the regenerative phase and proliferation phase. But on the other hand, if we have M2 macrophages, it's very good for tissue repair. Because this M2 macrophages, again, can induce angiogenesis and induce the proliferation of fibroblasts. So the focus is to shift M1 macrophages in tissue engineering studies. This is a hot topic, to shift M1 macrophages to M2 macrophages for wound healing applications. Considering this in mind, we have a big challenge for wound repair by immunoregulation. What is the big challenge? Here. In one hand, immune cells are very important to avoid infection. We need them in the wound tissue. But on the other hand, to get accelerated wound repair, like instead of 14 days to repair the wound in 7 days, we need to suppress inflammation. Yes? So there are two sides there. What should we do? What is the proper design of a hydrogel that can do immunoregulation, but at the same time can improve the regeneration? Because theoretically, they are against each other. Yeah? Any suggestion? Let's go to the next slide. This is a bit complicated. If we want to design such systems, we need to have really perfectly designed and taught system to get regeneration faster. How? The first thing to do, of course, it's obvious. We need to promote anti-inflammatory immune cells. Yeah, M2 macrophages that I showed you here. Yeah, we need to improve the expression of markers of M2 macrophage. But if we do this and we suppress neutrophils, we have a high risk of infection. We need to do some alternatives. What are the alternatives? Alternatives are to have something inside the hydrogel that can do the job of both M1 macrophages and neutrophils. Very easy. No? It became easy. We need antibacterial agent there to have the function of neutrophils. And the next thing is the drainage of exudates and phagocytosis. No? So this one also can be done. This is one of the jobs for M1 macrophages, but if we use proper hydrogels that has very good swelling properties, for example, then we can have a very nice system that can suppress inflammation and at the same time improve the proliferation. Even one step further, we can also incorporate some biomolecules inside that can induce M2 macrophage expression. And this is one example. So, mesenchymal stem cells. Do you know what is exosome? Exosomes are very, very small nanoparticles that are secreted from different types of the cells. Yeah? It can be cancer cells, immune cells, stem cells. They secrete, if you culture them in a medium, they secrete a lot of exosomes and extracellular vesicles, but they are very small. You would not be able to see it with naked eyes, of course, even not with the normal microscopes. So this exosome can be used for macrophage polarization, M2 macrophage polarization. They can be extracted from mesenchymal stem cells. This carbon peroxide can be fabricated, and plus catalase, they can be loaded inside the hydrogels for wound healing application. So the hydrogel can do this drainage of exudates. The exosome can do M2 polarization. This hydrogel, because of the polymers inside, can have antibacterial properties. And of course, this anti-inflammation comes from exosomes, no? So, the same as the previous study that I showed you about calcium peroxide, this calcium peroxide in the presence of the hydrogen, water molecules, sorry, can produce H2O2, hydrogen peroxides. And then this hydrogen peroxides, by using this catalase and catalytic activity, can convert to oxygen. So this is the job of the catalase. It can convert H2O2 to water. That's why it's included in this system. And if you see these results here, this is the very small and tiny exosome that I talked about, with the size of almost 100 nanometers. And the beauty of these exosomes are, in addition to miRNA and peptides that they have inside, for tissue regeneration, we can also load different drug molecules inside these exosomes. And if you see here the expression of different anti-inflammatory markers that were treated with these exosomes, IL-10, interleukin-10, and TGF-beta, tumor growth factor beta, that is an indication of anti-inflammatory responses. In both cases, when we have the final formulation with exosome, these anti-inflammatory responses are higher, and pro-inflammatory, which is M1 macrophage and neutrophil, is suppressed, which means a perfect environment for tissue regeneration. But these were with a lot of complex biomolecules or drugs inside. We can also think about much simpler studies or technologies. Do you remember I talked about scaling up and pharmaceutical companies that they don't consider these technologies for translation? That's why I did one very nice study. This was very, very simple studies in very low impact factor journal published, but this is one of the studies that I really like. So it's not always the matter of impact factor and the name of the journal. If you do really something that you are happy with it, then it's enough, no? So what we did was the formation of drug-free systems for immunomodulation. I was interested to know how hyaluronic acid can be used for tissue engineering. There are a lot of reports on that. And we tried to test 3 different hyaluronic acid: low molecular weight, medium molecular weight, and high molecular weight hyaluronic acid, without any drug molecules, without anything. To have the same properties, we tried to make these particles out of these molecular weights, 3 different molecular weights, with almost the same size, and almost the same zeta potential. So the charge and size would not affect so much the immunoregulatory responses that we checked in the next step. After fabricating these nanoparticles, we simply treat the macrophages with them and see what happens. And interestingly, we observed that these hyaluronic acid molecules by themselves can induce M2 macrophage polarization. What does it mean? It means that in the future, if we want to make this complex system for anti-inflammatory responses, instead of using X or Y or Z polymer, we can use high molecular weight hyaluronic acid to get this response, okay? So here you see the results. With high molecular weight hyaluronic acid, the expression and release of TNF-alpha was remarkably decreased compared to the other groups, the control group and low and medium molecular weight. We treat them with LPS and interferon gamma to induce M1 or inflammatory macrophages, and then we treat them with different samples. And you see that when we have the hyaluronic acid with this LPS and interferon gamma, the expression of TNF-alpha is decreased and interleukin-10 is increased, which means the shifting from M1 macrophage to M2 just by hyaluronic acid itself. I need to breathe a bit. Let's see what time is it. How much time I have? Around half an hour. Okay, I think it's enough to finish. Any questions so far? From audience? No? So you are not confused with different technologies and mechanisms that we can use for wound healing. Do you like it? Okay, good. So the next mechanism is angiogenesis. Now the question to you, why angiogenesis is important for wound regeneration? Why we need to make more blood vessel formation to get better wound regeneration? Yes, please. Mm-hmm. Okay. Thank you very much. Yeah. Oxygen and nutrient are the main two things that can be increased by increasing blood vessel formation. That's why we use angiogenetic molecules or some biomolecules or polymers that induce blood vessel formation in the damaged area. But there is one recently known factor as well that affects a lot. The other one is temperature. But do you know how? Any ideas? Mm-hmm. Yeah, that's true. When we have less angiogenesis, it means less blood delivery to the damaged tissue, and then the temper. Oh, sorry. Yeah. We have less transportation of blood to the damaged tissue, and the temperature will go down in the defect area. And it's known at the moment that if the temperature goes down, the proliferation of the cells will decrease. And this affects the regeneration of the wound. On the other hand, if the temperature increases to some temperatures, 2 to 3 degrees, do you remember my Monday talk that I showed you some nanomaterials that can increase the temperature of the damaged area to 40 degrees? Actually, the reason is that by increasing the temperature to 40 degrees, blood vessel formation is more, delivery of the blood is more, and then angiogenesis would be more as well, okay? And proliferation. So this is the answer. Oxygen delivery, nutrient delivery, and local skin temperature. There are several studies that I would like to show you about angiogenesis. One is about the delivery of the molecules that are very, very sensitive by using the hydrogel. In this case, this DMOG, dimethyl xylglycine, that can work very well to induce angiogenesis, but many researchers doesn't like it because they know that it will degrade very fast inside the body and it become inactivated. So using the hydrogels and nanomaterials together can solve this problem. In this case, this DMOG can be loaded inside porous nanomaterials, and then a patch can be made by polymers to implant it or place it on top of the skin, and then over time the drug molecule can be released to induce the angiogenesis. And by doing this, we can get, do you remember I showed you different trends of the release of molecules using polymers and what factors affect? This is one of the benefits. If you can sustain the drug release for many drugs that are very sensitive and become inactivated very fast, this is a huge benefit. By sustained release, we can get better therapeutic efficiency, and in this case, the expression of CD31 was meaningfully increased. The next example is the formation of the hydrogels by ion materials or ion-based cross-linking. I remember one of you on Monday asked about different cross-linking methods. I wanted to actually put inside this lecture how these physical hydrogels can be made by hydrogen cross-linking, by ion-based cross-linking, by co-surveyed technology, which means the change of the pH, but I was sure that there is not enough time, and that's why I just tried to make this one and just talk about the other one shortly as I did. So these PEG molecules with thiol groups on the surface or end of the change were mixed with Ag ions, and this Ag can make a bridge between the thiol groups to make the hydrogel. Desferrioxamine as an drug molecule that induced blood vessel formation was loaded inside, and the material was PEG. That's why it was totally safe, and as you see here, the cells can very nicely attach and grow on the hydrogel, and because of this ferrioxamine, they can induce angiogenesis. And this is one study that can be done with Huvex cells that we call it tube formation. You see these connections between the cells and making this kind of tubes from the cells, which means that these hydrogels are very good for inducing angiogenesis. When Huvex cells from the separate ones treated with the hydrogels become in tube form, it means that the expression of CD31 or VEGF is increased, and that's why these Huvex cells form these tubular structures or tube structures. More to that, the expression of CD31, as I mentioned, was checked in this study in the wound tissue, and you see that the expression is increased in the group that has hydrogel and desferrioxamine. And just to let you know, these green ones are also bacteria. So it was an infected model, and because of the Ag ions, the bacterial growth and proliferation is also less in the groups that are treated with the hydrogel. So let's go to the next one. I talked about drug-free tissue regeneration by hyaluronic acid, and this is more details to that topic that I showed you in previous slides, how drug-free angiogenesis can be done with hyaluronic acid. One possibility with hyaluronic acid was shown with my study, that it can convert M1 macrophage to M2 macrophage for tissue regeneration and suppression of inflammation. And the next benefit of this hyaluronic acid is drug-free angiogenesis. How? You know, in our body we have hyaluronidase. It's an enzyme inside our body. Everyone has hyaluronidase. By hyaluronic acid in the body can convert to smaller fragments of hyaluronic acid with lower molecular weight, and these lower molecular weight fragments can induce very, very mild inflammation or expression of inflammatory markers here, okay? And when we have mild inflammation, these two studies that of course not mine, shows that angiogenesis can be increased in mild inflammatory areas. That's why actually mild or very low amount of ROS in the damaged tissue can also be a good factor for angiogenesis and improving of the tissue regeneration, okay? So this is the main mechanism for hyaluronic acid to induce angiogenesis. And finally, the last one, very short, the last mechanism that can be considered, and I also explained in the introductory slides, is to mimic the extracellular matrix. This is an example. We know that fibronectin is one of the proteins that are in the dermis layer of the skin, and this is the real SEM image of the extracellular matrix of the healthy human. You see in lower magnification and higher magnification what is the texture and orientations of these fibronectins that I mentioned to you, that one of my colleagues is also working on the texture, surface chemistry, and geometry of the hydrogels. And this is the fibronectin scaffolds. Of course, it's very difficult to precisely and exactly mimic what we have here in the intact, healthy skin, but you see how close they are. Because the material is same, and the fabrication method was considered to be in a way that can mimic really the real structure of the healthy tissue. And this is extremely important for the tissue regeneration, no matter if it's for wound, brain, nerve. You need something that can mimic the extracellular matrix. Any questions so far? No. Okay. I need to be a bit faster, otherwise I cannot finish all the slide that I have. I showed you at least 6 different mechanisms for wound regeneration that in most of the examples that I showed you were a kind of monoregeneration, yes? Just singly was used. But imagine we develop some systems that has a combination of different mechanisms for immunoregeneration. So from now on, in different examples, I show you how we can switch from monoregeneration to multi-regeneration. This is again one of the studies that I did during my postdoc. We tried, and that was actually the basics of establishing our Capsamedix company that we used, that we develop 3D printed devices for diseases that are in the oral cavity. And the idea is to make very small devices, attach it to the tooth, and over time it can release drug molecules for oral cavity diseases. Can be inflammation, infection, whatever, okay? So what we did here in this study was to develop a very simple, straightforward hydrogel by just mixing polyvinyl alcohol by tonic acid with tonic acid. Tonic acid is again a polyphenol with a lot of OH functional groups, as you see here. This is the structure, and these red areas, parts, are hydroxyl groups. We just mix them together with PVA, and right after, within seconds, we can create a very nice hydrogels by hydrogen bonding, and we can get many different properties. The first one is anti-inflammatory responses that comes from tonic acid. Tonic acid is a really magic molecule in the nature. And again, it's in many different plants, okay? Why it's magic? Because it has anti-inflammatory properties, it has antibacterial properties, and it also has antioxidant properties. So we can get 2 different functionalities just by tonic acid that is not loaded inside the hydrogel as a drug molecule. It's part of the 3D structure of the hydrogels that we make. So we make the tonic acid with PVA, but get therapeutic response and regenerative response with what we have as a network of the hydrogel. Since we have the hydrogel with a lot of small porosities, we can load drug molecules there, and we show that it's very flexible and stretchable hydrogels for different applications. And this was the initial idea. We developed these kinds of holders for this hydrogel to place the hydrogel inside. The hydrogel was also sticky, so it can very nicely attach to the walls of the holder, and then in the next step, it can be attached to the tooth. And over time, it can release the drug molecule. What we load inside was doxepin. Why we use doxepin? Because it's a painkiller in mucositis. Do you know what is mucositis? No. I think most of you are not in the clinical side. Mucositis is the most common adverse effect of chemotherapy and radiotherapy. If you have seen patients that use chemotherapeutic agents or they receive radiotherapy, they complain a lot about pain, inflammation, and wound in the oral cavity. And there are several stages, stage 1 to 5 for mucositis, and in stage 5, that happens to many patients, they cannot even drink water because of the pain. The endothelial cells in the oral cavity is gone, and because of that we have inflammation, infection, and it's really annoying situation. Basically, on top of the side effects of chemotherapy and radiation therapy, mucositis is one of the reasons that you see that cancer patients are very, very thin because they cannot eat because of the pain that they have in the oral cavity, okay? So this was the idea. We load the doxepin, and then we did a lot of characterization to show that it's stretchable, self-healable, because we have mechanical properties and motions in the oral cavity. We should make sure that The hydrogel doesn't break, it's moldable as well, so it can get the shape of the holder that we have there, and it's also self-healable. If it breaks, it can be healed within seconds again. And then, in the next step, we studied how doxepin can be released. And interestingly, we also observed the release of tannic acid there. So tannic acid that was inside the network of the hydrogel, over time the hydrogel can be degraded and tannic acid can be released for therapeutic application. We did the study for over 7 days, and you see very sustained release of almost 4 days for tannic acid, and for doxepin was a sustained release of 7 days, and after that I don't show, but it reached also to plateau. We can get the pain killing effect from the doxepin, and also antibacterial effect that I don't show, antioxidant effect that I don't show, and anti-inflammatory results and effect that I show here with just tannic acid. Interesting, no? And we showed that the expression of TNF-alpha is nearly, like let's consider this case, 50 folds decreased compared to the control. Okay? We have 50 folds of suppression in the expression of TNF-alpha when treated with the tannic acid. So I showed you, and I, most probably you realize that I work a lot with the natural polymer and natural materials with the examples that I showed you: alginate, gelatin, tannic acid, gallic acid. Why we do this? Because the first thing is that the synthetic materials are in some cases expensive. Degradation can be an issue, side effect can be an issue, but in many cases natural polymers are very safe. Some of them we have inside the body, like gelatin in this example. So what we did here, again, we used tannic acid, but this time with gelatin. Again, we mix them and we get very nice hydrogel again by hydrogen bonding. That allantoin as a drug molecule can be inside to induce cell proliferation. And here again, we get multifunctionalities that I can skip because I explained what is the effects of tannic acid. A lot of characterizations for these multifunctional hydrogels are needed. What is the pore size? What is the porosity? And this is important. I explain you why in the first part of the talk. Viability is another issue that is very important for these hydrogels. Of course, when we put it on the wound tissue, we want to do the regeneration, not killing the cells that are already healthy there in the margins. So that's why we did the viability test. You see that this is just for the hydrogel, and this is hydrogel with the drug. And surprisingly, you see that even viability is increased over 100% when we have the allantoin with the hydrogel, which means that the proliferation of the cells has been faster in this case. That's why we get more number of the cells, and the viability value goes over 100%. We did again some hemocompatibility tests, and again, as I showed earlier in different examples, antioxidant effect and scavenging effect by using DPPH as a free radical. Let me see how much time I have. Okay, I would like to have some time also for questions. So I leave these parts because I showed in my previous examples and Monday talk as well. Let's go to some technologies that we use, and of course other researchers use to make different wound healing formulations. One of the technologies that we use in our lab is electrospinning. This is exactly the same machine that we have in our lab, recently bought, and one of my PhD students is working with the electrospinning machine that works with this mechanism that I show here. We have a pump, we have a syringe connected to a pump. Polymer solution is inside the syringe. We have electrical field, and by pushing the plunger, we can produce these nice fibers that can be collected in a flat plate or a drum that just turn. If we just collect the fibers in a plate, we get these kinds of fibers that there is no orientation. But if we collect it on a drum, since it turns, we can get really aligned fibers that, of course, we can play again with the thickness of these fibers that define the mechanical properties. What are the benefits of the electroresponse fibers? Many different benefits. We can play with the flexibility of the fibers by changing the polymers or thickness of the fiber. They can be breathable on the wound. Air can pass, bacteria cannot pass, which is very important for wound regeneration. Mechanical property is something that we can change and play with this electrospinning technology. We can prevent the penetration of the bacteria. We can load different drug biomolecules. Polymers that can be used are, of course, considered to be safe, not toxic. So these are the benefits that we can have, and simply in the polymer solution that I showed here, we can have drug molecules also there, and during the fabrication process, we can get the drug molecules inside these nice fibers. And the other importance of this technology is that due to these nano-sized fibers, the thickness is at nano range in many cases, we can have very high surface to volume ratio, which mimics the extracellular matrix that has the same property. And this high surface area is very important to have a very good cell attachment and growth. Of course, we have, with this technology, we can make different fibers that I show in these schematics. We can do blend electrospinning with single fluidics. We can do emulsion electrospinnings. We can have a mixture of different polymers, like two separate polymers in one syringe to make side-by-side electrospinning. So when they are dry, we have two different polymers in one single nanofiber that they are not mixed, or we can make core shell nanofibers, or even we can do electrospinning by 3 or more fluids or polymers, and each one can be composed of one different drug molecule. So consider having 3 different drug molecules there, one for inflammation, one for angiogenesis, the other one for antioxidant properties, okay? So these are the beauties of the electrospinning technology, that we can play with the polymer solutions and get different fibers. I showed you in the previous slide that we can have the random structure, we can have aligned structure that I skip. We can even get the porous microfibers and nanofibers with the electrospinning technology, as you see in this SEM image. How we can do that? The polymer solution that I showed you in one of the slides can be mixed with the drug molecules, of course, but also with some sacrificial agents. Okay, 5 minutes. These sacrificial agents, after electrospinning, with some solvents can be removed, and we can get these porous fibers that are used in many different technologies and fields, for water treatments, for biosensing, and for drug delivery application as well. And that's why you see a clear increasing trend in the number of publications with electrospinning technology. What we are doing in our lab with one of the PhD students is to develop electroresponse patches that can be used for oral cavity diseases and wounds in the oral cavity. I skipped that. Unfortunately, just 5 minutes, but I would like to get also, we had a lot of communication during the talk, but let's skip this. This is really pity, because this is also one of the technologies that we use. It can, these are very tiny microneedle patch. This is all of this with 100 needles. The size is almost like this size, and we have 100 very tiny microneedles on top of that at the moment is highly used for vaccination, for drug delivery applications. We can make these microneedles with different technologies. They can be dissolvable, hollow microneedles, coated microneedles with different materials. These are the benefits and the fabrication process that, if you are interested, later we can talk about that. There are also a lot of startup companies or some companies that they have already approved the formulation. In this case, this company works on these drug molecules to increase the blood concentration in the body, but for us, we are focusing on wound regeneration with these microneedles that I can talk with you later if you are interested. We can load different biomolecules, photothermal materials, oxygen-generating materials inside these microneedles, and then just attach it to the wound tissue and change the inflammatory phase and proliferation phase. So I will skip all of this. This one I would like to just show and then finish. Even we can combine microneedle technology with electrospinning technology. Also, one of my PhD students is also working on that. In the backing layer of this microneedles, these are, imagine the needles. It has a backing layer that needles are assembled on top of that. We need a backing layer, of course. This is the backing layer that, oops. This we call the backing layer, okay? And this backing layer, in this case, was made of electroresponse patch. So there are a lot of fibers there. And the benefit is that this part can mimic the extracellular matrix, high surface area, one types of drug molecules. Here, another type of drug molecule. There is no interference, and it can improve the efficiency of wound regeneration. This one, again, we can talk later. Pity, the 3D printing and the technology that can be used to make personalized medicine. But later, if you are interested, we can also talk about this amazing 3D printed technology for drug delivery applications, okay? So unfortunately, and this is one of, again, our studies recently published in Advanced Material for wound regeneration, and here we show that it's extremely important to have a communication and crosstalk between angiogenesis and neurogenesis for wound healing application. With that, I come to the end. Sorry, the last part was very fast. Now I'm open to questions. 1 minute is left. Thank you.
Thank you, Doctor. Thank you, Doctor. Now we can begin with the round of questions.
Hi, thank you so much for your presentation. It was very interesting.
Where are you? I cannot see you.
So I have two questions.
Can you raise your hand? I cannot see you. Who
Here,
are you?
I am here. Sorry, I'm in the back. Well, I have two questions.
Yeah.
The first one is about the stability of the hydrogels. I know that it depends on the material that you use to form them, but in general, how stable they are. I mean, under which condition they lose their mechanical strength and they are degraded. And the second question.
Let's answer first this
okay,
one,
yeah.
because I'm old. I might forget your question. Basically, we have a lot of optimization process in the fabrication of hydrogel. What I show you is the optimized formulation, but we play a lot with the amount of the cross-linking agent. We play with the concentration and amount of the polymers that we have there, and these are the factors that are considered for the biodegradation and degradation rate of the hydrogels. Okay. And the second question?
Okay. Yeah, the second is, well, for the antibacterial activity, is it always better if the active molecule is released, or are there examples in which the molecule is kept attached to the hydrogel?
Preferably, or let's say to have therapeutic effect and antibacterial effect, definitely we need to have the release of that antibacterial agent from the hydrogel, because there are two different phases. Hydrogel is there, bacteria is there. Okay, the hydrogel might be in contact with the bacteria, but if the drug molecule or metal ion is just inside the hydrogel or inside the patch, then it would not show antibacterial effect. Most probably the bacteria, because of the antibacterial property of the hydrogel, because these antibacterial agents are evenly distributed in the hydrogen. They cannot attach to the hydrogen, okay? But to get really good antibacterial property, we need to get the release of the antibacterial agent.
Okay.
And that's why one of the main studies in this kind of formulations is the release of the antibacterial agent. We need to get the antibacterial agent released.
Okay. Thank you.
Yes, please. Yes.
Thank you. Just do you know what is the state of the art in knee improvement? For example, if you know of certain implants in the cartilage area, so they can help elder people with this joint problems.
Unfortunately, I don't work on the cartilage and joint.
Okay.
But I see one of my colleagues, Professor Prashant, in the room. I know he's really expert in cartilage and joint. So he definitely knows what is the state of the art in cartilage repair. And I highly recommend you to discuss with Professor Prashant. Or if you have some comments, please. Yeah, of course. For you mean for schematics? Yes. Very good question. I'm very tough with the schematics and like eye-catching photos. And that's why I train my students to do that really perfectly, because one thing is to have a good lecture and presentation, but to have really the attention of audience, you need to have really nice slides. We use different things. Illustrator is one of the software that we use to make this kind of images. Yeah. You're welcome.
Hi, thank you so much for the lecture. I have a question that I don't know who asked it in the beginning at the beginning of the lecture, but for instance, you mentioned about the trachea or the 3D printed trachea for the child.
Yes.
But what happens if the child, it will grow, the child will grow up?
Yes.
So the trachea will also evolve through these kind of processes or evolution, or you need another implant for these kind of grafts?
Very, very good question. Thank you very much for that. Really, really good question. Here, one thing that I mentioned several times during my talk was the importance of biodegradation of the material. And I mentioned why we use the natural polymers, because most of them are biodegradable, okay? What you see as the implant, windpipe implant that I showed you, that was the initial structure made of a polymer, okay? But it doesn't mean that it stayed there. Actually, one thing that I forgot to mention during that example, or maybe it was intentionally because it was just in the introduction part and you were not in the field, was about the degradation. This polymer made windpipe that they produce is biodegradable. So the cells are there, but when they implant it, we have many different enzymes there. At the beginning, it's the ECM, extracellular matrix for the cell attachment and growth. But when the cells are attached, by growing, they also release and produce the real extracellular matrix and collagen there. So the collagen is produced there, the polymer degrades over time, and then the problem is solved. When the child gets bigger and bigger, like other parts of the body, wind pump grows as well.
So, okay.
At least so far, because the kid is still alive.
All right, okay, thank you.
Yes, please.
Hi, thank you for your lecture. About the holders that you use for the mucosity, it was?
For mucositis.
For mucositis. About those, how do you hold them then? Like some kind of braces that you put some resin and they use light or something like that? Because in the needles, well, the needles are the ones that hold themselves there, or you can use some kind of dressing in the other ones. But in those cases, in the mouth, there is a place that has really high humidity and things like that. How do you?
You mean how we stick it to the tooth? Yes.
How do you assure that they stick in them, that they don't fall, or someone eat them?
Yeah. Thanks again for paying attention to details. This is very important. I didn't show all the results, but the concept that we used to make was making a clipping technology. Okay, the holder is not directly attached to the tooth. First, we have a clipping agent, like a layer with a clipping that sticks with some highly adhesive glues to the tooth that are resistant to the water and moisture of the oral cavity. And then this holder clip to that to the other part. Basically, there is a connection between the holder and tooth. Okay? Yeah. Because anyway, we are expecting that the patients need to, after 7 days, because we have the release rate of 7 days, we expect that after 7 days they easily unclip and then clip a new one. Yeah?
Well, is that everything?
Yeah.
Well, thank you
If there
for
is no
Dr.
question.
Sally exposition. Really
Thank
nice.
you. Thank you very much.
Please, welcome. Now, we'll take a break and we'll return here at 2:30. Regresamos a las 2:30. And please, can you take all your stuff? Yes, please. Yes. Thank you. positions. Now it's time for Dr. Sarthak Misra. Dr. Sarthak Misra is a full-time professor in the Department of Biomechanical Engineering at the Faculty of Engineering Technology. He is also affiliated with the Department of Biomedical Engineering at the University of Groningen at the University Medical Center Groningen, where he leads the robotic surgery laboratory. He earned his PhD degree from Johns Hopkins University, Department of Mechanical Engineering, Baltimore, USA. He worked for 3 years as dynamic and control analyst at McDonald, Detwiller and Associates within the International Space Station program. He is a beneficiary of a European Research Council grant and has been honored with the Veni and Vidi Awards from the Netherlands Organization for Scientific Research. He is also a Link Foundation Fellow and a recipient of the Awareness of Spaceflight Award from NASA. Currently, he serves as the co-chair of the Surgical Robotics Technical Committee of the IEEE Robotics and Automation Society and the co-chair of the Biological and Medical Systems area of the IFAC Technical Committee. Dr. Sarthak's research interests primarily lie in the field of applied mechanics at both the macro and micro scales. He is interested in modeling and controlling electromechanical systems with application in mechanical robotics. Please welcome Dr. Sarthak. Would you like a microphone? Okay.
Well, good afternoon, everybody, and thank you for sticking it out. So thanks for the kind introduction. Thank you to the hosts for inviting me. So as I was introduced, my name is Sarthak Misra. I'm a professor at the University of Twente and also at the University Medical Center Groningen. As was introduced, I'm an engineer by training, so this talk is going to be very different from the talks that you've heard earlier this morning. And if you attended my lecture yesterday, you know that it was also very different than what you would normally heard. So yesterday I was talking about small miniaturized robots, so micro robots, milliscale robots. But today I'll be talking about larger scale robots. And these robots are medical robots that could potentially deliver the micro robots or nano robots that I spoke about yesterday. So the way I've structured this presentation or lecture is there'll be 2 parts. Thanks, that's great. There'll be 2 parts. So the first part is about needle steering, and then second part is about continuum robots. So after the first part, I'll take a short break, but also you can ask me questions, and then I'll continue with the second half. All right, so this is about steering flexible devices, and I mentioned these devices could be used to deliver the micro or nanoscale robots. So as that was introduced, I lead the surgical robotics laboratory, and the lab has 2 locations, one at the University of Twente, another at the University of Groningen. We have students at both places. So some of these things were mentioned yesterday, but for the sake of those who were not there yesterday at the symposium, I'll be repeating some of things. So if I were to describe in a few words what we do in the lab, it's to basically develop new ways, novel techniques to reach challenging locations within the body. And the way I think about doing this is by first developing preoperative plans. So using various imaging modalities, ultrasound, MR, CT, use these kinds of imaging modalities to develop models. Models to simulate the interactions of surgical tools with soft tissue. And then use these plans, models, to control a variety of instruments. These could be flexible needles, endoscopes, snake-like robots, and also going down several orders of magnitude. I'm interested in trying to track, control, and steer these micro-robotic systems. So the first half of this talk is about this part, needle steering. So I guess the first question you might have in your mind is, why is this guy interested in needles so much? What's so special about needles, right? So needle insertion into soft tissue is one of the most common minimally invasive surgical procedure. So needles can be used for biopsies. So in breast biopsies, if you have severe depression, needles are also inserted into the head. So they're used for deep brain stimulation. Needles are also used for biopsies in the lung, which is extremely challenging to do if there's a need of a biopsy in the lung. They're also used in therapy. So for example, if you have cancer in the liver, you cook various regions of the liver with a radio frequency probe. They're also used for brachytherapy, where you place radioactive seeds at very specific locations within the prostate, and then you kind of basically nuke the prostate. They're also used in fetal procedures. So when the child is still in the womb, for congenital defects, you can also do certain procedures in order to clear that. So in all of these cases, you can see that the needle must reach very specific locations within the organ. And what often happens is that the needle deviates from its intended path. And these deviations are due to just organ deformation, anatomical obstructions, physiological processes, just such as respiration or fluid flow. So one of the ways to mitigate these needle targeting errors is by idea of needle steering. So generally, needles have this symmetric tip, and when you insert something symmetric into soft tissue, it does not bend. But if you introduce a small asymmetry, like a kink here, and when you insert a needle of this manner into tissue, due to the interaction forces between the needle tip and the tissue, the needle naturally starts to bend. So it pushes the needle down. And now if you were to rotate the needle, the forces would act in the opposite direction. And then the needle would move in the other way. So you could kind of insert, rotate, insert, and kind of snake your way into the body. Asking a clinician to do this is extremely challenging. So what we have done over the years is to develop robotic techniques to steer needles like this. So these are basically passive devices. So there's no motor, there's nothing attached to the needle. But you basically, just due to the geometry of this needle, you can insert it into soft tissue. So what this talk focuses on is basically, first part of it is kind of how do you develop these preoperative plans using, develop these needle deflection models, and then use these models to control a variety of instruments, specifically using images to control the needle. So in this case, you can see an ultrasound probe that is moving over a slab of tissue to insert, to steer a needle. So there are various ways to model needles. So here's some representative work. So needles can be modeled by something called a bicycle model, where you model basically as a unicycle. So there's the front of the needle tip is kind of the front wheel of a bicycle. It can be modeled by modeling these needle deflection forces using various springs. Or this is some work that I'd done a while ago where you use continuum mechanics. So earlier this morning, there was discussion about nonlinear elastic models that to simulate tissue. So you can use continuum mechanics to also model the interactions of the needle with tissue. And then how do you control the needle? So you can control the needle by attaching a motor at the tip of the needle. You can, so these are two examples of it. Or you can, or in this case, a kinematics-based model is used, and right around here, the needle will be, near the obstacle, the needle will be rotated in order to reach the target. And this is completely done in an autonomous manner, but the idea is to basically show you that this is some work that was done in order to how to steer the needle into soft tissue. So this first part kind of covers two things. So first we'll be talking about these needle deflection models, that's on the left-hand side, and then the other is the using images to steer the needle. So first I'll be talking about two kinds of models. One is a kinematics-based model. So the basic idea of a kinematics-based model is that when you insert a needle into tissue, it moves on a circle. It moves on a circular path. So what we observed, and this is a confocal microscopic image, is that when a needle is inserted into tissue, a crack kind of develops at the end, at the tip of the tissue, and this crack is given by this value beta. And this is called the crack angle. So this kind of shows the idea. So this is a needle that is inserted into tissue, and when you rotate the needle goes on another circle, and that would be given by the red line. Now, if I were to include this beta angle, the needle would not move on this red line, but on a green line. And then using kind of standard frame transformations, you can relate the tip of the needle to the base of the needle. So for those of you who are not engineers in here, the key thing to remember in all of this is, in order to do this kind of modeling, you need two parameters. One is to know what this cut angle is, and other to know what this radius of curvature is. So that means you need to know what is the type of needle that you have and what kind of tissue you're interacting with. So that's the first thing, the two things that you have to remember from this chart. While in a mechanics-based model, you need to model all the forces acting on the needle. And these forces could be friction forces, forces acting on the tip of the needle, or distribution loads. And then there's something called the system potential, meaning that you have the work done in the system and the energy in the system, and this big pie is called the system potential. And then what you do next is you model your needle and with springs at the base of the needle, and then these springs have certain energy, and then you also have a tip force acting at the needle, and all of this can be combined and used in a mechanics-based model. So for those of you who are interested in how we did this, I'd be happy to go over it in a bit detail. But again, the key thing that you should remember in all of this is that in order to do a mechanics-based model, you need two things. You need to know what is the tip force, so what is this force acting at the tip of the needle, and then you need to know what is the distributed load acting. So these are the two things that you need to know in order to do this mechanics-based model. So now that we have this kind of framework of a kinematics-based model and a mechanics-based model, the idea was to compare how good are these models, so that we can use them to actually do something useful. So in order to do this, we have a simple experimental setup. Here's our needle with this angle alpha here, some gel, a gelatin powder, basically jello with some water and silica in there, and silica is added in there too, so that the images with the ultrasound images come out better. And then you want to find out the elasticity of the tissue. So earlier this morning, I believe Prashant was trying to explain to you about shear stress and strains. So once you can estimate elasticity in a non-invasive manner by passing sound waves through it, and then using the shear wave velocity, you can find the shear modulus, and from that you can find the elastic modulus. And so then you know what the elasticity is of this tissue. So you have the elastic properties of the tissue, you know what your needle is, and then you start doing an experiment. So the experiment was that you insert your needle into tissue, and then at around 30, so where is it? So around at 30 millimeters, you rotate the needle so it would go the other way. So then first we need to populate the model, right? So the question we had posed to you was, what is for a kinematics-based model, you need to know what this cut angle is and what the radius of curvature is, right? So the way to do this is that you would fit a circle to this plot, and you could find the radius of curvature, and then you could also, from your confocal microscopy images, find what this angle, beta angle, is. And then similarly for the mechanics-based model, you can find out what by fitting the forces in here, which you can estimate from having a force sensor attached to this. You can find out what the tip force is and also what the distributed load is, and then you do a least square fitting. So obviously, if you have any questions later on, you can ask me, but basically I'm going over pretty quickly on how you do this, but the idea is basically we want to compare those two models. That's what I want to do. I have a mechanics-based model, I have a kinematics-based model. I just want to compare. How good is it? So that's what I'm trying to explain to you here. So what we see is that, so we did our experiment, we took those parameters in there, and then we do another experiment, and we say that our needle needs to go inside tissue for 110 millimeters, and we need to reach a deflection of 10 millimeters and 20 millimeters. That's what the deflection at the tip should be. And so the model predicts that you should rotate your needle at 44.2 and 64.2. And then we take those numbers and we put it in our experiment. And let's see where the needle ends up. So the needle ended up at 10.8 and 20.6 millimeters. So fairly close to 10 and 20, right? So it's fairly okay. While the mechanics-based model tells you that you should rotate at 42.8 and 62.8 millimeters. And you end up at 10.4 millimeters and 18.3 millimeters. So again, fairly good. So the advantage of the kinematics-based model is that it's more computationally efficient, meaning it runs faster as compared to a mechanics-based model. But both models show good agreement. Next, so life is not two-dimensional, right? Things happen in three-dimensional. So we want to convert our models from 2D to 3D. So the figure looks complicated, but the idea is still the same. So for the kinematics-based model, now instead of moving on circles, you start moving on spheres. But the idea is still the same. You move on here and you move here, right? But you move on different spheres. But the concept is still the same. You want to need the cut angle and you need the radius of curvature. Instead of radius of curvature of a sphere, you need radius of curvature. Instead of radius of curvature of a circle, you need radius of curvature of a sphere. But that's the basic idea. The figure just looks a bit more complicated. The math gets a little bit more complicated, but the ideas remains the same. For the mechanics-based model, it also gets fairly complicated. You need to then decompose the problem into two planes. You have springs along the XY plane and the XZ plane, but you decompose the problem, find the system potential, but the basic idea still remains the same. You need to find out what the tip force is, and you need to find out what the distributed load is. So those are the two parameters you need. Yes, ma'am. Yes, sir. Why a sphere? Because you're moving in three dimensionals. So then that's why you need to do this in spheres. Yes, thank you for the question. Okay, so you have your 2D model and your 3D model. Now you want to compare how good are these. So there's various ways to do this. You can insert this into tissue and then have cameras track how the tissue is deflecting, but your tissue then has to be transparent. So there's another way to do this, and maybe this for some of you this may be interesting, not for a needle application, but for some other applications. So there's this idea of using fiber Bragg grating sensors. So what are fiber Bragg grating sensors? They're essentially fiber optic cables. We make cuts on them, and we pass light of a certain wavelength through it, and then which is the big lambda, and certain amount of light passes through, certain amount of light gets reflected back. But when you deflect this needle, there's a change in the wavelength. This change in wavelength is called a Bragg wavelength. And this change in wavelength is related to the strain, the epsilon in there. And this, with the strain meaning how much is the deflection. And using this value, you can then find out how much is the radius of curvature. So this is our little needle here. We make 3 cuts on it. Then we put our fiber optic cable in there. We have 3 fiber optic cables, fiber A, B, C, and these FBG sensors, or these little cuts that we put on them are at very specific places. So we know where it is because we made it. And then once from these equations, what you can do is you want to find out what this radius of curvature kappa is, and you need to find out the orientation. Right. So again, the equations shouldn't worry you. It's the idea that you should take. So this is what the needle looks like. You have your curvature is kappa. You take the first integral, you find out what the slope is, and from the slope you can find the tangent. If you're interested, I'll get into the details of this, but the math is not the most important thing. It's the idea that's going to happen. And then you can integrate this to find out what the tangent vector is. So this is what it looks like then. So this is a student of mine. So that's the needle, and then the fibers are put in there. So what he's doing is that he's deflecting the needle, and in real time you can get the shape of what the needle is. So you can see the application of this. You can put these fibers on any instrument, and you stick it inside your body. You know exactly where things are. So you don't need ultrasound. You don't need MR. So this is this kind of technology is also now being used on aircraft planes. So they also put sensors on the plane so you can know exactly what the deflection of the plane wings are. But so now you can get the idea of how you can change from the strain value and get the curvature and then reconstruct this. And all of this can happen in real time. So now we have this kind of a technology, and we want to now find out how good is our mechanics and kinematics-based model and then compare it. So this is our little setup. Here's our needle with our fibers in there. And we kind of do 3 experiments. We deflect it in one plane, we deflect it in another plane, we deflect it in an out-of-plane case. We basically want to see how well it compares to our models. And we also insert this needle into gelatin, so slab of gel, and then also want to have different cases and want to compare then how well our models are. And we have 2 cameras, one on the top, one on the side. Basically, cameras are our truth model. So what do our experiments tell us? It tells us that for the experiments in free space, when you're in plane, the measurement is really good. When you're out of plane, it's also fairly good when you compare it to beam theory, meaning you can solve these equations analytically. So we have confidence in our FBG sensors. How good am I doing with time? And then we wanted to do these experiments in the gel sample and compare them to our mechanics-based model and kinematics-based model. And we see that both the kinematics and mechanics-based model do well. And but we see that when you go deeper into tissue, the kinematics-based model does not do so well, and the reason is that when I started this lecture, I said there's this cut angle happening and some beta is happening there. Well, that beta value is not constant. When you go deeper into tissue, that beta value changes, but in our models we just take one value, and that's the reason why we think that the kinematics-based model, as you progress deeper into tissue, performs worse. But anyways, both of those models are fairly nice. It's nice. Yes, ma'am.
Why will the beta angle change during the insertion of the needle?
So the idea is that when the needle goes into tissue, or even when a nail goes into the tire of your cycle or a tire or anything, a crack happens, and this crack propagates through. And this propagation changes as you move forward. That's the reason why. You're welcome. Same happens when a bullet hits you also. If a bullet were to, I hope it never happens, but if it were to hit you, then there's a crack happens in front, and then worse things happen after that. Okay, so we have our 2 models. That's great, but we want to do something useful with it, right? So what do we do next? We want to use them to control the needle. So that's when we come in here. So we have our 2 models, mechanics-based models, kinematics-based models. That's great. That's very good, you can say, but then what do you do with that, right? You want to go somewhere inside the body. So how do you use that? So we decided to use the kinematics-based model just because it works faster, so it works in real time, and we can control the needle, since our main game is to control the needle in real time. So how do we do this steering method? So this occurs we start with the 2D case. So I'll just run you through the flow chart, how this is done. So you insert your needle. You know where the tip of the needle is. Let's say you have an ultrasound probe or a camera, and you find out where the tip of the needle is, and you know where you want to go. That's where you as the user kind of prescribe it. So the first check is done. Has the needle tip reached the target? And yes, then just stop the insertion. But if it's not reached, then you check the radius of curvature of the needle. Is it greater than the length of this from the center of the curvature of the needle? So this L is, if C is the center of the curvature of the needle, and L is the radius, does that match the radius curvature? So if it does not, then you rotate the needle 180 degrees, and if no, then you keep continuing. So that's the basic idea, to do it in 2 dimension. So again, if you are not an image processing person, you can just gloss over, just stare at it. But basically, you want to find out where the needle tip is. So you use standard image processing techniques. So you filter the image, do a binary kind of thresholding, detect where the needle tip is, and then you can do use standard techniques to find out where the centroid of the needle tip is. So then you know where the needle tip is and you know where your target is. Okay, you have that information with you. Next, you want to do some experiments. So we designed 3 experiments. The first experiment was you have a target and you want to go in a straight line to reach this. And there's no anybody involved in the loop. Everything is happening autonomously. The next case is that you want to reach the needle in a curved path. And that's the case 2. And the third case was that your needle is moving, your target also starts to move, and you want to chase it down. So how do you do that? And then first you have to do these with camera images, and then next you want to also do them with ultrasound images. In first one, you want to move along a straight line, and then in the case 5, it's moving along where the target is also moving. So I'll show you now video of these cases, basically. So this is the experimental setup. We have your needle, you have your ultrasound probe, a phantom, and now now we have case 1. So here the needle is moving towards the target, and it performs 19 rotations to reach the target. So this is basically a kinematics model running in the background, and these rotations are happening based on the algorithm I said. In the next case, it takes 6 rotations to reach this, and it gets a bit more fancier after this. The target starts to move, and so then the needle has to chase it down to reach the target. And this happens in 2 rotations. It's able to figure this out. But next we do this with ultrasound as the imaging feedback, so the images are not that great then, but it's still able to reach along a straight line with 3 rotations, and then when the target starts to move, it kind of chases it down and catches onto it. So, but you get the idea. There's a model running in the background. There's images that are used to track this, either camera or ultrasound images, and we do this in 2D. And this just kind of summarizes the basic error. It just tells you that how good the needle insertion was and the number of rotations what that was done. So basically, just summarizes what was told. Next, we wanted to use not just ultrasound but also FBG sensors. So just to recap, you have your FBG sensors. You get your strain value. From the strain value, you get your curvature, kappa, and your psi. And then you interpolate it and then reconstruct this 3D shape. Then you want to do this in 3D. So 3D gets a little bit more complicated. So instead of now moving on a circle, the needle has kind of a cone in front of it, and you must want to make sure that your target always remains in that cone. And in order to do that, you find out, so this is the figure on the top kind of shows you that the red dot is the target, and the needle must always remain in that cone. And then you perform the appropriate rotations in order to make sure that the red dot remains on this circle. And as soon as the red dot gets on that circle, you rotate the needle. And how much do you rotate the needle? By the angle theta. And from standard trigonometry, you can find out what your target location is. And that's basically what's going on the side there. And now we show the experiment where we use FBG sensors to move it. So that's the needle. It has the FBG sensors on it. And then so that's where FBG sensors are. And then we kind of prescribe that. So first experiments are in soft tissue phantom, and then we want to insert the needle in very specific locations. So the insertion distance was 110 millimeters, and we wanted to reach a specific target, and we found out that the targeting accuracy was around 1.3 millimeters. So the reason it's rotating in that manner is that it's based on the position of the needle tip, based on the target. It figures out then when the rotations have to happen and by how much, and then it kind of reaches the target. It'll run through the whole process, and it will eventually reach the target. And this kind of just summarizes how long it it took, and basically tells you how many rotations. We also did experiments in biological tissue. So this is a piece of chicken breast, and then the needle is being inserted into it with the FBG sensors, and then it reaches the target, but we also do then a scan after it to show what the path was. So the basic idea is that you want to reach accurate locations in the body and then deliver a drug there. So this basically shows you how the needle moved and based on the reconstruction. Next, we wanted to also use ultrasound as the imaging modality. So how do you then reconstruct something in 3D with using 2D as your imaging as your ultrasound feedback? This is the setup that we have. We have a needle. The phantom is number 2. The ultrasound probe is number 3, and then that's the plane of the image is number 4. So this is what you get. So this is what the image looks like. So the reason I show this image is that the needle tip looks like a dot, but like a comet. It's called a comet tail artifact, and you want to kind of find out where that needle tip is. So from your images, you can find out, you can know how much the needle deviates. So that's pxy dot and pxz dot. You know what the insertion velocity is. You know what this v is. And then based on that, you can find out what the x position is. So then you have your location of your needle in 3D. So then you need to find out where your needle tip is, and then once again you do some standard image processing, apply a filter, do some thresholding, remove certain speckle within the image, and then do a transformation, and then find out what the centroid of the needle is. So that's the basic idea of how you find out the tip of the needle. Then based on the needle tip position, you can do standard frame transformations. So if you're, again, not from a robotics background, this is not relevant for you, but basically you do a frame transformation, meaning if you want to know one point in space with respect to another point, how do you do these transformations? You can find that out. And then basic trigonometry can be used to find out what the orientation of the needle tip is. So now, using 2D ultrasound images, you can find out something in 3D, and then use this to track your needle tip. And then you can do experiments in open loop, meaning that you insert the needle, but you actually don't use that information for anything else. But you can also do it in closed loop, where you use information of the needle tip to kind of feedback and steer the needle accurately. And you use basically an observer. What that means is that based on the current position of the needle and what happened in the past, you can use it to improve what's going to happen in the future. So that's what we do here. And then I'd like to share the actual experiment with you. So this is needle steering in 3 dimensions with ultrasound as imaging feedback. So first, it's the same setup, but instead of FBG sensors, now you use the ultrasound probe. So what I'm trying to show here is that you can track the needle in 2D and steer it. So this was the kind of the first video that I had when I started this presentation. So the reason why we made a transparent gel is so that you could actually see how the needle is deflecting. And in ultrasound images, you can see that the image does not look very well, but we're still able to track the needle tip as it's being inserted into tissue. And then we also did, and then once it reaches the target, we kind of do the targeting accuracy of it also. So this just kind of shows how the needle tip moved and a reconstruction in 3D. We also did experiments with biological tissue. So it depends on where you buy your biological tissue. So we bought this. We bought this from a cheap grocery store. So there's a lot of water in there, and that's not good for our experiments. But you can see when you look at a slab of chicken tissue that how it looks like. There's a lot of other junk in there, and then you want to move towards a target, and that's what we're trying to demonstrate here, is that you can accurately track a 3D point with 2D images, and that's what the idea is. And this just shows you the accuracy with which you could do it. As we were doing this, we also got contacted by Siemens. Siemens is a company that makes ultrasound equipment. And they wanted us to develop this technique into a breast scanner technique. So basically, we use the same idea of tracking things in 2D and developing models in 3D for using that in their breast scanners. And this is kind of what shows. So using the same framework that I had, we have 4 kind of videos running here. So the first one is the online path planning going on. So what is happening is that, let's say there are disturbances while the needle is being inserted. There's a million different paths being planned every second in order to reach the target. Then we also have in the video on this top right is basically there are obstacles along the way, and the needle has to kind of replan its path in order to reach the target. The human body is not flat. We have curves on our body. So how do you do needle steering when the body is curved? And that's what's shown in this video on the bottom left-hand side. And finally, on the right-hand side, the patient is moving. Things are moving when you do this. So even if there are patient moving, how do you account for these disturbances and then insert the needle into soft tissue? But the idea, the framework is still the same, but the controller has just got more and more advanced, and that's what I'm trying to demonstrate here. So this is our experiment that we did with, this is a breast scanner. So instead of a mammography, you can use ultrasound also for scanning breast tissue. But the way it's done, used to be done is that the clinician really didn't know how much to press and how to use the information to actually diagnose it. If there's a tumor, is the tumor benign or is it something cancerous? So we developed a controller for this to basically scan the breast tissue. So this kind of shows the experimental setup where we use this breast scanner that is kind of moving on this tissue sample and then using the information from the images to basically reconstruct the tumor. There's a slab of basically a grape that's inside there. We put physically a grape or an olive in there and basically to reconstruct this shape and provide that information to it. And not just one, but several of these things we put inside, obstacles, and then reconstruct the shape of the object. And then all of this is kind of done online, so the clinician can get this information instantaneously. And then kind of we provide even the shape and the dimensions of it. So basically, to conclude this first half of the lecture, basically I showed you that we had these kinematics-based models, the mechanics-based models. Both have good agreement. And then used an idea of using optical fibers as shape sensors, and then reconstructing them to use them to steer a needle. But I also showed you that you can steer a needle with camera images, with FBG sensors, but also with a 2D ultrasound probe, and then use it to guide the needle into a designated target. So what's more is going on this project? So a few other things. So all that ugly looking equipment, we've actually transformed it into a single kind of tube. So now this is actually a human cadaver. So at UMCG, in our building on the basement, we have cadavers where you can do experiments. So the idea here was that you wanted to steer needles for taking biopsies in the lung. So using a CT scanner as the imaging modality. So this was a project that we had, and the idea was to show that you can use this for taking biopsies in the lung. And the reason is that generally when biopsies have to be done in the lung, clinicians don't want to do it unless it gets very big, because it's really challenging to go into the lung. A lot of other bad things can happen also. We also developed MR-compatible robots. So once you go into an MR scanner, you cannot use standard electronics. So we had to develop an MR-compatible robot, and this is specifically for taking biopsies in the prostate. So the patient kind of puts his legs there, and then he goes into the MR scanner, and this kind of takes the biopsy. Generally, when a biopsy has to be taken, the clinician has to run into the MR scanner room, then run back outside, run inside. So now what he or she can do is basically sit outside in the control room and guide this robot either autonomously or guide it or just by pressing a button, and then steer it into the patient in order to take biopsies. And this can be done in a very accurate manner. When I started this lecture, I was talking to you about needle having a little kink or a bend. But what if you could now change the bend of this needle as you're moving inside tissue? Then you have a needle with unlimited steerability. So this has a needle tip with a tip that's kind of moving. And as the needle gets inserted, this tip moves, and you can kind of steer it into the object. And then we also inserted FBG sensors on this, and then use this to kind of guide the needle to a target. And that's what is being shown in this little video here. So the needle shape is being reconstructed by these FBG sensors, and then you use it to kind of steer the needle. So what is being shown here is that the needle is being inserted into a phantom or a gel that has two different stiffnesses. And what we're trying to show is that basically it moves with a certain curvature in the first sample, and then once it hits the other gelatin sample, it kind of goes into another gel stiffness, other gel with another stiffness, and then progresses forward. How much time do I have? 1 hour more, okay. No, it's good, it's good. So, and now what we are demonstrating is that you're using the shape of the FBG reconstruction to use to reach a target, which is the blue dot here. And basically we show that you can reach this target accurately by changing the tip of the needle. Okay. We can also demonstrate that you can do needle steering with haptic feedback. So what's happening here is that a user is kind of guiding the needle tip, and that's a haptic device, so basically a robotic device. And what's special about this experiment is that the student was in a lab in Italy, and we were doing experiments in the Netherlands. So you can do this in teleoperation also. So, and what we wanted to explain or demonstrate is that when you provide certain kinds of feedback, do you improve steering accuracy? So when you don't provide visual feedback, provide visual feedback, there is some kind of force or haptic feedback, will it improve targeting accuracy? And that was what this experiment was about. So the user is kind of rotating that knob to rotate the needle, and there's cues given to the user saying that you should rotate it now, rotate it this much, rotate it that much, and then it kind of guides the needle to the specific target point. And this was the experiment where the student was in a lab outside Netherlands, and we in Italy, and then the experiment was being done in our lab in Netherlands. But you get the basic idea, so I'll move on. So that was the end of my first half of my presentation. I'd be happy to take any questions you might have right now. I don't think I have anything more after this. I know. Yeah, sure, please.
So how do you visualize? Because you're moving between, you are not seeing what's...
At the experiment, let's say the experiment is happening there, and I'm standing here. There's an ultrasound probe. So in the image, in the video, there was an ultrasound probe kind of moving on the tip of the needle, and that information was used to kind of feed the information back. And then on the screen of the user, it kind of says rotate 25 degrees, rotate 30 degrees, and there's like a little monitor that kind of tells you how much you should move. Yeah.
Fantastic presentation. One question: how precise and exact is the movement of the placement of the tip of the needle, especially when we think of, in terms of very critical spaces within the body. Let's say, I don't know, the brain, where there's a very narrow area, maybe the needle doesn't touch the exact position depending on the exactness or
So I think I get the question.
Yes, thank you.
You ask me how precise is this rotation. So there are two kind of key things, right? The rotation or the deflection of the needle depends on the material properties of the needle or whatever coating you've put on it, and the stiffness of the tissue, right? So if I were to take the needle and just now move it on air, it's not going to deflect because it's not a stiff medium. But if I now have a slab of gel and I put it into tissue, it would deflect. But if I have a stone in front of me, it can't even go through it. So basically, so it depends on the kind of the ratio of the stiffness, the flexural rigidity or the stiffness of the needle with the stiffness of the tissue. Depends on the diameter of the needle. So if you want, if you want to do an intervention in the brain, first you have to have a very thin needle and a needle that can also have a large curvature. So because the brain tissue is very soft, so you need a very stiff needle. So for the brain application, for example, you don't need to have too much curvature. But let's say you want to go somewhere in the liver, then the amount of bending that you need to do is quite dramatic. So it really depends on the application. So that's why also for different kind of application areas, clinicians use different thickness needles. So for moving into the lung, for example, currently needles, the clinician uses needles that look like screwdrivers. While for example, if you were to take a biopsy in the thyroid, they're extremely thin needles. So it just so the question is valid, and so depending on the stiffness of the needle versus stiffness of tissue, you can adjust the radius of curvature and also change the bevel. You can have that also. If there's more bevel angle, it deflects more.
Thank you, doctor. My question is there any way to know in real time the stresses in different regions of the needle to prevent breakage, or there's a limit in the curvature of the needle?
So that's a nice question. So the next half of my talk, I will show you how those FBG sensors can also be used as sensors to get force information. So from force information, you can then kind of go back and then calculate how much is the stress. The other thing you can do is attach force sensors at the end of your needle and use that information to calculate what the stresses are. And all of that can be done in real time. But those are the only ways in order to, I guess, find out. And you can use maybe with ultrasound images, you can also use that to backtrack and calculate what the stresses are in your system as you move forward. Yeah.
Okay, thank you. I have another question. Are you considering that the index refraction at the interior of the needle is the same as the value known for air? I'm questioning this because you said that you use the fiber optics for total internal reflection.
Yeah, so that's a great question. So the assumption we make is that the refractive index is the same in air, and sure that causes some errors, but then again with the closed loop in the system, we use that to kind of correct our errors.
Okay, thank you.
My question is, how do you actually in vivo designate the target?
I'm sorry?
How do you designate, pinpoint the target?
How do you designate?
Yeah, because in vivo, the target can be moving. You cannot just target the target. So how do you manage to reach it?
How do you manage to reach the target?
In a vivo model, a target that is moving in a vivo model.
So in one of the slides, I had showed you that we had work with Siemens as developed this breast scanner thing to move, right? So that you do that preoperative scan, and then from that scan, so this is before you do the operation, before you do that scan, you kind of find out that these are the targets I want to go to and investigate. And once you know where they are, you feed that into your experiment, and then that tells the needle that that you need to go here. And then when you are, let's say, doing your experiment, and let's say your target is also moving, then you have your ultrasound. You need to have your ultrasound probe kind of scanning the whole environment. So that's the only way you can find out where your target is and where your needle is in real time. That's the only way. Or if you use 2 ultrasound probes, but that's rarely used.
All right, and there's a way that you could just GPS it, like a chip, a microchip, nanochip?
You could do that on the needle tip. You could do that. You could put something called a Hall effect sensor that sends out magnetic fields that could be used to track the tip. But those are kind of big, so when you want to limit how much, you have size limitations.
Right, thank you.
Welcome.
Thank you, Dr. Mishra, for your great presentation. My question is related with the temporal resolution with the target. I mean, for example, if you want to make a biopsy of the heart, maybe in auricles or verticals, what about the temporal resolution of this system to track or to extract the tissue in this area?
Okay, so if you use ultrasound as your imaging feedback, you're limited by the spatial resolution. So you can't go too deep, so about 4 to 5 centimeters deep. And we have demonstrated that with ultrasound as your imaging feedback, you can get up to 2 millimeters accuracy. And if you use FBG sensors, you can get sub-millimeter accuracy. So that's those are the numbers we have. So you can maybe go, if you use MR images, maybe you can get better. But that's kind of, ultrasound is the most common one used.
Hi, doctor. Thank you for your presentation. My question is this. Do you have to calibrate your needle each time it rotates? Well, I mean, I know not every time it rotates, but how long does it take till you calibrate it again? Because you mentioned that you can perform surgeries or perform whatever the procedure is, like from long distance.
Right.
So how often do you calibrate your needle?
So the calibration is done, you do it for a specific needle and a generic case for a tissue, because tissue changes as you go inside the body. But we account for those inaccuracies by using our FBG sensors or our image feedback. So the answer to your question is that you calibrate it the only time you change your needle. So once you change a needle, you have to redo the thing again. Yeah.
Thank you.
You're welcome.
So if there is no other question.
I saw a hand there in the back. Is there? No? Okay. If there's no other questions, I'll just take a 5-minute pause, a 5 to 7-minute pause, and then we come back in. I'll just restart my computer, and we come back in, what time is it now? About 3:35-ish? Yeah, 3:30. Yeah, okay. Thank you very much. I'll be waiting. there are several clinicians doing this. So one to kind of grab onto tissue, but there's several people doing this kind of task. So these are the kind of application areas that we're interested in. So in the area of tendon-driven robotics or snake-like robots, these are some examples of literature out there. So the examples on the top are on the top left-hand side is with basically cables running through it. The example on the middle is basically like old when you have old cars antennas that can go out like a telescope. So they are basically systems that are with concentric tubes that are running through them. Or you can have basically discs with cables on them. You pull these cables and then you can deflect your needle. So one of the challenges in doing this kind of stuff is you need to know where your device is. There's various ways to localize or track your device. You can use the one on the top is magnetic resonance images, so MR images. You can use CT images, that's computed tomography. The advantage with MR images is that you have very high resolution, but then you need MR compatible devices, right? So that makes things challenging. You can use CT images, but then you get radiation or radiation dosage to the patient. So that's not something good. You can use ultrasound images, but then the quality of images is really poor. So the spatial resolution of the images is poor. Or you can use something like electromagnetic sensors. So attach magnetic sensors to the tip of your needle, and these can be used for also tracking. The disadvantage with electromagnetic sensors is that they get affected by the metal in the environment, and in an operating room, there's a lot of metal in there. So then we come back to the FPG sensors that I introduced. They are one way of having localizing instruments. So FPG sensors, as I said, basically fiber optic cables with little cuts on them, as shown in this inset, and then you have an interrogator that kind of fires the light signal through it and then uses it to give you the strain information. The reason for using FPG sensors is that they're very fast. You get at least at 20 kilohertz. That's really fast for doing processing. They are electromagnetically inert, meaning that they don't get affected by the metal in the environment, and they're also biocompatible, and they can also be sterilized. And also they're very small. They're up from can be 80 to 250 microns in diameter. So they're really advantages. So FPG sensors are an alternative to other conventional localization methods. So this is an example of one of the earlier continuum or snake-like manipulator that we developed. So basically it has these discs, and then it has cables running through them, and it has a center disc, and on the center disc is basically you have your FPG sensors. And the way it works is that when you pull your cables in a certain way, you can move it left, right, upwards, downwards. That's what a continuum manipulator is. Again, the basic idea how to model these things is basically you discretize your shape of the continuum manipulator into various rigid segments, and then these rigid segments are considered like rigid links, and then you do standard robotics calculations to find out what the deflection of each of these links is, and use that to basically evaluate what the shape of this continuum manipulator is going to be. And this basically shows you what it looks like. So the image on the right-hand side, on the left-hand side is basically your continuum manipulator being you're pulling your cables. There's a motor attached to the back of it, and then you pull your cables in a specific way, and then it moves up and down. What you see on the right-hand side, the green one is the model using the rigid lig model, and the left-hand thing is the reconstruction using FPG sensors. So what I want to show you is basically that the FPG sensors can reconstruct the shape and track this continuum manipulator in real time. So you can do it for the single bend case, so move one way, or move it for a double bend case. But the idea is that these are the kind of errors you get for in the reconstruction, about 2.5 millimeters or 2.15 millimeters. So then you want to use this information to actually control the needle. So how do you do that? You need to use the model to reconstruct the, to use it to find out what the curvature is, then use the information from the FBG sensors as the real-time information of the tip of the needle, and then use this to kind of feedback into the system in order to kind of, let's say, avoid an obstacle, which is this red blob. So the challenges in all of this are that you need to solve an inverse kinematics problem. What that means is that you know where the tip is, but using that tip information, you have to go back and calculate what the shape information is. So that's not an easy task to do in most cases. So that's challenging. Next is that there might be various loads acting on the needle, means there might be some forces acting on the needle, so you might have to compensate for that. And you have to estimate the shape of the manipulator in real time in order to do feedback control. So what I'm showing you here in this video is basically that the idea is that the task given to this continuum manipulator is that go around this red blob by avoiding it and then reach a target point. And that's what we're showing here in the video on the bottom. There are two sets of cables attached to the center disk, and then the rest of our two cables attached to the top disk. In this example here, there's a dynamic obstacle, meaning that that red blob is going to be moving in some random manner, and the continuum manipulator has to kind of avoid its shape. So has to avoid this and reshape itself. And the way this is done is that you have your model, you have your FBG sensors, and the controller, and this information is then used to kind of reshape it. And in the end, it has to reach that target. And I've also mentioned to you that there was a question from the audience how FBG sensors can be used to measure force. So based on the strain information that you get from the FBG sensors, you can use this to estimate also the force. And what we are demonstrating here is that the continuum manipulator is pushing onto a spring, and attached to it is a force sensor, and basically shows that this tip, the force actually is changing, and that is you can estimate force value. So in this case, the task given is move along this reference trajectory and then hit that spring. And what we wanted to see is that so the red line kind of shows the data from the force sensor, while the blue line is the data from the FBG sensors. And basically it shows that there's a change in the reading, and that tells you that how much force is being applied. So basically the FBG sensors can also be used as a force sensor in addition to use as a shape sensor. So as I said, so these FBG sensors can be used as force sensors, and the applications that we had in mind were for neurosurgery in the brain, but also in colonoscopy procedures. So in this specific case, we had a similar idea, same FBG sensor, but in this case there were 24 FBG sensors, so 8 sensors on each fiber, and then 3 different fibers. And then this was our experimental setup. And what we are seeing here is that you have your continuum manipulator moving in a specific path, and there's a string attached to it, and the string is kind of pulling it, and you're recording certain force values, and we compare them, and we compare this value using something called a Kosarat rod model and the rigid link model. The Kosarat rod model is an exact model for developing such kind of continuum manipulators. Rigid link model is something that which I discussed or mentioned in passing in the last couple of slides. But the advantage is that the error value is also less, and it's also computationally economical, meaning that the calculations happen in real time. One application that I'd like to discuss with you using this idea of continuum manipulators is using this robotically actuated delivery sheath. So the idea here is to use, we had a project here to repair mitral valves. These are generally quite challenging to do, where you need to open up the heart to do this. And most of the people who get this kind of procedure are older people, so they can't have this surgery done. But you can do this surgery in a minimally invasive manner, going through the jugular vein here, or going through kind of couple of incisions in the rib cage area, and do this valve repair. The project that we had was to develop this kind of robotically actuated delivery sheet. So it has basically 72 cables running inside this little continuum manipulator. You move these cables in a very specific manner and get the deflection. So I'll quickly run through how we do it. So we have a model of this manipulator, and then we you calculate its inverse based on the kinematics. So based on the tip of the position of this instrument, we calculate its shape. Based on the shape, we calculate its tip. So that's the forward part is, you know the shape, whereas the tip is, that's the forward kinematics, and the inverse kinematics, you know the back, you know where the tip is, and you want to calculate the shape. And then you have an ultrasound image that tells you where the needle tip is, and you segment this, and this is what we want to show here. So the task given to this device is that kind of move on a circular path. So we are tracking the tip of this device, and then that's what the kind of the blue cross is. And basically we are able to, for different trajectories, move along a circle, square, kind of a figure eight, and these are the various accuracy values. Next, we want, since the application is in heart applications, and we wanted to kind of simulate this, so we have a heart simulated model. So this is a heart simulator that mimics both the complete 6 degree of freedom motion of the heart. So this is basically a bovine or a cow heart, and the idea was that, and we also pass fluid through this. And the idea was that you want to demonstrate that you can go track not just the valve, but also the tip of this device, and then as the heart is moving, you can kind of then also do the procedure. And the image here on the bottom left is basically showing that. The image on the bottom right is basically showing that in addition to using ultrasound as your imaging modality, you can also use electromagnetic sensor. So that little box here is the electromagnetic tracker, and then you have these electromagnetic sensors that are attached to the tip, and basically you're reconstructing where the tip position is. But the basic idea in all of this was to show that you can use these tendon-driven systems for interventions in heart procedures. Even when there is fluid flow, and the motion of this heart model is basically based on realistic patient data that we gathered and then programmed it into this model. And basically we were demonstrate that with the electromagnetic sensors we get about 0.5 millimeter error in tracking the tip of the needle, while for the ultrasound it's about 2 millimeters. Another example is in endoscopy. So the idea here is that you have the endoscope moving into the patient, the clinician inserts it, and also has these knobs that are outside kind of to control the tip of the endoscope. But we want to automate this process so that the clinician can actually focus on the procedure intervention, meaning doing the diagnosis, while the tip of the endoscope is kind of moving automatically and guiding and going through the gastrointestinal tract. So the way to do this is that we developed a markerless method to track the tip of the needle. So basically by putting few markers on the tip of this endoscope, we were able to track the tip of the endoscope and then use this into a controller to track the tip of the endoscope. And that's what's being shown here. So you have this endoscope in a gastroscopic model, and the tip of the endoscope is being moved externally, and we are basically overlaying it with a reconstructed image and demonstrating that you can track the tip of the endoscope in real time with 2 millimeter errors. So you can, in addition to using tendon-driven, so the big disadvantage with using tendons or cable-driven devices is that you can't miniaturize them. So, so if I were to now use a system where I have a flexible device that is steered into the system, but I use ultrasound, I use magnetic fields to kind of steer the device, so I can do this in a wireless manner. So then that would make it possible for me to miniaturize it. So the next half of this lecture is basically using magnetics to kind of steer your flexible device. So there are various ways you can do this. You can use magnets. You can put ferromagnetic beads at the tip of your device. The disadvantage with this is that it causes images, artifacts in your imaging system. You can use permanent magnets at the tip of your instrument. The disadvantage with this is that you're limited by the size of your flexible device. Or you can use electromagnetic coils, so miniaturized coils that you can attach to the tip of your endoscope, to the tip of your flexible device. The disadvantage with this is that one, there could be heat issues, meaning that the temperature in the coil would change. Would change, but also very few people know how to make miniaturized coils that are like as thin as paper, and that you can put around your catheter body. So this is an example of where we use basically a catheter, a simple catheter. We attach a permanent magnet at the tip of it, and then put it into this coil setup. This is a simple Helmholtz coil setup. And then the task given here is that kind of track the tip of this device as it is moving in a figure 8. So this is what it looks like. So here's your the image on the right-hand side is showing a catheter with these permanent magnets attached to it, and this is what the ultrasound image looks like. That's the center image. So we need to do some modeling for our controller. Standard modeling that you might look in an undergraduate textbook in mechanics. So basically there's an input torque that is generated from the dipole moment of your permanent magnet, and that dipole moment matches the moment that you get from your external coils. And due to this interaction between the external coils and the magnet at the tip of it, the catheter bends, and the amount of deflection is depends on the flexural rigidity, depends on EI. So the Young's modulus and the first moment of inertia. And so we feed this into the controller, and what you see in this image here is that an ultrasound probe is tracking your catheter tip, which has the magnets on them, and these the catheter is kind of moving in a figure 8, which is something like this. And then and basically the task given, and what you see here is that the blue circle is the target point that we are given, the reference point, while the red circle is what is being tracked from the ultrasound image. And we see that we are able to do catheter tracking in closed loop with a mean tracking error of about 0.4 mm. In addition to that, we decided how can we improve the tracking accuracy? So in addition to having ultrasound as an imaging feedback, we decided to put these FBG sensors again on them. And that's what we have here. So you have your catheter, we put your FBG sensors in there, and then also have your permanent magnets in there. We have two kinds of FBG sensors. One is FBG sensors that are kind of single fibers that are attached at the tip of the catheter. We also have a different kind of FBG sensors called multi-core FBG sensors, and what these are basically all the fibers clumped together which makes them a very thin, one single thin fiber. There are about 7 of these fibers all glued together, and then you can insert it into the lumen or insert it into your catheter. So then you get better accuracy, it's smaller dimensions, and also it's more precise than these single core fibers. And that's what this video shows here. So basically you're taking data from the FBG sensors, data from the ultrasound, fusing them together, so using both that information to track the tip of the needle. And what we see is that with this fusing of information, you can get a mean error of about 0.18 mm. So that basically the idea here was to demonstrate that fusing various sensor data improves the tracking accuracy in an intervention. So not just using FBG, not just using ultrasound, but combining both of them. So next we wanted to develop new kinds of magnetic coil systems. So this system is called Big Mag. So the student who developed it thought it looked like a Big Mac, and that's why it looks like. So it's a kind of 2 coils. The top 2 are like the bun of a hamburger. There's a workspace in between. There are 6 coils here, 3 coils on the top, 3 coils on the bottom. It generates, it's about, from the floor, it's about this high, so taller than me. It generates very high magnetic fields and field gradients. And what you see on the bottom right is basically a catheter with permanent magnets attached to it. And we wanted to demonstrate that you can do steering with this system. There are cameras, one at the top, one at the side, and what we're demonstrating here is that you can, as this catheter is moving inside using the magnetic fields, basically you're able to track this catheter tip in real time with 0.59 mm accuracy. And this basically is showing how the system moves. So there's the bottom 3 coils are kind of rotating, the top 3 coils are rotating, and the task given here is that the catheter should move kind of in a spiral manner. So the idea was to show that you can get a tracking accuracy about 0.5 mm with a speed of about 3 millimeters per second. Next, we wanted to show that you can actually use this system for something useful. In clinical procedures, you often have to grab onto tissue to take things out. We wanted to demonstrate that a system like Big Mac can be used for grabbing onto tissue like how a cowboy does to catch a cow or a bull, use it like a lasso. So basically what you have here is that you have the catheter with the permanent magnet attached to it, but you also have a little coil at the end of it. So what is happening here is that when you turn this coil on, it attracts the permanent magnet, and then the permanent magnet kind of closes up, and you can increase the amount of current in the coil, and it kind of increases its grip. And the idea is to remove polyps from the intestinal tract. And that's what's being shown here. So it goes into the Petri dish, you turn the coils on, it gets attracted, and then it starts to move this slab of tissue. What we also demonstrated is that the maximum grasping force is about 0.1 Newton, which is kind of sufficient to grab onto this slab of tissue. But basically to show functionalities of these magnetic systems. The next system that I like to show you, a magnetic system I like to show you, is this ARM system. It's called Advanced Robotic System for Magnetic Manipulation. It is basically a large magnetic coil attached to a robotic arm. So basically you can scan the whole body of the patient to do the intervention. So this kind of shows you how it was done. So it's the ARM system has this, as I mentioned, a robotic manipulator with a 6 degree of freedom robotic arm. So it has 6 joints that kind of move. A coil is attached to the end of it. And there's also markers to track the patient motion. It also has a catheter insertion system, meaning that you can insert a catheter into the patient, and this can be done in an autonomous manner. So that's what's being shown here. The system was essentially designed for endovascular intervention. So procedures where you enter through the groin, through around here, and have to reach this area. and there are passive markers on the body. So as the patient is moving, you can also track the motion of the patient, and then use this to change the shape of the robot arm. So it's envisioned that the experiments that we do, basically the robot arm would be kind of move across the patient while the catheter would be inserted into it. This is a slide I'd shown you earlier, I think yesterday, but the idea is the same basically. So now instead of having external magnets, why not have internal magnets? So this is a magnetic catheter system where you have coils that are kind of attached onto the catheter. There are 3 coils here. You insert them in, you turn the coils on, they open up like a flower, and then you can kind of release your drug or your robot, and then you can manipulate them, and that's what's being shown here. So you insert it into the patient, you change current in the coils open up, and then it kind of guides the micro robot, and that's what's being shown in the bottom video, where the idea was to move the micro robot in a kind of a triangular path. I think there was a question last time that, or yesterday when I was doing my presentation, was that if you inject a robot into the body, and if you say you want to get it out of the body, how do you do this? And the solution to this that we came up with is this magnetic capsule delivery system. So it basically has a permanent magnet attached to the tip of the coil, but also has this coil system, which can be used to not just fire the coil, but also use it to attract the capsule back. And that's what's being shown in this video here. So you basically have it releases this capsule, and now you turn the coils on, and that it can attract the capsule back. So this is one mechanism whereby capsules can be not just delivered, but also be retrieved. On the inset video on the top right, you can basically see that the coil is being kind of maneuvered in order to guide this capsule robot. The black thing is the little capsule that is kind of ejected, and then it kind of attracts it back. Another example of a flexible, magnetically actuated system is this. So this is a requirement that we had actually from clinicians. So when you have blockages in your cardiovascular system, they basically need to kind of use a drill to kind of clean it up. There are various ways to do this. We came up with this magnetically actuated system, which basically changes the lateral motion into rotational motion. It has a combination of both permanent magnets and also electromagnetic coils. And the next video kind of shows the idea. So it is basically has a kind of a drill attached to it. Turn the coils on, and that makes it basically like a motor. And then what we are showing here is by changing the drill bits of these various of the device, we are trying to show that you can actually clean up the occlusion, so the blockage that is there. And so this just kind of shows you the experimental before and after. So you get around 35% reduce in stenosis. So stenosis basically is the amount of blockage that you have, and this is comparable to what you would see if, or comparable or even better than what you would see in the conventional system. Some of the other things that we're doing is basically developing these kinds of flexible probes. So this is a silicon PDMS designed catheter tip. It is a soft robot, but with magnetized at the tip of it. And now we apply magnetic fields and can basically guide it through this complex vasculature. So the way this is done is that you have you make this mold of silicon and PDMS, then you magnetize it in very specific directions, and then when you apply the magnetic field, it can rotate in left, right, or in any configuration, and that makes it a soft magnetic device. Another example is on the right, is basically using shape memory polymers. So shape memory polymers are basically devices that when you increase the temperature, they change their shape, but they also remember that shape. And then when you cool them down, they can go back to their original shape. So in the example on the right, polyurethane based, that's an example of a shape memory polymer. So where basically you can the idea was to go through that really complex vasculature and then change the shape of it and then lock it in that position and then grab onto a polyps and remove it out. So basically what you're seeing there is that it's changing its shape, and then there's a laser tip attached to it basically to show that you can reach various configurations. So once you heat it up, it becomes a bit soft, then you can bend it with magnetic fields, and then once you want to lock it, you again cool it down and it stays in that shape. And now you're heating it, so that's now I'm describing the video on the right. And then you want to now retract it back. Next, you wanted to show to remove a polyps. So it basically goes into the intestine or into the stomach, and then grabs onto the polyps, and then returns back. And finally, just another example of a continuum manipulator. It's basically a manipulator with kind of flexures, so just cuts attached to it. So by just having clever designs, you can come up with these manipulators and attach magnet to it. And then when you apply magnetic fields, you can deflect it and move it in very specific configurations. So this is, again, not with a tendon, but by just cutting flexures. So these cuts, by making cuts on them, they can deflect and bend in very specific ways. And then you have a camera system to kind of track onto it. So I think this is my last slide. If there are any questions on any of the topics, either the first half or the second half, I'd be happy to answer them, or even about what I discussed yesterday if you were there and something was unclear. But thank you again for inviting me, and thank you for sticking around. Questions, if any? There's the woman there.
Hi, thank you for the talk, for the lecture. Thank you. So you mainly work from the neck below, right?
For these, so yeah, there was one application or one project that we had where we had for neurosurgery, where basically you had to go drill a hole and then use FBG sensors to track it. I didn't have time to discuss that, but yes, that was one application. But most of our cases are from neck below.
All right. Okay.
So
it's only one project for the brain.
Except one for the brain. That's where we had this procedure where developed this special needle, attach FBG sensors to it, because it's just very difficult to track inside the brain, and that was one example that we had.
Yep. Thank you.
Welcome.
Hi, thanks. Hi. One question. Your magnetic catheter, it just have like 3 degrees of freedom, right? And
sorry, what? I have
the magnetic catheter.
Yeah.
It has like 3 degrees of freedom, right?
Correct.
And the other one have more, right? Because you can have encounter curves like an S, and this one is just 1.
So with a catheter, with a magnetic catheter, let's say I put, I don't know, permanent magnets on it, you're right. So I can move left, right, up, down, right? So that's kind of, and I can rotate, but yeah, that's fine. So, but while with the flexible needle, you're saying that you can do, you can make curves. So with the example that I had here, for example, here by introducing by using a soft permanent magnet that is attached to the tip of this catheter, I can do much more complex shapes. So with permanent magnets, you're right. I cannot do complex S shapes, or I have to have permanent magnet, catheter, permanent magnet, catheter, and then I can do complex shapes. While with having a soft magnet where is it? By having a segment which is basically like a spring, but very soft, I can get a complex shape like this. Or the image on the video on the right-hand side, I have a shape memory polymer 1, and then shape memory polymer 2, and they're trained at different temperatures. So one is trained at, let's say, 35 degrees, other is trained at 42 degrees. So when you make it 35, the first one can kind of move left, right. And when you make the 42 degrees, the other one can move. So that way you can make complex shapes.
Okay, so it can be like an coil array, right?
Yeah, it can make it like
so you can control it one by one?
You can control it step by step. So but you rightly said, just using permanent magnets, at one point you will not be able to. So
you cannot avoid, for example, some obstacle.
Yeah, then if you want to make complex shapes, you need to do something like this.
Okay.
One way to do this, yeah.
Thank you very much.
Hi again.
Hi.
Yeah, my question is more like, I think you mentioned it yesterday, but I'm curious about what makes you switch from aerospace research to bioengineering.
Ah, okay, nice question. You're right. The short answer is that the short answer is that the underlying theory between space robotics and medical robotics is the same. So they basically are both working extremely challenging environments and where you cannot make a mistake. Right, in both. So that is the and the basic fundamental robotics, mechanics, dynamics is almost the same. So that's how I had the transition. The second, I think, is a more personal reason is that I finished my master's and I thought I wanted to study more, I wanted to do more things, learn more things, and that's how I got in. I wanted to do a doctoral degree. And the third reason is that when I was working in that company, so I worked in it's a big robotics company, and they have a space robotics division, which is very big, but they also had a medical robotics division. And in this medical robotics division, they were taking the technology that they developed for space robots and moving into using the same ideas to develop robots for neurosurgery. And that's when I kind of got the idea, let me try something else out. Okay. That's why I did. So and if you want to know more, I basically, after I did my master's, I wasn't sure if I wanted to do a PhD or not. So I wanted to experience working also. So that's why I took that career path also.
Okay. All right. Thank you.
You're welcome.
Hi again. I'm sorry, I do not know if you mentioned it before, but this has been nagging me. Especially in the injection tube, it has a hollow center. So how do you manage it doesn't get clogged with tissue, especially soft tissue when it travels? Does it have a cap or something to prevent it?
Yeah. So I did not show this but in some of our recent work, when we do these experiments, or actually most instruments, they have one channel for light, for example. If you take a medical instrument or endoscope, it has one channel for light, and in the other channel it has a water channel where you pass water or fluid to kind of clean things around. So so we do have a system. When we try to take this one step further and do it for actual preclinical trials, then we have a water channel for also cleaning. And in our applications, most of the areas are kind of open spaces, either in the stomach or gastrointestinal tract, where you don't actually kind of puncture into tissue and it gets clogged. But we do have a water channel and a camera channel and another is a delivery or retrieval channel.
Right. Thank you.
You're welcome. No more? Okay.
Hello, Professor. In the first part of the lecture, you talked to us about the mechanical model for the deflection of the needle. And I saw a figure that looks like a spring. I was wondering if you modeled that using a harmonic oscillator approximation. And if you did that, why it is valid to use that approximation in the model?
So it's not exactly a harmonic oscillator. It's just a spring in there. So there's no damper in there, right? And so the spring, the key is that is valid. Well, it's an approximation, right? So the tissue does not behave like a spring, or because it's linear elastic. It's a linear elastic spring. So, but it's an approximation in there, and the key is that you have at least a first approximation, and then you have better control. In the control phase, you kind of adapt for these, for the errors that you have done. But the key is here is that you need to somehow estimate the stiffness of these springs, and that is done in your calibration phase. So that makes it an additional work that you have to do in these mechanics-based models. But it's no damper in there. There's only a spring.
Okay, thank you.
Any more questions?
Okay, then thank you very much.
Thank you, Dr. Mishra, for your conference and for your lesson.
Sorry? Okay, sorry. Thank you very much. Thank you very much.
Thank you.
Announcement for you? Is there an announcement?
Yes.
Okay.
The coffee break.
Ah,
If you
okay.
want some coffee. Well, next we will have the coffee break. It's only 15 minutes of coffee break, and then we will return for the last lesson of the day. That is in charge of Dr. Patrick. So enjoy your coffee, and we'll see you in 15 minutes. lesson in charge of Dr. Patrick Van Richt. So I will read a little bit about him. Dr. Patrick Van Richt is a researcher at the University of Groningen, whose research projects focus on the development of new dynamic biointerfaces to understand the relationship between material properties and cell behavior. Therefore, Dr. Van Richt and his research group developed high-throughput parameters and technologies to investigate biological phenomena, depend on surface parameters, but also created a new coding approaches and delivery strategies using nanohydrogels as vehicles. The projects that are carried out within the research group are both fundamental and applied and have a strong relationship with the clinical and industry. Likewise, Dr. Patrick has been president of the Dutch Society for Biomaterials and Tissue Engineering, leader of the research program Nanobiotechnology and Advanced Therapeutic Materials, editor of Biomedical Polymers of the Journal of Polymers Open Access, and guest editor of Polymers, Bioactive Materials, and Tissue Engineering A. That's amazing. So please give a huge welcome to Dr. Patrick.
Yeah. So thank you for this kind introduction. So it's nice that you're still here. I think I'm tall enough to do it without. Sorry. I'm very happy that I'm the last speaker. I find you all very, very brave that you remain perseverant and join. So yesterday I told a little bit about nanogels. And today I would like to cover a little bit a similar topic, of course, but I would like to expand with a little bit more examples, but also a little bit more explanations. And what I hope that at the end, through the research that we do, that you also get a little bit the philosophy and the translations that we did along the way. All right, so can I just ask who was here for my talk yesterday? All right, so then I think that I can skip a few slides and I have to make sure that I don't make the same jokes. All right, so if we're talking about nanomedicine, I'm not going to dive directly into nanogels, but first you need to appreciate a little bit the system that you're working with. So basically my group is divided in two. So we're looking at nanobiomaterials, namely nanogels, but we're also looking at macroscopic interfaces, macroscopic materials. And the underlying phenomenon that we are interested in is how do cells deal with materials. And that is not only for implants, but also if you want to deliver drugs, how does a nanoparticle, how does a microparticle interact with a biological system? So what we learn from a macroscopic point of view, we can also apply it to a nanoscopic point of view, and vice versa. So if we're looking at the human body in all its complexity, then what we of course immediately see is that we as a human being are smack in the middle. But we are quite complex because we are built out of several tissues. So here you see the lungs, you see the brain, the heart, but that tissue again is of course made out of different cell types, extracellular matrix, and if you look even further down, a cell is again composed out of different organelles. It has the nucleus, it has our genetic code stored, it continuously produces proteins, it divides, it behaves in a certain way. So if we are looking then at the overall system, it ranges from macro to micro to nano. Meaning, okay, if we want to intervene, if we want to treat a disease, if we want to treat an illness, we can do that on the macro scale. And here you can see a nice image of the whole body. You can here see an intraocular lens, what Theo was also talking about this morning. So these are generally macroscopic items, right? If you then go down, then in the micrometer range, then you're dealing with cells. If you go in the lower micrometer range, you're dealing with bacteria. You can even go lower, and then you're talking about
intracellular aspects like ribosomes, endosomes, but if you even go smaller, you're talking about proteins and even single molecules. Our body spans from sub-nanometer all the way to meters. So you need to appreciate that you need to intervene at the right level. All right, so how do we intervene at different levels? So how would we, I hope everybody can still hear me, I still have quite a loud voice and I'd like to move around a little bit. So how would we intervene with a disease on a sub-nano level? Or on a nano level? Molecules, yeah, what kind of molecules? Yeah. So how would we intervene for a disease? Medicine, right? This is paracetamol. We all take it frequently. So if we intervene on a subcellular level, even on a nano level, we apply drugs. Right? We use drugs. So what would we use to intervene on a microscopic level? Surgery, and what do we do with surgery? Yeah? Cut and slice, but do we also need something maybe? Prosthesis, implants. Yeah? So if you're talking about a microscopic way, you're talking about implants. The intraocular lens, the hip implant. Oh, okay, sorry. I did not realize that they were also online. So then we do it with a microphone, not a problem. Yeah. So we have the nanoscale, the sub-nanoscale, we have the macroscale, but what about the microscale? How do we do this? How do we intervene? Nanoparticles, yeah. And of course, Helder already had, and also Ali, already discussed quite a lot on nanoparticles, microparticles. How do we apply them? And we always come up with this kind of overview, right? So we have here a whole bunch of different nanoparticles. These nanoparticles we can control in terms of composition. We can control their shape. We can apply some chemistry to make them targeting. But they, of course, also have already surface properties and chemical properties on their own. And we can tune this. All right, very nice. I always like these overview images because they immediately give you exactly what you want to know. But if I'm looking at this picture, I get a little bit frustrated. So what am I missing here? Proteins, yeah. No, think a little bit about the title of the presentation. Nanogels, yes. You know, we have here wonderful particles, and I don't debate whether they are good or bad. They are all wonderful because they all serve a purpose. But of course, we have here inorganic nanoparticles, iron oxide. We have here quantum dots, dendrimers, micelles. But we do not put nanogels in there, which is a pity because already, well, I think 15 years ago, Krzysztof Majewski, one of the pioneers and the godfathers of controlled radical polymerization, they already wrote about micro and nanogels for drug delivery applications. So already 15 years, and very more recently, basically the godfathers of microgels and nanogels, André Piech, Walter Richtering, but also Thomas Hellweg, so these are all Germans. So Germany is known for their nanogels, microgels. Yeah, that is basically the mecca. That is also where I learned it. But also Andrew Lyon from the US. So also they had a very nice review on their applications. So why don't we put nanogels in there? Well, I hope that after this summer school lecture, if you ever write a review about nanomedicine, you place nanogels in there. All right, because why? Why would we place nanogels in there? For the very simple reason that we can very nicely control the chemistry. And we can use a whole range of different monomers. We can make them in different shapes and different geometries. And we can do post modifications on there very nicely. We can add peptides, and we can add ligands. So we can do a lot with these particles. All right, but what are actually nanogels? So in order to see this, I would first like to ask, what is a gel? Three-dimensional arrangement, yeah, of? Polymer, yeah. So I hear all these buzzwords, and they are all correct, right? So the definition of a gel is basically a non-fluid colloidal network. Yeah, so basically you have a network, and a network that inhibits flow. Well, that is a little bit the gist. So how does it look like? I think everybody knows more or less in their gut how a gel feels, or sees like and feels like, you know. But what this illustrates here, so here you see a plug in the drying state, and if you make it wet, it swells up. Why? Because it has a good affinity for solvent. And the solvent that you want to use, of course, depends on the combination of solvent with monomers. Let's assume that we want to make hydrogels. So in this case, you put it in water and it really swells. Here, you can nicely see a gel, a transparent gel. We call it a gel because you can do an upside down test, it doesn't flow. And if you look at it with an electron microscope, then you can see here all these nice networks. It's very porous. And the fact that it can hold up water means that the polymers are really hydrophilic. And so if we have a gel that is for water purposes, we call it a hydrogel. All right. Oh, now I'm a little bit too quick. So basically what makes a hydrogel? Right? Because if we just take polymers that are dissolved in water, we don't automatically get a hydrogel. Right? So if we want to make a hydrogel, we need crosslinks. So the crosslinks, they are crucial for preparing a hydrogel. This can be covalent, but this can also be due to physical interaction. So an example for physical interactions is, for instance, alginate hydrogels. Yeah? You mix calcium, chloride, you mix alginate together, and they form a hydrogel because you have ionic interactions. But for the sake of argument, because in the remainder of the lecture we're dealing with covalent crosslinks, so I will focus a little bit on that. So how do we actually prepare a hydrogel like this with covalent crosslinks? Well, then I would like to show you my favorite chemical approach. And why is it my favorite chemical approach? Because it is easy. I like things to be easy. Because if it is very complicated, it is difficult for new students to master it, but it's also more difficult to translate it to the clinic. So the Summer School of Helder yesterday was all about, okay, how do we translate it? What do we need to do? If a company, if you go to a company and it has 10 steps, forget about it. So simplicity is, in my case, or in my opinion, a very beautiful thing. So if we're looking at a hydrogel, and these are then, this is a very general hydrogel, so this is BHEMA. What we see here is we have BHEMA, we have bisacrylate, and this forms a nice hydrogel if you apply free radical polymerization. So for those who don't know exactly what free radical polymerization is, so basically what you have is a initiator. So this initiator is usually a peroxide. This peroxide cleaves homolytically. So that means that this bond, which contains 2 electrons, is homolytically cleaved, creating a radical. And this radical is prone to react with double bonds. This double bond flips open, so one of the electrons pairs with the radical, and the other radical, or the other electron, stays there. So that is the initiation step, and you create a new radical that, again, can react with a double bond, which goes on and on and on. It is really robust, right? So if we use PMMA bone cement in the clinic, it is based on free radical polymerization, right? It can withstand water, it can withstand proteins, you can do it with cells present, it always runs. unless you have too much oxygen there. But in principle, it's really, really robust. And it's really diverse. So yesterday I told you, okay, there are a lot of commercially available monomers. And this is just a very, very tiny selection. So these you can all buy easily at Sigma. Here you see the PMMA. You see here, so these are more hydrophobic. You also see branched ones. These are more hydrophilic. So these are acids. These are ethylene glycol based. So this is a single unit, but you also have them longer. You have styrene, of course, but also all these other types of polymers that are acid responsive, base responsive. Of course, below here we have the thermal responsive polymers. And this is just a very, very, very tiny selection. And in addition, it is generally not extremely difficult to make a new monomer. So these reactions are generally quite easy to do for a skilled chemist. So we have an enormous library of possible functionality. And that is, of course, tremendously useful. So what are now actually nano and microgels? So for those who were here yesterday, I compared them to a sponge. Not because on a fundamental level they behave the same as sponges. Yeah? The physics behind it is different. But it is about the idea what you can do with a sponge. So the sponge can take up water, and if you apply force or another stimulus, you can squeeze out all the water that it contains. Well, this is also what I showed yesterday. So there are a lot of functional sponges. So what can you actually do with a sponge? Well, you can take up liquid, not only water, but also complex fluids. That is really crucial, of course, because if you want to take up something like medicine or like biological samples, then it is very convenient that a sponge is able to do this. You can also interact between interfaces, so the sponge can actually go to an interface. It withstands mechanical force. You can apply force for it to deliver something, and it remains stable. And that is very, very important. And of course, you can use it as a filtration device. So a sponge is a porous network. This porous network has a distribution of pores. If you have smaller pores, then the more larger pollutants will remain, and the smaller pollutants or the desired compounds that are smaller can pass. You can change this, so you can change your filtration or separation potential. All right. Here we go away from sponges. Yeah? So from now on, we go only for little sponges, mainly nanogels, but I also have a couple of examples at the end for microgels. And yesterday I showed you this, but I did not pay too much attention to it. I just told you, okay, if you have a nanogel or a microgel, it is actually a combination of 3 different factors. So it behaves like a macromolecule. It is a colloid, it's a particle, but it also behaves like a surfactant. Well, what you can see here is that it's nicely porous, but it also means that on the outside it's a little bit more fuzzy. The swelling really depends on the environment. So you can imagine that if you have a hydrophilic nanogel or microgel and you add a solvent that is less compatible, then it will not take up that much anymore. So the environment in which it resides is really indicative for how it behaves. But why do we attribute these 3 properties to a nanogel? Well, first of all, we can see it. Right? So if we, for instance, so here this is an example where we put nanogels on a surface and we look at it with AFM, you can really see that these are colloids. So nice particles, they have a defined shape, a size, we can see them, we can detect them, no problem. So that is quite easy. But why is it a macromolecule? Right? Well, in principle, it is one continuous network. They're all covalently attached. If it would be perfectly defined, we would call it a molecule. But since it's not perfectly defined, we had a distribution of molecular weights, we call it a macromolecule. But also, it behaves like a surfactant. And this is an example that we did a very long time ago. So this is an oil droplet, and This is stabilized with nanogels, and this is a hydrophobic surface that we dipped in a aqueous suspension of nanogels. And if you then look at the end, you can really see that you see this capsule, and within the shell of this capsule there are all these nanogels. Also, if you look at a planar hydrophobic surface incubated with nanogels, you really see that they stick onto the surface. They really want to go at the interface. So maybe a question, maybe an intermediate question for those who attended Theo's lecture. So which scientist was actually also looking at colloidal type of structures at interfaces in biology with biomaterials? Who remembers? So in this case, they are nanogels, but if we look at implants, then also proteins are prone to do this. Anyone remember the name? Where smaller proteins are continuously exchanged by larger proteins? Vroman, very good. So the Vroman effect. So also there you have this interface colloidal stabilization. And nanogels do this as well.
Just one question to clarify something. So, in this case, I mean, nanogels, of course, I agree that they have to be cross-linked, but covalently. But also, do you think are macromolecule or many macromolecules that have non-covalent interactions could form a real nanogel? That's the first question.
Can you make a nanogel out of non-covalent crosslinks?
Yeah,
that is
that's
basically
the question.
Yeah. Yes, the question is yes.
Okay, so the second is, I mean, when you go into the nano, smaller, sometimes you can get like quasarbates. So the question is, what is the relation of the nanogels with the quasarbates, which is basically solid liquids, kind of sponges, no? I mean, very liquid-like things.
So coacervates really depend on the environment, right? So coacervates, they generally form when you are in something dehydrating system, so high salt content, where basically then the macro are the polyions, and polyanions and polycations are basically slightly dehydrated, where they then interact with one another. So that is, well, it's not completely a gel. It's usually materials that have water pockets on the inside. It's slightly dehydrated, but not completely. I don't think on the nanogels they are coacervates. Maybe you could make nanogels out of coacervates, but that is something that I don't know. I don't know any examples.
Yeah.
But if I would make, so if I would be able to miniaturize alginate into a nanoparticle cross-linked with calcium, then that is not a coacervate. That is just a stabilization based on charge. All right. So they really have. Oh, sorry. Yeah. So they really have these three attributes, right? And that means that you can play around. And in the past, we played around a lot with emulsions, making capsules, making smart capsules and interfaces. But what is also interesting is a nanogel, and particularly the way we make them, they are not homogeneous in distribution, in density. So there is usually a slightly denser core, and it becomes a little bit more fuzzy towards the end. That also means that you have quite a nice hydration shell. And so it's almost like you have individual polymer strands coming out of it, which are still part of the network. And this is, of course, very convenient because that is really hydrated. And if we are talking about really hydrated systems, they are prone to not foul. So they can withstand, to some degree, protein fouling, so this corona effect. But also, if we choose the system correctly, we can make them stimuli responsive, so switching usually means switching between size. This can be temperature, this can be pH. Currently we are also doing light. But this is absolutely fantastic because we can use those kinds of attributes. So there's a whole range of things that you can do to synthesize them. We prefer the precipitation polymerization because precipitation polymerization is really easy. So basically you are at the interface between solubility. While the monomer is soluble, the polymer is not. So if you initiate the polymerization, which is a free radical polymerization, it starts to polymerize and it starts to collapse on itself, forming a colloid and then growing. It's really easy. You add a stabilizer and in the end you stop the reaction and you purify it. So what do we generally use? So NIPAM and NIPMAM are really nice monomers for nanogel preparation. Why? Because you can just increase the temperature. So if we do it at 70 degrees C, that is way above the volume phase transition temperature. So the precipitation or the temperature where NIPAM is precipitating. We add our cross-linker and it immediately forms polymers. This goes within 10-15 seconds, half a minute tops. If you do this, and this is a very nice control, that is also why I really like nanogels, in particular with precipitation polymerization, you will immediately see whether your reaction has worked. Sometimes my students come into the office and they say, well, we have nanogels, I think, but my solution did not become turbid. So, well, if it does not become turbid, you do not have nanogels. Because as soon as they polymerize and they form these particles, so these particles, they will scatter light. So the solution will become turbid. If it is turbid, you know that you have success. Instantaneously. And that is all because of this lower critical solution temperature. What happens really is that hydrated water, so this is poorly hydrated, but it's still hydrated, but if you increase this temperature, you increase the water dynamics, the polymers don't really like it, water goes away, it collapses on itself. This is where all the responses come from. Whether it's pH, whether it's temperature, the underlying phenomenon is always the same. All right, so now you know a little bit nanogels. Why do we need nanogels? Why do we want nanogels? So now I will go and discuss a couple of the examples that we have in the lab. And alongside I will hopefully convey a message to you how we design them. And what is each time the next step? How do we think in terms of translation? So it looks like a very simple particle, and it is, intrinsically, but you can do quite complex things with it. So you can change the chemical functionality, you can change the responses, you can tune the responses, you can play on the mechanical properties, but also in how you distribute your monomers. So you have all this control. So this control, where to start? Right? Because there are thousands of monomers. There are, well, I think hundreds of different applications. So where do you start? Well, basically we divide it in two. So we do suspension applications and development, but also interface, because they really like to go to interfaces. So I will just mention a few examples, a few more than I discussed yesterday, and I will also discuss them a little bit more in detail. So yesterday I stuck a little bit like, hey, this we can do, but now I will also show you a little bit more the underlying data, why we think that it works like this. And that goes from drug delivery to interfaces, bioactive materials. This we also do incorporate into 3D printed hydrogels, but I'm not going to discuss that, but I think it is a nice next step, going from suspension, single particles, to coatings in a 2D towards a 3D environment. All right, so if we're talking about material biology, then it is interacting materials and cells. That is the interaction, that is the interface, what I am interested in. So that is something that I would really like to investigate. Whether that is mammalian cells or bacteria, so I prefer mammalian cells, that is my major interest, but with bacteria, working with bacteria is also very convenient and very interesting. So if you look at cellular uptake, so nanoparticles being taken up by cells, that can be done via a couple of different routes. I'm not familiar with all these routes, but luckily my colleague Inge Zuhorn is, so she's an expert in cellular uptake, which mechanisms are being done. But what we're looking at is, hey, can we cross a barrier? How can we make it more complex? Because what you do generally is you look at cellular uptake, so you add a nanoparticle to a cell or a cell layer, you look, do they get in? But that is not fun. Well, it is easy to do, but it is not really applicable because you want to deliver it somewhere. So we thought, okay, let's go one step further, and Inge is working a lot with the blood-brain barrier. That is her passion. So we thought, okay, it would be a good idea to bring passions together, nanogels and the blood-brain barrier. We knew that stiffness is an integral part of interacting with cells. We heard the talk of Prashant this morning, right? It was all mechanics. It was all how does nature interact with mechanical properties? How does it evolve? How are cells dealing with this? So we knew from literature that a decreasing stiffness is an increasing uptake. There was literature about it, but that was just single cell layers, and we were interested in the blood-brain barrier. So the blood-brain barrier is actually basically a endothelial cell layer. So these are very close packed. So there are a lot of tight junctions. So only very, very tiny molecules can pass through the tight junctions. Everything else needs to go through the cell layer. And that means that it needs to go from the blood side to the brain side. That means that it needs to be taken up, but also be excreted to the other side. And this is what we thought, okay, if we apply nanogels with different stiffnesses, then they must have a different interaction. And whether that is uptake or whether that is excretion, we did not know. We just tested it. So we tested 4 different nanogels. So we based it on this, and we added a fluorescent monomer so that we could also track it and quantify it with fluorescence. And what you can see here is that just by changing these ratios, some of the reaction time, so it's not always a linear thing, so that if you wait twice as long, that the particle becomes twice as big, right? So there is a lot of experimental effort that you need to put in. But if you do this, then you get very nice particles, so in the range of 170 to 200 nanometer, and one large one which is about 420. So this gives us different stiffnesses, different swelling ratios. So these are either a 20 kilopascal in the low range up to 350 in the high range. All right, so I would just want to highlight one aspect from this slide. Because what you can see here is these ones, we designed them to have the same stiffness. They have the same ratio of cross-linker to monomer. But you see that there is a difference in deformability. So if we measure at room temperature and we measure above the phase volume transition temperature, and we can divide those values so it shrinks in size. And basically if you divide that over each other, then you get the swelling ratio. This one is higher, so the large one is higher than the smaller one. Why would this be? And in essence, I already gave the answer. So this is also an intermediate question to see whether you pay attention. There are different sizes, yes. But we use the same ratio of monomer versus dimer, versus crosslinker. But still we have quite, well, a bit of deviation in the swelling ratio. What would be a possible explanation for this? It has something to do with this. It has something to do with this. Sorry? The string? Yeah? Yeah. Well, you know, it's a free radical polymerization. So you have reaction kinetics, right? This molecule has 2 double bonds. So this one is consumed a lot faster in principle than the monomer. The reaction time was a lot longer. So in the end, you will most likely only get monomers reacting still on the outside, but they are still part of the network and they will still shrink. And the more they grow out, and the more you have your hydration layer, so the larger your particle. So in this case, most likely because of the longer reaction time versus the reaction kinetics of a crosslinker versus a monomer, caused a little bit of deviation. All right, so that is an intermediate question just to see whether you are still using your brain. All right, so we first looked at, so the system that we had is a collagen hydrogel with on top a densely packed endothelial layer. Really packed so that you can see the tight junctions, so nothing can go in between the cells. If we then look at the uptake, so we can harvest the cells, we can look at, okay, how much fluorescence do we have in the, on the inside of the cell. If we quantify that, then we see that the stiffer hydrogel, the stiffer nanogel, is being taken up more than the soft one. So that was already contradicting literature that we found, which is not necessarily a bad thing, because we use a different polymer, but we also use a different cell type. So this is also something that I really like. Nothing you can really predict in advance that clearly, because as soon as you change something, it might behave different. It is really difficult, but it's also something really interesting. We also detect that there was not really a difference in size. So even 420 nanometers would be easily taken up, and not a problem. Which is also, again, something interesting, right? Because if we talk about nanoparticles, then you usually aim for below 200. Well, here we have 420, and it doesn't matter. So again, something different that nanogels have over other nanoparticles. So based on these results, what we could conclude is that if we increase the stiffness, we increase the uptake. All right. But that is only part of the story. So if we want to look at transcytosis, going from the blood side to the brain side, we also need to look at the second step, namely excretion. Well, if we then look at, okay, how much of the particles are actually going towards the other end. And we can see that at the basolateral side we see less stiff particles and more soft particles. If we do the calculations, the transcytosis versus uptake is twice as high for the soft particle than for the hard particle. Meaning that uptake is, even though it's poor, the excretion is very efficient. And if you look at, so lower the stiffness, better the transcytosis. And the transcytosis is all these 3 steps combined. If you look at this layer, so what we see here, well, I think it's yellow, but it might also be green. I'm not that great with colors. So here you see the actin staining of the phalloidin staining from the endothelial cells. So this is the layer. This part here is the collagen gel, and what you see here in red, those are the nanogels. If you look, so this here you see it from the top, here you see it from the bottom. And what you see here, so I only took the soft one and the hard one, but you can also already appreciate that close to the membrane here, or to the cell layer, you see a lot of nanoparticles on the other end. A lot less for the stiff particles. And you do see some overlap here with the cell layer. So for some reason, the stiff particles either get stuck inside the cell or are being excreted back to the blood side. Well, if you have a soft particle, it goes poorly in, but once it goes in, they really want to kick it out, but on the other side. So that is, of course, very, very interesting, because again, that play says that if we play on mechanics, we can really tune what happens in biology, right? So low uptake still results in higher transitosis. And as it also mentioned yesterday, this allows us to redesign our particles. Can we make something dynamic? Can we make something hard in the beginning and soft at the end? All right. So I think closely related to this, because here we made, of course, use of a fluorescent dye, but the fluorescent dye was used for tracking, so for quantification. It was not designed for release. But we did a study, and I will skip this a little bit because these particles Heller already discussed as well a bit, and these are the standard particles for delivery. This, of course, is delivery for hydrophilic compounds, this more for hydrophobic compounds. A nanogel can do both, right? So that is another reason that nanogels should be taken up in this overview scheme. But to that I come a little bit later. So here we did a very nice study, and it was very, very simple, but it also will trigger the next step in designing nanogels that are in fact dynamic. So what did we do here? So also what Theo said this morning, looking at degradation of polymers via the macrophages, adding different inhibitors, and one of the inhibitors that was added were the inhibition of esterases. And esterases, once it was inhibited, the decrease in mass of the biodegradable polymer was lower. So the esterases really take a role in the degradation of esters. And that is also something that we wanted to use. So we designed a system where we took a FITC dye and with a conjugated acrylate. We just bought it, right? So this is not a complex design, and this is also something that you can buy. It's called now blue acrylamide, and here you have an amide bond, which is more stable. We use NIPMAM, we use BIS. We initiate it with a stabilizer. This you can forget for now. So what we end up is a nanogel with a labile compound and a stabile compound, which we can track by means of fluorescence. So the idea was, okay, if we add a particle with a labile label, fluorescent label, it will be cleaved and it will diffuse out into the cell. If we have a stable label, it will remain with the particle. Well, we made these two nanogels, one with a labile, one with a stable label. We introduced them to the cell layer, and what we can see for now blue, so this is the amide conjugated system, is we see all these punctuated dots. So every dot is not just one nanogel, it's probably a cluster of nanogels, but you can see that they are all localized. Well, if we look at the fluorescein signal, so the labile conjugate, we see a very diffuse pattern. So the dye is able to diffuse throughout the cell. That can only happen when the dye comes loose. Why does the dye come loose? Because the ester is cleaved. That is that was in our mind the only explanation. So we also mixed them. So in these experiments, we did one experiment with the acrylamide, one with the ester. Then we said, okay, let's put them both in the same system and see what happens. And we see exactly the same. So in the red, we see this very nicely punctuated pattern. In the green, we see it much more diffuse. We also see some, still some punctuates. So it's not completely liberated yet. And in the overlay, you can still see them both. Of course, you need to prove that it is because of esterases, right? So the first thing that we did, and I did not show the data here, but you can, so if a particle is being taken up by a cell, it's usually in an endosome, and in the endosome you have a low pH. A low pH is a catalyst for ester hydrolysis. The nice thing is that if you're working together with a cell biologist, Inge said, oh, but we can switch off this pH effect. We just have a small molecule which we add, and then we prevent the pH drop inside endosomes. So we did this, and we still saw the same pattern. So this means that there was not an enhanced hydrolysis simply because the pH dropped. Very important, because otherwise you look at the wrong effect. So then we still, of course, needed to show that it was because of the intracellular environment. Because what you could also think is that, well, it already is cleaved in the culture medium, and then it diffuses inside the cell. In our mind, that was not very logical, but still it is a possible explanation, so you have to investigate this. So what we did was, so the nice thing of these nanogels is that if you apply ultracentrifugation, you can just spin them down. And then you can analyze the supernatant. So we did this experiment, so we used the FITSI labeled nanogels. We incubated overnight in water. We incubated it overnight in buffer. We spin the nanogels down and we analyze what is in the supernatant. If the supernatant is fluorescent, then there is a little bit of, then there is a cleavage. What we could see is that if we mix it in with buffer, we have indeed a little bit of cleavage of this dye. However, and now comes the trick, what you can do is just grow a cell layer. And rather than putting the cell, rather than putting the nanogel inside the cell, you can also just open up the cell, suspend all the enzymes that are inside the cell, and mix that into your nanogel system. If it then still works, then you know that it has to do with the enzymes present inside the cell. So that is what we did, and you can see that there is a massive fluorescence output, meaning that if you add the intracellular components to your system, your dye is being cleaved. If we do the same with acrylamide nanogel, so where the Nile blue is conjugated via an amide, then if we analyze the suspension of the nanogels, then we see that there is fluorescence of the nanogels. If we then spin it down, either, so we incubate it in water and we incubate it in cell lysate, overnight, if we then spin it down and we analyze the remaining fluorescence, there is no fluorescence. So the dye is still connected to the nanogel, meaning that it is a stable conjugation. All right, so now we have two particles. And at the time we were thinking, okay, theranostics is a very novel approach in imaging and treating. And here potentially you have a delivery vehicle that you can also track, because you have two different systems. But that means that it is not a fluorescein or a Nile blue. No, you need them both in the same particle. Well, luckily it was as easy as this. Rather than just one dye, you add two dyes, you do the same synthesis, and you get them both in. Well, if you then do the experiment again, then again you see this very nice diffusive pattern of the green and the punctuated spots of red. Meaning that, again, the fluorescein is being diffused out by ester cleavage, and the Nile blue is remaining into the nanogel stable, right? So you can nicely envision, I hope, that also these are just fluorescent dyes. But you can also exchange the fluorescent dye for a drug. And so in this case, you can conjugate a drug via an ester, and that is being cleaved, and you can still track the particle. But now come back to the shape-shifting or the so the dynamic mechanical properties for the blood-brain barrier. So knowing this, what would be a logical approach? So if we go back, now I have to go back a little bit. So what would be a logical approach for redesigning this particle with the information that we now know? So what do we know? That a stiff particle is being taken up more efficiently. But if we want to have it excreted, it needs to be soft. So we want to maximize the uptake, but we also want to maximize the excretion. So what would you advise us to do in order to achieve this now that you know about the intracellular stability of different bonds? So what should we do? Yeah, William. A core shell? Yeah, so what would it be?
So the pH inside the cell will be dissolved waste outside, leaving a softer nanoparticle.
Well, if you would dissolve the shell, then you would probably leave with a stiffer particle, because most of the density is in the core.
Yeah, but and if you try to make a stiffer particle, and in the middle, a softer particle.
Okay, and how would you then do it? Because there is an easier solution. It is very simple.
I saw that there was an image of a particle with half, with like half a polymer and then half another polymer. So I think that would be probably an approach, like something that is stiffer on one side.
Yeah. Let me ask another question. What makes for the stiffness inside the hydrogel? The crosslinks, right? So if we are looking at this molecule here, so are these molecules, which one is causing for the crosslinks? The bis. So what is the bis? It is an amide. We can also make an ester. So if the ester is cleaved, then the crosslink is gone and the particle becomes softer. So the logical next step would be to repeat this reaction with the blood-brain barrier, but then instead of using a amide-based crosslinker, we just use an ester-based crosslinker, because intracellular it will be cleaved, it will be softened, and it should be. So that is actually the next step in the design, but that is based on what we found with the blood-brain barrier, what is important to cross it, combined with another study where we identified, hey, we can make use of selectively the intracellular environment. So for this idea, these two experiments, and so these two studies were quite crucial. All right. So if there are questions, then you're always allowed to interrupt me, right? I have more slides, but it is not necessary that I treat them all. So you're always allowed to interrupt me. So this is the system that we so far described, right? So we have here NIPAM or NIPMAM, we have BIS, we have different acrylates with dyes. And here, chemistry is key. And I have another example where in fact we exploit this chemistry and the alteration of the chemistry. But what is the chemical diversity in these two systems? And so at this point, I already said, well, chemical diversity is great because we can do so much with it. But this is the point where you should be critical. That's true. So you need to choose them well. In this case, they're all, so what we used here is all biocompatible. But here I would expect, well, Patrick, you're advocating for chemical diversity, but the both systems that you introduced here, they are NIPMAM and BIS. There is no chemical diversity. Right? The 1% of fluorescein that we added, that doesn't do much. So far we did not exploit the chemical diversity yet. So this chemical diversity, that is what we're going to discuss next, because this is very interesting. So of course, if you want to interact with cells, you can do this in different ways. You can add ligands, ligands that actually recognize receptors, which drive cellular uptake into a certain pathway. You can also just do it by changing the surface properties chemically. Or you do a combination of these. So what we wanted to do was making a bioactive nanogel. And bioactive in the sense that we wanted to tackle bacteria, bacterial resistance, right? So we wanted to make the life of bacteria a living hell. So we thought, okay, we do this in a double way. We try to make a system that intrinsically, the particle is intrinsically already killing, but we can also add antimicrobials to basically finish the job. And we based this design on quaternary ammonium compounds. Well, what are quaternary ammonium compounds? So there's basically a quaternary ammonium, so a nitrogen that has 4 covalent bonds, which is then permanently charged. And we know that these quaternary ammonium compounds, as you can see here, are interacting with the membrane of a bacteria. And what do? Because the surface of the bacteria is negatively charged, so these positively charged, they are really attracting to the surface, but because they are amphiphilic, and so here one of these tails is a long aliphatic tail, so basically they partially dissolve the bacterial membrane. So that was our vision. Okay, if we stuff this in the nanogel, then we have a small deformable particle that if it sees a bacteria, it will just rupture, right? So this is the synthesis. So we used NIPAM in this case, we use a tertiary amine acrylate, we use our BIS, stabilizer, initiator, easy peasy. So this is just again a precipitation polymerization. Now we have a chemical handle here, because this is still a nucleophile. It is still able to be protonated. So we did a quaternization with methyl iodide, so then we add one methyl group to the nitrogen, or we reacted it with bromododecane, and then in that case we add a C12 tail to this nitrogen. Yeah? So a very short one or a very long one. And of course we expect that the very long one is doing the trick, but we wanted to make the very short one, so the methylated one, as a control. Right? Because you really want to know where the effect that you find is coming from. So in this case we have one that is positively charged because of taking up a proton, because it's a base. This is a quaternary ammonium, but then with a methyl group added, and here a long aliphatic tail. So this is the characterization of the particles. So this is them, so we nicely see that there are particles, there is a certain distribution, it's all good. But in our mind, we added, so if you look at this structure here, and now I go one structure back, so you have here a quaternary ammonium, and there's a long aliphatic tail. Here we use CTAP. CTAP is a quaternary ammonium with a long aliphatic tail. So it is a surfactant. It is a soap. What does the soap do? It aggregates. It aggregates into micelles. So what we thought was, hey, maybe these chains, these hydrophobic chains, maybe they will see each other inside the network. Maybe they start aggregating in there. And this is something that we wanted to investigate, whether that was in fact true. So what we did was we took our, so we used Nile red. So Nile red is a bathochromic dye. So it changes its fluorescence intensity on the environmental polarity. So in water there's hardly any fluorescence, but in a hydrophobic environment the fluorescence shoots heavily up. So this is a very convenient way of identifying whether there are hydrophobic domains there. It is also used to determine the CMC in surfactant systems. And this is what we saw. So if we have our nanogel and we mix it with Nile red, we don't see any appreciable fluorescence. If we mix Nile red with our quaternary ammonium C12 nanogel, all of a sudden we see a large output of fluorescence, meaning that there are hydrophobic domains present. And then we thought, okay, then we can also do this with a hydrophobic antimicrobial. And in this case, triclosan, and triclosan has the same effect if it comes into a apolar environment, the fluorescence or the absorption changes, and that is also what you see here. So we created a particle that intrinsically has the ability to interact with bacteria because of these quaternary ammonium compounds, and additionally we created hydrophobic pockets inside this nanogel where we can store hydrophobic drugs. Right? So in the first part we had a hydrophilic component attached covalently to the network, and now we have hydrophobic drugs solubilized inside hydrophobic domains that are inside the network. All right, so these are our 4 controls, and this is what we tested. So we tested 4 different strains, and we determined the MIC and the MBC, so the minimal inhibition concentration and the minimal bactericidal concentration. And what we saw was that if we look at just the tertiary ammonium nanogel and the nanogel that was quaternized but just with a methyl group, we see that there is no appreciable antimicrobial activity, which we also did not expect. However, this was initially a little bit disappointing because we thought, okay, the C12 quaternary ammonium compound, that will really do the trick. Well, we do see that there is a concentration in the microgram regime, but it is not really right? We were a little bit disappointed. But still, we could find that there is a concentration at which bacteria are being killed. So that means that the nanogels do have an interaction and an affinity towards the bacteria. So of course we tested triclosan. So triclosan is known to be in the low microgram per milliliter regime in terms of killing effect. But, and this was the really awesome part, because if we take our nanogel, we solubilize our triclosan inside the micellar domains, all of a sudden, and I will show it here a little bit more in detail, so we compare triclosan, this is triclosan, versus nanogels where triclosan is hosted inside these hydrophobic domains. And what we see is if we look at the minimal inhibition concentration, for some strains it's 1,000-fold more effective. If we look at the MBC, it is up to 800-fold more effective. That means that you can get away with 800 times lower concentration to kill the bacteria. Right? We were really happy with this because that actually meant, so yes, our particle itself does not kill, but it is bioactive because it does interfere with the bacteria. So this is the mechanism that we propose. We have no hard proof for this, but what we envision is that is happening is that you start out with this attraction, because the quaternary ammonium compounds, they are positively charged, so you still have this attraction to the cell membrane of the bacteria. Then the aliphatic tails inside these micelles, they start to intercalate into the membrane, right? Because we know that quaternary ammonium compounds with long aliphatic tails, they do this. They tend to dissolve the membrane. But of course, you have to realize that you have a lot less freedom here. So these are not free-floating quaternary ammonium compounds. They are trapped to the network. So probably the effect is not strong enough to completely dissolve the membrane. But since the micelle here is opening up, intercalating, and thereby it does make a pore inside the membrane, and it actually pushes the triclosan inside the bacteria. Because what happens most of the time with a hydrophobic molecule and a membrane, it gets stuck in the membrane. So the bioavailability to do the trick where it needs to do it is inhibited. But now you open up the membrane and you actually shoot in the drugs. It's much more efficient. So we were really pleased about this. And then we said, okay, we have it as a suspension, but we also would like to make coatings out of these particles. And this is very recent work, and this is I really like it. I have to say it because one of my PhD students, a couple of my PhD students worked on it, but I find it very elegant because it's simple. And if something is simple, it makes me happy. So what we did here is we made NIPAM APMA bis nanogels. So we create a primary amine in there. Primary amines in water, they take up a proton, they become charged because it's a base. What we then do is we take a material, and it can be any material. It doesn't matter. We stuff it inside a plasma oven to oxidize the surface just a little bit. It renders the surface negatively charged. If we then spray, and that is really, well, you know, the spray that you have at home, maybe for the perfume, that is the spray. So we fill this bottle with, that it can spray, we activate the surface, we spray on it, we let it dry, we wash it, it's done. We have this very nice homogeneous coating of nanogels. You see the individual nanogels here. We did this for glass, we did this for polycarbonate, for PMMA, Teflon, you know, a whole range of medical relevant materials. And what you can see, so here, this is the native surface without nanogels, and below you see the coated versions. So just plasma treated, sprayed, and then dried, washed. Nothing more. And you see in each case that there is a very nice dense packing of all these nanogels. Really easy, really applicable. Anyone coming in the lab can do it within a day. It is that easy. So if we then focus first on glass, because glass is nice because it's transparent, so we can find some interesting properties. So this is also something that I told yesterday, so I will go through it a little bit quick. So we looked at the anti-fouling capabilities of these coatings. What I said, so these coatings, so these nanogels, they are really hydrophilic. So if they're really hydrophilic and swollen, then if a bacteria comes and it needs to push it, then of course there is a penalty, an energy penalty. If the system doesn't allow it, the bacteria does not attach. So we made different nanogels with, nanogels with the same diameter, but with different stiffness. And what I also told yesterday is that if you apply these to a surface as a coating, then of course they start to deform because they interact with the surface. The softer they are, the more they deform, the thinner your coating will be. Well, these are the coatings, and now I missed the. Well, I missed the anti-fouling capabilities. Well, they're here. So what we could see is that we are about almost 3 orders of magnitude, a little bit less, 2.5 orders of magnitude, less in bacterial attachment if we apply this nanogel coating. Of course, there is deviations in the thickness of the coating, so it is not completely fair to compare a 10 nanometer thick coating with a 60 nanometer thick coating. So we did a lot of experimental effort in order to make larger nanogels that would resemble the same coating thickness. And what we could see was that indeed if you have a soft nanogel but with a good thickness, then it is more efficient than a equally thick coating with a slightly stiffer nanogel. So the thicker the nanogel and the softer, the better the anti-fouling capability. So we knew, based on this study, that it is anti-fouling, right? All right. So if something is anti-fouling, and of course, Theo discussed this morning as well, because what happens when you introduce something into the body, proteins will absorb. As soon as proteins are absorbing, then you know, okay, cells will respond to it. So an anti-fouling character on a surface will maybe reduce the foreign body response. So we did a study where we coat Teflon and polyvinylidene fluoride. So this is in fact a hernia mesh. We looked at it with IVIS. So IVIS can pick up fluorescence quite easily. And what you can see is that once we coat it, you can see that there is fluorescence detected there. This is how it looks like. Then of course, before you want to test it in vivo, you would also like to see whether it is stable. So here we have Teflon coated and we stored it in a shaker with PBS for 3 weeks. And we could see that after 3 weeks, we could still see homogeneous fluorescence. We also did it in serum. So PBS is kind of mild, it lacks all the proteins, it is not that complex. But also if we expose it to FBS, then we see that after 3 weeks we still see fluorescence. So the coating remains there even though we shake for a long time in an aqueous medium. So then the next step is of course to test it in vivo. Since we have fluorescent micro nanogels as a coating, we can study it with the IVUS again. So here you see a sham. So this animal, this mouse underwent the surgery without the mesh. Here we implanted a mesh that was not coated, and here we implanted the meshes that were coated. And you can still see that after 13 days, 2 weeks, we still see that the fluorescence is there. So even in vivo, at least for a short period, it remains stable. We did not test it longer, but that will be in the next phase. But then, of course, you want to identify whether it also does something. Well, we did histology on these, and here you see the whole of the mesh. And so this one is the coated, this one is the uncoated. So this is the table that summarizes. To our, well, disappointment, we could see that after 13 days, there were hardly any differences between the coated and the uncoated. So there were no really long-term effects, or long-terms, well, it's not really long. But what we could see was that after 7 days, we did see some appreciable differences. So we could see that there is only a slight number of surfaces that are with collagen. So if you look here, a lot of the collagen in the coated ones is not there, meaning that there is not an excessive deposition of collagen. What we also see here is the formation of foreign body giant cells that actually are involved in the foreign body response. Also there, we see that they are not there for the coated ones, but they are there for the uncoated ones. Unfortunately, that effect is gone after another week. So we do need to tweak it a little bit. But at least it behaves correct. So there is no strong adverse effect. And very short term, there are some differences. But something that I really like, and I also, we discuss this more often, what I really like about this is not so much the whole system. Of course, I like the system, but this is really a team effort. And this is something that I do want to highlight, because if you look at the names here, so Patrick van der Wel, he's at the Faculty of Science and Engineering. He's a biophysicist. He is an expert in solid-state NMR. Without him, we could not determine how much of the different components are in our nanogel. Nanogels are extremely difficult to quantify. The molecular weight is difficult to quantify, but also the exact composition, because they are too big to study properly with solution NMR. Quantitative FTIR is a bit difficult. So Patrick really helped us with identifying with solid-state NMR and identifying how much of each component is actually inside the gel, which is extremely important because you have to realize that every monomer that you use has a different reaction kinetics. So that means if you insert a 10:1 to 0.1 ratio of 3 monomers, does not mean that your polymers reflect the same composition. If the 10 is reacting really, really slow, then the balance might be completely different, right? And that is something that you do need to know. Of course, Theo, you all heard Theo this morning, cell biologist, crucial for looking at the foreign body response. Max Witjes is a surgeon, so we wanted to have input in how do we apply it, what is relevant. We have here Jelmer Solema, colleague of us, expert in how to do imaging. Henny van der Meij, professor in microbiology, and the chemist. So it was all these disciplines that were absolutely needed to pinpoint what was happening at every stage of the research. And I think that is something that I really, really enjoy, because you make use of everybody's expertise. And you create a very nice result. All right, so we now know these nanogels are antifouling. Are you still all okay? Or do we need to do some exercises to keep the blood flowing? I have no idea what time it is. So if no one stops me, we will be here still tomorrow, right? So I think I'm now more or less quarter of my presentation. No, so I still have about 30 slides, I think. So I think so it was not even 6, right? 6, okay. And we have until 6:40. Oh, okay, good. Then we can slow down a bit. So we designed nanoparticles. We now know that they are anti-fouling. We know that they will be there and remain there in vivo. We have highly effective antimicrobial nanogels. So then, of course, a logical next step is, can we take the nanogel that we designed for suspension killing? So the first example was all suspension killing. Can we transform that into a functional coating? So we now know how to make coatings. So what we did was we took the same 3 nanogels, so with a tertiary amine there, modify it with an aliphatic tail, and introducing triclosan. So we have basically 3 different surfaces. One that has no quaternary ammonium there, one with a long aliphatic tail, and this one is loaded with triclosan. It's very easy. So this we already knew, the preparation of this nanogel. There's also, because it's exactly the same as our previous work, and we know that if we plasma coat the surface, so the quaternary ammonium is already positively charged, so it will stick to a negatively charged surface. So it was really convenient to take one and the other and combine it. If we then have this coating and we dip it in triclosan, then it will be taken up, because there are micelles inside these nanogels. So it is basically the same procedure. If we analyze it, it still looks exactly the same. So all the nanogels are still there. And then we looked at the anti-fouling character. And I have to add that this is under flow. So what we do is we take our glass slide, we add our coating to it, and we put it in a flow setup. That means that we have a bacterial suspension, and we flow it through a flow cell across our surface. Well, what we then saw was that the nanogel had quite a good reduction in the number of bacteria adding to the surface. It was not as good as our previous nanogel. Yeah, so our previous nanogel was about 99.7% reduction. This was, I think, 97%, 97 point something. So why would it be worse? So this is our nanogel, which is this one. So there is NIPAM, there is BIS in there, and there is our tertiary amine. Why would the anti-fouling be a little bit worse? Because it is kind of logical. So what is the charge of the nanogel? Positive charge. What is the charge of a bacteria? Negative. Yeah? So there is enough charge there that some bacteria will remain. And in fact, it even becomes worse if we add our quaternary ammonium nanogel there. Why does it become worse? It does not become more charged. So if it doesn't become more charged, why would it attract more bacteria? So what does the quaternary ammonium compound do? It intercalates into the bacterial membrane, right? So there is and a charge interaction and a hydrophobic interaction of these long aliphatic tails. So in fact, this is not a really good anti-fouling compound. It became a little bit better, but not really if we added triclosan. So we were a little bit disappointed in this. Then of course, we look a little bit further, we look a little bit deeper, because now we're not only dealing with anti-fouling, we're also, of course, dealing with something that kills bacteria. And also this, again, has been done under flow. So the samples that in the previous experiment, after flow, we take them out and we characterize, okay, what happened to the bacteria. What we do see is that most of the bacteria are dead once we add triclosan. And we also have more dead bacteria if we have our quaternary ammonium nanogel there. So it is less antifouling, but it is more killing. So there is a balance. How far do you need to go? What do you prefer? Do you prefer antifouling or do you prefer killing? Right? Do you prefer it under flow? Or, and this is a little bit what saved us, because in all honesty, these are not highly impressive results, right? The concept is interesting, but it did not work under flow that drastically. But my PhD students said, we also did it under static. So what they did was a parafilm test where they added the bacteria and then put a parafilm on top. So they have a suspension of bacteria there, they immobilize it with a piece of parafilm, and then they look at the colony forming units. And what we could see is under static conditions, it went down almost 6 log units in terms of colony forming units. And that is impressive. So the static conditions saved us a little bit in making it a little bit more impactful. So under flow, could be improved. Static conditions works very, very well. But it shows that you can take something that you learn from suspension and add it as a coating. And we did not do anything different, right? We just applied our universal coating approach. So I hope that I took you a little bit along in our way of thinking. So what can we change? How do we change it? And can we change it from, can we apply it from one system to another system? How does it behave? What can we learn from it? Because so far, we applied it first just how do we deal with barriers? Does it go in? How much is secreted? If we put it inside cells, how can we actually release something that is on the inside? Can we make it bioactive? Can we translate our nanogels into coatings in a universal coating for any implant, because you don't want to reinvent every time a new coating? How does it behave in vivo? Can we apply it really as a bioactive coating? And what I will not show is how we implement it in 3D printing. Because I hope to come back in a couple of years maybe here again, and then I will tell the next phase of the story. But, of course, I would not only want to highlight the things that we do, right? I like what we do, but I also like what all my friends are doing. During the first lecture of Theo, someone noted neuronal regeneration. And yesterday, after the lecture that I gave, someone came to me, so are you also doing tissue engineering? And I had to admit, well, I don't. But I do know people who do. So I really wanted to highlight this system because I also find it very elegant. And it solves something which is extremely difficult to do. So this is the work by Professor Laura de Laporte. So she is professor at Aarhus University, so the RWTH. And what she does is absolutely amazing. So what she does, she designed microgels that are anisotropic. So in the beginning, I showed you this nice overview of, hey, we can have different particles. We can also control the shape of the particles. Well, she can control the shapes. Right? We just make spherical nanogels, but she can make anisotropic ones. And what she does is she incorporates magnetic nanoparticles in there. So she makes use of another material to introduce new properties. And then what she does is she mixes in these micro anisotropic microgels inside a polymer matrix that chelates. And once it chelates, they get stuck. But here comes the trick. Because she has an anisotropic structure filled with magnetic nanoparticles. She applies a magnetic field to it. All the structures, they align to the magnetic field. At that point, she is introducing the cross-linking. So she has this directionality inside a 3D matrix, right? When I first saw this, I was like, wow, right? We do a lot with anisotropic structures, but only on 2D, because we find 3D too difficult, right? But this is how to do it. And her idea was to use this for neuronal growth, as an injectable for making neurons grow again in the proper direction. So this is basically how she does it. So she has a mold, so something like a Teflon type of material. So she fills it up with these pre-polymer materials, with the magnetic nanoparticles. She seals it up, she cures it with UV, so it's UV-induced cross-linking, and then basically she pops them out and puts them in water. If she then applies in solution a magnetic field, so here there's no magnetic field, so you see that they are pointing all directions. She applies the magnetic field and they all align, right? So they instantaneously do this, and they all point the same direction. And then, so what she then does, so she takes the neuronal node in the neck of chicks, so baby chickens. They take them out and she puts them inside the microscopic hydrogel. But inside the hydrogel, she puts these anisotropic microgels, anisogels. And with increased concentration and a magnetic field, you can really see, so red is all the neuronal cells that are growing out, and green is here all the microgels. And you can see that they're all in the same direction. And you can see that all the neuronal growth is in the same direction. And this is something that she uses, or wants to use, as an injectable for spinal cord injuries. Right? Normally, you would put in a scaffold, and the scaffold is porous, and all the pores are in one direction, to make sure that the neuronal growth is in the right direction, because it's not so much for neurons that they don't grow, but they grow in the wrong side. So if you have two ends, they want to go towards each other, but they do this, not going to work, right? But rather than putting in an implant, she just does an injectable, she places it in a magnetic field, she polymerizes the system, and all the neurons can grow in this direction. I find it absolutely amazing. So this is something that I really wanted to share with you, because I think this makes it even more worth to place the nano and microgel in this box. So if you are really interested in this, so they wrote a really nice review here, particularly on clinical applications, together with Alexander Kuhne. So Alexander Kuhne used to be in Aachen, but is now in Ulm, and he makes all these nanogels using microfluidics. So also there, you see that the technologies that, for instance, Helder also discussed, are also applicable to microgels and nanogels. If you want to know more a little bit about the antimicrobial approaches using nanogels, then you can read this nice review. They are both reasonably recent. But I hope to have convinced you that nanogels in the future should be an integral part in the box of nanomedicine, because it is incredibly versatile. All right, so these are the people who did all the work. So without them, all these systems were not possible. Sometimes so this is ice. You don't get probably much ice in Mexico. We don't get ice in the Netherlands either, or at least not that much, maybe 1 or 2 days a year. But every Christmas we go for curling. We make a small competition out of that. So these are the people that all do the work, and they do fantastic work. So thank you for sticking with me all to the end. If there are questions or remarks, then you want 2 microphones?
Here, hey, Professor. Thanks for the really interesting talk. So I had a few doubts, a little questions there. So first, like in the monolayers, when you do the coating, do you know if they're like monolayers or they're several layers?
Yeah. So what we do is, so that is a good question. I didn't show it, but what we do is we make a scratch in there, and then we look at the height, and we really see that it's a monolayer. And that we do on glass, because if you look at all the other materials, so they are kind of rough. So you have to do it on something which is really hard, but also relatively flat, so that you can actually do the height. But it is indeed a monolayer.
Okay. And the other one is on the nanogels with triclosan that are loaded with it. You measured the antibacterial effect. Did you measure the cytotoxic side effects of that?
Yeah, yeah. And again, I did not show this. They are toxic. So whatever you do, so positive charges for cells, they are generally very toxic. But what we found, and that is a little bit where you have the playing field, because we have a 1,000-fold or 800-fold increase in activity from the antimicrobial side, we only need a very, very low concentration of our nanogels. So the concentration that we needed is far below the concentration at where it is toxic. So yes, it is toxic, way more toxic as when we would just use a NIPAM nanogel. They are not toxic, right? You can actually bury them and they will still survive almost. But we can, so they are toxic, but the effect is far below the toxicity. But it's a very good question. Sorry?
Which is the application of nanogels of biotechnology?
So what are the applications of nanogels in biotechnology? What do you exactly mean with biotechnology? So is that so generally we focus on biomedical? Do you mean that, or so in principle I would so we're now quite far in developing this as a coating. The coating is really easy. But the problem with, and that is also a little bit the problem in general with translation, is that you, so it can be stable, so we tested inside an animal, it remains stable, but the way how we deal with it in cell culture and in research settings is that we take our mesh, we coat it, we dip it in 70% alcohol, we rinse it, and we put it in. But that is not how a surgeon does it. Right? So we were thinking about, okay, how can we translate it? Is it something that you would do at the end? Yeah, but that means that you need to do quite a lot right before surgery. That is something that you don't want. So you would preferably want to do it already at the beginning, before it ships. So one of the things that we looked at is sterilization techniques. Right? So we took all these different, and there was a tremendous amount of work. So I feel really sorry for the PhD student who needed to do this, but in the end it was a really nice study, because what we did, maybe I should go back to these, to this overview, because that makes you appreciative for how much work it in fact is. So here we have 10 different materials. So we tested 6 different sterilization techniques, right? So for 10 different materials, of course in triplicate, with each time a technical duplicate, and we used things that we generally had in the lab, but also what industrially is being used. Yeah? So in Groningen, near the hospital, we have Steri Noord, and Steri Noord is the place where all the instrumentations are being sterilized. So they have a couple of techniques for that. we just gave them samples, coated, and said, well, please take these coatings along in your sterilization process, and then we reanalyzed them. Not only for, is the coating still there, but also, does the coating still function? So does it still inhibit bacterial adhesion? So that was a tremendous amount of work, but what we could find was that it depends on the material combined with the sterilization technique, whether it remained stable or not. So we could pinpoint for which material combinations it would work and for which ones it would not work. So I really think that these could be very interesting coatings for implants, but also for sensors. And I did not show this, but it's actually a very, very funny story, because we were doing these anti-fouling approaches, of course. And then a group from Portugal approached us and said, well, we have a fantastic biosensor, but it would work a lot better. So they had biosensors on a chip and with certain ridges, but the idea was that the bacteria should only be at the bottom, because then the sensor would be a lot more accurate. So they said, well, we want to have the top anti-fouling. So basically what we did is placing nanogels on top of these ridges selectively, and immediately all the bacteria went down to there where they needed to be. And so also for coatings in these, not in vivo, but for sensors to direct how things are behaving, I think also there would be quite a nice possibility.
Thank you very much.
But of course also for drug delivery, right? Nanomedicine.
Hi, thank you. I have two questions. I don't know if you can ask them. The first one is since nanoparticles are so small, how did you quantify how much of your nanoparticles were releasing the drug? Because right now my imagination just gives me for, I don't know, like a dissolutor, like for capsules, something like that, but you will need like a lot of sample. So how do you quantify that?
How do we quantify the release?
Yeah, like how much of the drug was being released by the nanoparticles in the conditions that you want them, like the pH, temperature, thing like that?
So let me just, which one is it? So let's take the, so for this one we looked at it, because here it is relevant, because here we have a drug that is non-covalently bound and it can actually leave. We incubated it in water and we tried to detect it, but we could not see any release, because triclosan is very poorly soluble, and only when we would wash it with ethanol and more organic solvents, then it would be leaking out. But we did not find any leakage, so it still remained there. Which also explains, so this also explains the killing effect, because even under flow, so if you have flow, you each time have fresh water on the interface. So under flow, it would wash away a lot quicker than when you just dip it in. And even there, all the activity remains, so it would just not come out. We did not test any other release because, well, we did not have any other release to measure. Right. So for the release of these of the fluorescent dye in our intracellular ester cleavage, there we just measured the fluorescence. So based on the fluorescence, you can just make a calibration curve, and then you can determine the amount that was liberated. Right. But for other systems, so we are also dealing with other systems where we try to quantify release of hydrophobic compounds. That is incredibly difficult because they are not soluble.
And the second one, well, right now, and where we're working with micrometric scale particles. And since you already know the difficulties of trespassing barriers in the body, would you recommend that these particles pass to in vivo, the in vivo stage, or just keep going down on the size?
Sorry, can you repeat?
Yeah, we're already working with microparticles, like they're 100 micrometers. And since you already know the difficulties of trespassing barriers in the body at nano scale, would you recommend that these microparticles we continue on the in vivo stage, or we could try to keep going down on the size of these particles?
Yeah, so I think there is a limit in what. If you're talking about a cellular uptake and passing to the brain side, there is absolutely a limit. So you should there have small particles. But it also depends a little bit on the particles. So generally, so there's also what we showed is that so for PLA particles and that sort of thing, you would aim for below 200. But if you have nanogels, so Andrew Lyon, so one of the authors on the initial review that I showed, so they did an experiment where they pushed a 1 micrometer nanogel through a filter of 20 nanometers. And it just came out as 1 micron in the back. So these are really deformable. So then the size is so it doesn't apply anymore. Well, it applies, but in a different regime. So I find it a very difficult question to answer.
Thank you.
Hi, thank you so much for the lecture. I have two questions. First, how do you determine the degree of cross-linking at that scale?
Well, basically, we don't assign a degree of cross-linking. We just look at the deformability. So particularly if you, so let me just.
The stiffness?
Yeah, it's well, I don't think it's here. It's somewhere. So what we are looking at. So so the degree of cross-linking. It is not something that we are immediately thinking of. We are thinking more in terms of what does it do to the properties. So if you have a high degree of cross-linking and low degree of cross-linking, then you can determine that by how they behave. And with behavior, I mean, so if you, so what we do is we measure the hydrodynamic radius at 20 degrees C, and we measure it at 50 degrees C, which is then above the volume phase transition temperature, so they shrink. And if it is really highly cross-linked, then that difference is smaller as when they are not highly cross-linked. We can also just measure the stiffness of these nanogels with AFM, and then you will see that they are lower in stiffness. So that has to do with the degree of cross-linking, but we don't put a number on the degree of cross-linking.
Okay. And the second one, do you work with clay chemistry?
Clay chemistry, no. No. That is a good addition, but we are not there yet. And that is also a little bit with, so I showed you just a small overview of the number of acrylates that we can use. On top of that, there are all these very interesting secondary chemical reactions that you can do, right? Click reactions are one of them. So there are so many different combinations that you can do, and they are all extremely all extremely interesting. So it is really difficult to make a good choice. What do you want to do? So yeah, it is a very good intuitive, but we did not do it yet.
Yeah, no, sorry. Thank you.
Yeah, thank you, Patrick, for the nice teaching lecture. Amazing. I have actually two technical questions about the work that you showed from Professor De La Porta with this anisotropic nanogels or microgels that she's working with. The first question is that how she can control, of course, X and Y direction and turning the direction of this material is very important. But in your 3D network of nanogel or microgels, we have also Z, not just X and Y. The first question is that, are these anisotropic structures by magnetic field evenly distributed all through the nanogel or microgel, or they are just in one Z level? This is the first question. And the second question, it would be great if you know about that and share your idea, is that how she can control not shifting all these anisotropic structures to the N or S pole of this magnetic field.
Yeah. So that so she uses a ethylene glycol based hydrogel. So of course that is already quite more viscous than just water. So within an even mixing, so the diffusion is not that quick. So there needs to be a ratio between how long you apply the magnetic field, because if you apply it too long, then they can also move towards it. So there is a balance. Right, so how exactly the details on that I don't know, but I'm pretty sure that they did it very, very particular, because she's very precise in all these things. And so this was the paper at 2017. I think by now there are already 10 papers on this with different cell combinations, with muscles, with so they did a lot of work, and it always really, really works. And the nice thing is that it's in 3D. I think that is particularly interesting.
Thank you.
Well, hi, doctor. I have a question. Have you ever explored use pH-sensitive polymer instead of a thermoresponsive polymers like SNIPAM? Because I think the pH of the cancer cell, for example, is acidic, so the polymer maybe shrink and release the dye or the, I don't know, the antibiotic or whatever.
Yeah. No, that's a very good question. So we do use so the tertiary amine and the primary amine nanogels, so they are pH responsive. We also use acrylic acid nanogels, and they behave based on pH. And so we do also. So if we do the titration with acid and base for the amino nanogels, we also see them swell and shrink based on pH. The difficulty with cancer is that the pH that is deviating is a little bit of an awkward pH, right? So there are not really monomers available that shift, because you need to be at the pKa what the environment is of the cancer, and that is a little bit of an odd pH for the monomers that we generally use. So for that we would need to redesign a new monomer, which is a really good point that you make, and we probably should do it, but then I need to talk to a real chemist, because I'm no longer a real chemist.
Hi, Patrick. Very nice talk. You really made a strong point in favor of the nanogels, and so I have two questions. The first is, why did you move away from proteins? And the question is, how can we combine these two worlds? No, I mean, I understand the functionality, the versatility of the polymers, but also, I mean, we know the proteins have some reason to be, no, and they could be combined. The worlds are different. The proteins have other logic, sensitive, some of them, but also advantages that maybe together can be even better.
Yeah, no, so I totally agree. So from proteins, I did not specifically move away per se. But the thing is, there are only limited number of ideas that you can actually do. I had a very talented master student at one point, because I had the idea of using, because it is known that these polymer networks are stabilizing proteins. So you know that the work of Francis, right, with the TMV, where they put a polymer coat around it, and then all of a sudden they can put it in organic solvents, and the virus remains intact. And that really also inspired me, in the sense that what if you can deliver proteins that that have a specific function, and you can deliver it into the cell, and then it takes over the function because the protein is not being expressed properly, right? And then I had I pitched this idea to a master's student, a biotechnologist, and he was extremely clever. And what he did was he put in a horseradish peroxidase inside the nanogel, and then it remained active. It retained its catalytic effect. He could also switch it on and off, and then he moved away. Right, and it is still a very interesting idea, which we want to explore in the future. But I think, particularly, also what's in the last example of Laura, these combinations, right? So it's not just magnetic particles, it's not just gold, it's not just nanogels, but these combinations of all these functionalities. That is where we really find the next generation of materials, I think. So I did not move away per se from proteins, it's still in my mind. But I also think that there is something that we definitely can do together, also relying on the self-assembly of proteins, where we can add nanogels as templates, and really do some cool hybrid stuff. Right, and where it is good for, I don't know yet, but I know that you already said that you're in favor of playing around, right? So can we do it? What is the function? We don't know yet, but so I think that there is still a lot of ground to be taken.
Hi, thank you for the lecture. It was really cool. I have two questions. The first one is really quite simple, and it is: have you done this research in vivo organisms like complex?
Sorry, the last part I didn't.
In vivo organisms, like complex ones. Have you done it?
Complex organisms?
Yeah, like humans, for example.
Oh, no. No, we had,
mice?
So in mice we applied the coating. So right. the anti-fouling coating. But we did not apply yet the antimicrobial coating. So this is, so you can see it from the publications, this is all the work from the last 2 years. So we are now at the point where we say, okay, what can we explore? So for this, we did not test it further in animals. No.
All right. And the second question is a bit hard to explain, so just feel free to ask me back. And the thing is that in one of the slides you presented this nanogel which contains these particles that were capable of reorder the membrane in order to penetrate, right? And the thing is that nanogels, as long as I understand, they have not exactly a good control of the liberation. So a molecule like this could be really dangerous in a full organism, especially the administration way, because it could just reorder all kind of membranes, damage barriers like blood-brain barrier, and so how could you approach to resolve this problem? Because it seems like really tricky and quite dangerous, and how do you control that after penetrating the tissues that you wanted to, it actually release now the degrading membrane compound?
Yeah. So in all honesty, I would never ever apply these quaternary ammonium compounds to anything, right? And that is something that. So generally, I first like things to see whether they work, right? So there are always 1,000 questions that need to be answered before you can actually apply it. But the question always is, are you already thinking about all these questions before you know that it works? Or do you first do a quick route to see whether it works and then re-engineer your system to such an extent that you know, okay, that is what we can implement. So basically where's my bioactive? Yeah, here. So if you look at these ones, so there is absolutely no degradability in there, right? Which is already the first thing that we need to tackle. Yeah. Luckily, we can quite easily exchange these ones for esters. Yeah, so we will be able to degrade them into smaller fragments. So that will already be one part of the journey. After that, so if you look at this structure here, so you will still have then fragments of all these quaternary ammonium compounds. If they are liberated and they are integrating into cell membranes, they are doing stuff. So also there, I would probably start redesigning it so that also the tail can be cleaved off. So in the future, we need to go further and implement how do we break down the particle. So this I would not introduce yet, because for that it's too preliminary. On the other hand, so the amount that you use is really, really tiny. So if we do, so we did these toxicity assays on cells, and we do see that if you apply it to a low concentration, they're not toxic. But that does not mean that they don't have any effect. So they don't kill, but how does it? What is the behavior of the cells which are being taken up? Right? They are not killed, but maybe they are transformed into something different. We don't know. Right? So we have to redesign this particle to such an extent that it can be cleared, that it is less prone to do stuff.
Right, thank you. And just one last question that just came to mind. You say that you don't have really a way to control the release yet, releasing this, but there's maybe a chance that you could integrate the nanogels microstructure and make it maybe fusionate with the drug delivery that Dr. Santos told us, to make it like a coat that is quite sensitive and then dissolve to expose the nanogel, the sponge.
Yeah.
Maybe.
Yeah. So one of the things what would be so you can make these really nice copolymerization systems, right? So what you generally see is that if you so the volume phase transition temperature for NIPAM is 32 degrees. If we do a copolymerization with a tertiary amine or a primary amine, you will see that this shifts. Because if you compress the nanogel, so all these amines are charged, you want to, so then you also need to bring charges together, so you need more energy for compression. So I could imagine that if you play on, even if it is a fraction of the pH, so if you introduce a thermoresponsive polymer that is collapsed at 37 degrees. So you have your compound, you put it inside your nanogel, you collapse it, it is entrapped, but you have a copolymer there which is also pH sensitive. If then all of a sudden it goes to a regime where it becomes protonated, then this volume phase transition temperature changes, and then it is still able to open up. Right? So there are all these tricks that you can do to tune it to such an extent that you're at the edge of, okay, now it works, now it doesn't, right? Because you have only very small windows where you can play with, right? So you cannot go from 30 degrees to 50 degrees. You have to go from 37 degrees to maybe 40, 41 degrees, right? Because if you have inflamed tissue, it is not going to be 50 degrees, right? Or you need to play on responsive polymers that are triggered by light, or an external a magnetic field, or something else. So I think there are still a lot of combinations that we did not test. But I'm pretty sure that there are combinations there that do the trick.
Right, thank you.
Hi. My question is why you choose the spray coating. I think it's difficult because you may have agglomeration of the particles. And the first question, you say you need to wipe the surface to maintain or to obtain the monolayer. Why you choose that technique and not deep coating or Dr. blading maybe? It's because it helped you in the size of the nanogels in the little droplets, or why?
No, so spray coating. So we did not come up with the spray coating. So in fact, if I go all the way to the end, so at one point I visited my postdoc supervisor in Potsdam, Alexander Beuker, and I came into contact with Klaus Duschel. And Klaus Duschel was collaborating with Thomas Helbeek, and they developed this spray coating. It is very, very convenient. It is absolutely, because the beauty is that you activate the surface and you spray on it, and yes, initially there will be more on there than a monolayer, but since these particles, they are charged, right? So they are not prone to agglomerate because there is this repulsion of charge. So it is quite easy to wash all non-bound particles away. Dip coating is also a possibility. But what is really nice of the spray coating is that you can add a little bit more, so you can push it. So when it dries out, it really occupies all nooks and crannies. So the whole surface is really densely packed. And we found that works best with the spray. And I think also in terms of translation, if you want to go for larger objects, you just need to spray it, right? I think in terms of concentration and the amount that you need, I think you need far less. So that's why we do the spray coating.
Hi. You show us the results against Staphylococcus. So I would like to know if you evaluated the activity against gram-negative bacteria. And I have also another question. Which are the most suitable methods to get the minimum inhibitory concentration of the nanogels?
So basically what you do is you make a suspension and you apply your so different concentrations, and then you plate it, and then you count how many colony forming units there are. Okay. I have to admit, but is Brandon here? No, Brandon is not here, too bad. I have to rely then on my colleagues, either Brandon or Henny, because the specific. So what you do is you plate it out and you make dilutions, and then you count the number of colony forming units. So that is a pretty standard assay. For the flow, right? So that is something that we do quite often. The live dead stain is something that you can do quite easily. It's just adding a stain where bacteria either light up green if they are alive and red when they are dead, and then you can just count them.
Okay.
So in generally, it's difficulty is not so much in the technique, but in making everything really precise, right? So to avoid any errors in pipetting, to avoid any contaminations, to avoid any of these things.
Okay. And did you see activity against gram-negative bacteria?
Sorry?
Did you see activity against gram-negative bacteria?
I don't know.
Oh, okay. Okay.
No, I don't think so. Again, I have to admit. So I think these were gram-positive bacteria that we tried, but I don't think. So usually we go first for the usual suspects. There's the S. aureus, and so I think we tried it. It did work, maybe not as well as the gram-negative.
Okay. Okay. Thank you.
Hi. Thank you for this stunning speech. And my question is related to my college one. Is it possible to change the bioactive molecule that you insert inside the matrix of the gel? And how, if this is true or possible, how you can do it? changing the integration of the positive charge ammonia, or how would be the strategy for this case?
How to change that structure? So what you could envision is that. So what we do now.
Yeah. For example, instead of using triclosan, we can, for example, put some hydrophilic molecule, bioactive molecule.
Yeah.
How it could be possible to integrate to the matrix of the PL.
So for introducing hydrophilic molecules, I would always go for these labile linkers, right? So if so, let me see if I can have triclosan back here somewhere. No, it's not here. Where is my triclosan? Oh yeah, here. So, well, this is a little bit of a. So here you see. So it is a hydrophobic molecule, but it has a handle, right? And that is one of the projects that we're now also doing. Is we want to introduce a hydrophilic antimicrobial,
or changing for such another molecule.
Well, so what you can do is just modify this, right? So this is hydrophobic, but let's assume that we have a hydrophilic molecule and it maybe has an alcohol in there, or an amide, or so these are handles that you can use to modify and add acrylates to it. So it is, for many functional groups, it's not that difficult to add a acrylate group. And then you can just introduce it into your polymer network by adding it in your nanogel system. So I really think that